Hello students, welcome to Vedantu Neet English. I am Miss Gopika.
And I hear... Okay,
Class is not cancelled. Please stop spreading fake news.
Who's telling class is cancelled? We are here already. Good morning, good morning, good morning. So good morning each and every one of you. This is Baswaraj sir. This is Gopika Ma'am. Welcome to Vedantu Neet English students. So good morning each and every one of you.
Yes. So hi students. So first time we are doing a morning class together and we are
Yes.
totally energetic.
Energetic.
Yes, but I hope all of you are also energetic because it's going to be a really helpful sessions. We have seen everywhere that mind maps are sold for some amount Okay, but here we are giving it for complete free and we are going to do your entire 12th syllabus with the help
Entire 12th biology in one video with the help of
mind maps. Okay.
So we'll be doing alternatingly. First chapter ma'am will take, then second chapter I'll be taking. So one after another, every single we'll be doing one by one.
Yeah.
Okay, ma'am?
Yes. I hope we are clearly audible and visible. Okay. I would request all of you to just listen to the class. I do not want any other, unnecessary comments, spams, nothing, students, because it's a very important class. Such a class will not happen again. Okay. And it is always better to watch it live than record it. So shall we start? Yes. Hello. Good morning, all of you. How is your energy? Is it high? Please let me know in the chats. Okay. Yes, we are audible and visible. Okay, come on all of you, let me know in the comments, let me know in the chat if all of you have the energy. Okay, let's start. Okay, before we start the class, all of you mark your attendance because attendance is very important. After that only I will start the class. Okay, I need to see the attendance, then we are going to start the class. Chalo, do it faster. Yes, 1 second students, let me refresh. I'm not able to see your chats. Yes, okay. Yes, okay. So yes, all the students with proper focus be here in this class. I do not want any spam. I do not want hi, bye, anything because this session is very important from Neet, Board, PUC, from all the point of view. Okay, you are going to get the entire mind map also for free. We will share the session like we promised. Yesterday's session also is already shared on the Telegram. The notes also is being shared. Okay, shall we start? Okay, yes. Yeah, I want all of you to smash the like button and let me know the attendance, and also come on, put the yes, and also students, something I wanted to tell you. Okay, let's start. Where is the fire emoji? Where is the fire emoji? I will not start until you put fire emojis. Come on, all of you. And likes also hasn't come. Likes also hasn't come. So much conjuice in the morning, early morning. Okay. So students, first chapter we are going to do is sexual reproduction, flowering plants. We are going to keep it crisp. It's a revision. It is not a explanation. Okay. Shall we start? Okay. Yes. So look here. 1 second. What is happening? Yeah. Okay. Let's start with your first thing, flower structure. Okay. So all of you, is it visible clearly? I think it's visible. Okay. Is it visible? Yes. Okay, very good. Awesome. I don't think a true student should ask the duration, should be bothered about the duration. But before that, mark your attendance. All likes, all attendance quickly. Okay. Yes. Okay, students, look here, all of you. We are going to start the flower structure. So here you can see we have your normal flower, LS of the flower. What in all do we see? We have the anther, filament, petal, stigma, style, pistil, ovary, ovule and sepal. But this is not the catch. The catch is you need to know which is the essential part of the flower and which is the non-essential part of the flower. So if I have to mark the essential part of the flower, stamen, pistil becomes the essential part of the flower. That means which are helping in reproduction. And then what is this non-essential part, ma'am? Non-essential part is the part which does not directly help in the reproduction, but it definitely helps in attracting the insects for, pollination to happen. So they just do the other part of the reproduction. Got it? So the non-essential part. Is it too bright? Is it too bright? 1 second. So little, no, right? It's fine. Okay. Okay, it's fine only, I guess. Okay. So look here. So we learned about the essential and non-essential part. The question can come for all of you, essential and non-essential part. So I hope it is clear. Stamen becomes the essential part, pistil becomes the essential part, or in simple word, andrecium and gynoecium. Okay, that means essential because they help in reproduction. Non-essential means your sepal, petal, your calyx and corolla, which does not directly help in reproduction but is helping in pollination. Okay, this is your first part of the chapter. Now look here. Okay, look here all of you. This part, when you go, here you are able to see the male gametophyte. Okay, the next portion is your male gametophyte. So here, students, this is an exact image from your NCERT. Yesterday also we discussed this. Day before yesterday also we discussed this. That is, when we take a dissection, okay, when we take a section of your anther, we will get some few layers, right? We get few layers. What are those layers? Those layers are very simple. Those layers are nothing but your, how did I tell you to remember it? Okay, we have elephant eats my tomato. That means epidermis, endothelium, middle layer and tapetum. Now, what are these layers actually doing? They are basically helping or protecting the anther, right? Inside the anther, who is there? Male gamete is there. That is the reason what we tell, no, essential part. That means ultimate, students, ultimate of this chapter is we need to form the pollen grains and we need to form the egg. Fusion should happen, fertilization should happen, which should lead to the formation of seed. That's all. That is the ultimate destination of this chapter. For that to happen, what do we need? We need our young anther to develop. For that, we have some layers. Okay, I have made complete notes of what each layer is, that you can definitely check it out later. So look here, we have the first layer, epidermis, then we have the inside layer, endothelium. Epidermis and endothelium are single layers. Single layer. Okay, got it? And look here, middle layer and tapetum. Middle layer and tapetum, out of that, your tapetum is very, very important because this provides the nourishment. Okay, this provides the nourishment. It also helps in synthesizing what? All of you in the chats, it also helps in synthesizing what? Okay, all of you in the chats, quickly. So yes, tapetum, and then you can see inside, okay, in the this entire thing I told you for what? For the protection of your pollen grain. But just like that pollen grain will happen, no, even pollen grain has a mother, right? Even we have a mother, same way. So microspore mother cell will be inside. To protect all this, all these layers are to protect your microspore mother cells. Okay, now look here students, after that, if you come here, you will be able to see the female gametophyte. So all of you know microsporogenesis means your microspore mother cells will undergo meiotic division, form your microspore tetras. They will be attached to each other, and that is called with the help of callase, we will separate them. Right? After that, they develop slowly and they form the pollen grain. Okay, very good, very good. Not only callase, they also form sporopollenin. Okay, sporopollenin. Yes, good job, Sanvi. Rinin, okay, yes, Vaishnavi, good job. So sporopollen, Mega, Drishti, those who are new to this channel, do not forget to subscribe to the channel and do not forget to hit the like button, because that's the only way we feel loved. That is the only way we get to know that we were able to help a lot of students. Okay, so do not be hesitant, students. It's only one like. We are not asking you to pay anything, because you know mind maps are sold for some amount in most of the places, but we are giving it for free. Okay, yes, look here, after understanding the male gametophyte, okay, your entire microsporogenesis, megasporogenesis, microsporogenesis is simple, we form the pollen grains. Okay, you have your vegetative cell, generative cell, exine, intine, all that will be formed. Now we'll come to the female gametophyte. Okay, look here students, female gametophyte has who and all? All of you tell me this, the simplest female gametophyte, what is there in the female gametophyte? Simple ma'am, stigma style ovary, right? Stigma style ovary. Inside the ovary, who is there? Ovule is there. The ovule that we have, or the ovule that you have to study, okay, is your anatropous ovule. Okay, anatropous ovule. Now, I have given a trick to remember these two examples. Students, they can ask you the question how to give me an example of apocarpus and syncarpus. Very easy. What is this? Is it syncarpus or apocarpus? This is syncarpus. MSP and MAM. Okay, this, what is MSP? Again, your multicarpillary syncarpus papaver somniferum, which you studied in your morphine, okay, opioids. Here you see MAM. This is what multicarpillary apocarpus michelia, This is how you'll remember this example. If this image is given, you're going to write the example. One is Papaver somniferum, one is Michelia. All my students know this? Yes or no? Okay, got it. Anatropous ovule means what, students? The funicle will not come in straight line with the micropyle and the chalazal end. Okay, here, what you need to remember? You need to remember first is the funicle, the stalk. Okay, first you need to remember the stalk. Then you have to remember the hilum. Okay, the attachment point. The attachment point. Very good, this T. Okay, very good. Yes, you have to remember the attachment point. Then, there is a portion, okay, there is a portion where the integuments are not present. You'll be like, what are integuments? Integuments are nothing but protective layer. Because inside that, who is growing? This is the female gametophyte. Who is growing? Egg is growing. So, this outer and integuments will protect the embryo sac. And the portion where the integuments are not present is called the micropyle. Okay. Now, that end is called micropylar end. The one that is opposite to the micropylar end is called the chalazal end. All my students know this? Yes or no? Now, after the embryo sac and the integument, there are some layers which provides nourishment, which is called the nucellus. Okay, and this is all about your female gametophyte. And students, anatropous ovule is very, very, very important. Okay, very important. Fine, got it? Are we clear on this? Yes. Now we understood pre-fertilization events. Now we have to understand for post-fertilization event, or for fertilization to happen, we need pollination to happen. Here, who is going to help? Your non-essential parts are going to help. So, all of you look here, there are 3 types of pollination mentioned in your NCERT. That is nothing but your autogamy, gametogamy and xenogamy. Autogamy means self-pollination, simple. Okay, gametogamy means on the same plant, okay, on the same plant there's one flower and one more flower. These two gets pollinated. That means from one flower to another flower of the same plant. Okay, that is called as gametogamy. Clear or not? Are we good to go? Then xenogamy means simple, cross-pollination. Okay, cross-pollination. Here you have under your this paragraph only you will have clistogamous and chasmogamous flower, right? That also you have to remember. Clistogamous always closed. Okay, chasmogamous exposed. Okay, clistogamous, chasmogamous flower. Now, agencies, that means who are the people who are going to bring this male gamete all the way to the egg. Simple. Abiotic factors are wind and water, and biotic factors are animals and humans. Okay, we walk around, it touches our dresses, and then we scatter it somewhere. So these things. Now, examples. Students, examples also is given in this mind map. You do not have to go anywhere. Don't take any tension. Okay. Yes. Wind pollinated is your, again, grass. Water pollinated, very, very important example is your Vallisneria and Hydrilla. Okay. Sometimes they have also given Vallisneria's image and asked. Okay. Now, insect pollinated is Yucca. Okay, insect pollinated is Yucca. Again, they show a kind of mutualism. They help each other. Okay, the moth will lay eggs in this Yucca flowers in the ovary, and then they help each other. So that's why Yucca also is very important to remember. Any doubts so far? Any doubts so far? All of you, let me know in the chat box. Yes, correct. Entomophily, zoophily. Okay, students, any doubts so far? Fast, fast, tell me. No, right? Yes? Any doubts? No, right? Okay, fine. Now, look here. Next one is devices for cross-pollination. So, first we understood the flower where all this essential part is there. Then we understood the male and the female gametophyte. Then we understood how to bring these both together. Now we are going to understand how to do cross-pollination, because self-pollination leads, continuous self-pollination leads to inbreeding depression. So, we always prefer doing cross-pollination. Now, for cross-pollination to happen, what are the things that we need to keep in mind? So, look here, devices for promoting cross-pollination, or in other words, to avoid inbreeding depression. To avoid inbreeding depression. Got it? To avoid inbreeding depression. First point is simple, students, self-incompatibility. That means the pollen-pistil interaction will get stopped, or the self-plant, like imagine the stigma will start rejecting its own pollen grains. That's all. Self-incompatibility. Okay? Self incompatibility. Gaitonogamy is nothing but, look here, this is one plant, this is one flower, okay, this is another flower. From here to here, transfer happens. That kind of a condition is called gaitonogamy. That means in the same plant, two flowers, okay, different flowers are crossing, okay. The pollen grains from this flower will fall on the stigma of this flower. This is called as gaitonogamy. Is it clear? You have put a card name, I don't know why. Don't you have a good name? Yes. Now, look here. Devices for promoting cross-pollination. First thing is self-incompatibility. Arrangement of anther and stigma at different position. Always we know if the stigma is in the lower position and anther is above, easily the pollen grains will fall on this. Easily the pollen grains will fall on this. Now what we are going to do? We are going to make the stigma longer. If the stigma is longer and the anther is smaller, then how will it happen? Right? Simple. Okay, simple. So we'll change the position. Next one. Production of unisexual flowers. That means forming only one flower, like one with the male part or one with the female part, right? Your staminate, okay, staminate and your staminate flowers. So what basically, that means any one of them, the male part will be there or the female part will be there, okay, got it? Yes or no? Fine. Now, pollen releases and stigma's receptivity not synchronized. That means time delay will happen. Time delay will happen. This way pollen-pistil interaction will not happen. That means one will mature later, one will mature early. If both matures at the same time only, this kind of a pollen-pistil interaction will happen. But if one person is maturing late, another person is maturing at different time, when we change the maturity time or the different timing, no, that time, what is going to happen? This delay or this kind of a delay we can cause. This is to avoid inbreeding depression. Okay, this is to avoid inbreeding depression. Now, cross-pollination happens, students. Now, let's go to pollen-pistil interaction. One of the most important thing is your pollen-pistil interaction. Now, for pollen-pistil interaction means what? We need to take this male gamete all the way to the embryo sac and we have to allow it to fuse with the egg. So, I had told a story in my class about the marriage, proposal and all that. You can check that video for understanding this in a better way. Now, students, simple. Stigma has accepted the pollen grain. Stigma will be like, yes, okay, fine, we are good to go. Let's proceed with the proposal. So, that means after the chemical interaction, they will be ready to go further. After the chemical interaction, they'll be ready to go further. Normally, two families are talking. After the conversation, what they'll be like, okay, we can proceed with the marriage. They say like that, no? Same thing is going to happen here. And now, because of that, what will happen? The pollen tube will grow all the way and with the help of filiform apparatus, very important. With the help of filiform apparatus, the pollen tube will be allowed inside to go and fuse where? To go and fuse where? To the egg. Okay, yes, perfect, Pranav, chemical dialogue. Okay, chemical dialogue. Got it? Yes or no? Yes. So, after that, what happened, students? Now, like family spoke, everything happened. Even the guy was allowed to go inside and meet the, bride and all, the groom and bride met. So, after that, fusion has to happen, right? Fusion has to happen. So, pollen-pistil interaction will only happen if they reach the right compatibility. Will they marry wrong people? No, we will not marry wrong people, no. We will always try to understand their character, their background, everything. We will marry the right people. Only when right compatibility happen, the pollen-pistil interaction will happen. So, the question can come in assertion and reasoning in a different pattern. Okay, different pattern. So, keep that in mind. Okay. Yes, filiform apparatus will guide the pollen tube to enter all the way towards the egg. So, all of you know your embryo sac, we have your egg apparatus, we have your antipodal cells. Egg apparatus have who and all? We have their synergids and egg. Antipodal cells are there on the top. In the between, we have a central cell or your polar nuclei. Now, one problem has happened. The problem is that with this doctor guy, one more bank manager came, his friend. His friend is like, no, I think even I should get married because I'm so jealous about you, even I want to get married. At that situation, what is going to happen, students? Okay, at that situation, what is going to happen? Your double fertilization will happen. That means, we were supposed to do only one marriage. We were only planning about one marriage, but two marriages happened. Okay, elder sister and younger sister got married because with this doctor fellow, his friend also came. So, that is called as double fertilization. That means, first your syngamy happens, that is the actual marriage, the male plus the egg, actual marriage. Triple fusion happened. This is the bank manager with the sister. So, that means again male gamete plus your polar nuclei or your central cell, okay, to form your endosperm. to form your endosperm. Got it? That is why we call it double fertilization, because when we decided for one marriage, 2 marriages happened, so double fertilization. Got it or not? Okay, got it or not? So, double fertilization is a very favorite question of board people, and NEET also, they won't ask directly like that, but they'll ask you the number. Triple fusion students, what is going to happen? It's going to be 3n, and this is going to be 2n, and your antipodal cells are going to be n. So, the question can come like that. Like yesterday we discussed the question. Okay, got it? At least tell understood or not, students. What is this? Sleeping, huh? Early morning you're sleeping. Okay. Now look here, all of you. Now fusion has happened. We are waiting for the zygote formation. That means after marriage, we are waiting for your other events to happen, right? So during that, what is going to happen? The synergids are going to degenerate, antipodal cells are going to degenerate, and then we will start having the primary endosperm cell and primary endosperm nucleus. Okay, the nucleus, that is pen and peck. Pen and peck. Easy to remember. Pen, peck, pen, peck. Okay, one is the nucleus, one is the cell. Cell is different, nucleus is different. Okay, and all of you tell me how much is your embryo sac? It is 7-celled, 8-nucleated. Okay, 7-celled, 8-nucleated embryo sac. Now students, look here. Okay, now after double fertilization, what has happened? We have our endosperm. We have our endosperm, right? That is your, what is that? Pen, or your, after the pen develops or matures, it becomes your endosperm. Now what are we waiting, students? Simple thing, okay? If endosperm is present, that means completely endosperm is not utilized. Endosperm is for nourishment, but all the endosperm is not utilized. That kind of a seed is called albuminous seed. That kind of a seed is called albuminous seed. Clear or not? Okay. Peck, pen, clear. Peck, pen, clear. Okay. Albuminous seed. Then we have, imagine the endosperm which was dead. Okay, endosperm which is dead had to completely, the endosperm got completely utilized by the growing seed or the growing embryo or the growing zygote. That time the endosperm is not there in the seed. That kind of a seed is called non-albuminous seed. So if endosperm is present or it is endospermic, it is called albuminous. No endosperm is there, non-albuminous. Easy to remember. Easy to remember. Okay. Yes, examples also are given in your textbook, right? Now, so students, now that happened, now we are waiting for the development of your embryo. That means what? Your dicot and monocot seed. Your dicot and monocot seed. Chichu, chichu. Dicot and monocot seed. So it is very easy. Dicot and monocot. Yes. Okay. So look here. Your dicot and monocot, students. Why is this not going? Yeah. Dicot embryo and monocot embryo. So what do we have? Properly in morphology, you would have studied this, your scutellum, aileron layer, your coleorhiza, coleoptile, all this you would have studied there. But here you just have to identify, okay? In case this diagram is given and if they ask you to label, you should be in a position to label. Okay, you should be in a position to label. So here, students, you will have your scutellum, which actually makes that cotyledon, that first, what is that? Cotyledon's proper differentiation will be starting from your scutellum. Okay, after scutellum, you can see coleoptile. Okay, that is basically to protect your shoot. To protect your shoot or to protect your plumule, you have your coleoptile. How do I remember? In coleoptile, there is a P. That P and plumule, please connect it. Okay, and then we have coleorhiza. Okay, rhiza. R is there. Root. To protect the root or to protect your radicle, there is coleorhiza. Then students, who is there? Now dicot. Dicot, what is there, students? Here you will see plumule, cotyledons. 2 cotyledons will be there and you will have your radicle and root cap. That's all. Root cap is basically to protect because it has to go all the way to the soil. Clear? Now what has happened, students? Imagine normal post-fertilization events had happened, but the farmer is not very happy. Farmer feels like, maybe I want the variety that, I want such a different type of variety of flowers, fruits or whatsoever. That time, the farmer will decide to do something called as artificial hybridization. That means to make his own desired flower, fruit or anything, he will try to artificially do self-pollination. So what is that? What do I mean by that? Okay. I basically mean that he is going to do emasculation and bagging. He is going to do emasculation and bagging. What does that mean? It is one of the major approaches of crop improvement by using desired pollen grain. Desired pollen grain will be put, and it will, or so what they will do? They will basically remove the anther. They'll remove the anther and they will bag it. They'll cover it and keep it until they want to put a desired one. All they do, or some kind of changes they will do, so they get the desired one. Next thing is they do bagging. It is a dynamic process which involves, this won't move, which involves pollen recognition followed by pollen tube growth. Okay, pollen tube growth. Again, so that two are emasculation and bagging. Emasculation is basically removing the stamens, and bagging is you cover it with a butter paper. Butter cord, some paper they have mentioned today, butter paper, I guess. You can use even the polythene bags. I have also seen people using polythene bags. Yeah, even in experiments, usually people use polythene bags because otherwise it's too difficult to find this butter paper or whatever paper it is. Okay. Then students, now embryo formation, everything we understood. Now last step is to see the type of fruits. Okay, to see the types of fruit. Look here, fruits are of two types, your true fruit and false fruit. True fruit means what? Yes. Okay, true fruit means what? Sound is okay, no? Yeah, butter paper only, no? Yes, I'm correct. Okay, yes, no emasculation is required in unisexual flower students. That is a condition what they were telling, no? If unisexual flowers are there, then inbreeding depression is avoided. So that way also this person can always change and do any kind of thing. But in flowers or plants which have continuously planned to do self-pollination, we have to do some kind of artificial hybridization. Now look here. When the fruit develops from the ovary, that means what it has to actually do. You are a NEET aspirant, you should prepare for NEET. In between, if you go to prepare for JE, you become a false fruit. Okay, you should be a true fruit. What? So, when a fruit develops from ovary, that is mango, example is mango, that is called as your true fruit. And false fruit is what? When parts of other than ovary, from wherever the fruit is arising, like apple is from the thalamus. Can I call it a true fruit? No, I should call it a false fruit. Clear? Example apple, that is from thalamus. Ho gaya. Done. Now, some some important terms are given in your textbook. So let's do that terms. First is parthenocarpy. That means development of fruit without fertilization. Okay, without actual fertilization, the entire fruit will be developed from only one part, maybe say from the female. That is called as parthenocarpy. Okay, parthenocarpic fruit. What is the example? All of you tell me. I have taught you the example. Parthenocarpic fruit, which is the one. Then polyembryony. That means what? Occurrence of more than one embryo in a seed. Usually one embryo is what we decide because, yeah, one embryo is what we decide. But then what happens? Multiple embryos will be formed. Okay, multiple embryos will be formed. All of you give me the example. Example. Very good. Banana. Awesome. This is the example of, your parthenocarpy example is banana. This is orange. I think orange is not given in your NCERT. No. Apomixis. Production of seed without fertilization. Every single student confuses parthenocarpy and apomixis. Students, one is development of fruit without fertilization. Development of seed without fertilization. Do you understand the difference? Do you eat the seed or the fruit? You eat the fruit. So fruit, when it is developed, that is called parthenocarpy. Seed, when it is developed without fertilization, is called apomixis. Is this clear? Is this clear? Yes, citrus, lemon. Yeah, citrus they have mentioned. Not exactly orange they have mentioned, only citrus. So all the citrus fruits, okay? Lemon, orange, everything. Did we do everything? Okay. Now, significance of seed dispersal and formation is not. Seed dispersal help the species to colonize in other areas, and many fruits have mechanism. No, nothing important. Did we do everything? Outbreeding devices we did. Students, yes. So I told you to avoid inbreeding depression, those devices are called as outbreeding devices. Okay. Did we do everything? Check, all of you check once again with your NCERT. I think yes, we did everything. Seed dispersal is only. No. The insect, just this is there, the agents. Students, mesocarp, endocarp, you know that coconut, your drupe fruits, okay? So that you have to know. Which one are we consuming, which we are not consuming. So coconut and your mango, Please remember that image in your head, okay? Are we eating the mesocarp? Are we eating the endocarp in both of these fruits, okay? Yes or no? Fine? Yes, students, your development, yesterday I have already done your globular heart-shaped embryo, globular shaped, heart-shaped, mature embryo I have already done yesterday. So that is not here. That is not here, okay? Done? Diagrams I have already done in another session, so I am not doing it again, okay?
Ma'am, all this banana and fruit and everything, actually remember I was telling you other day that strawberries, raspberries and
They're not actual
fruits. Yes. Students, no, they're fruits but they're not berry.
Yeah, they're not.
So do you know, students, I'll tell them the fact also today.
Yeah.
I wanted to make a short on this but I never made. But students, do you know that strawberries, raspberry, all of these are not actually berries. You know what is a berry, students? Berry is a type of fruit which develops from your a superior ovary, which is multicarpillary and syncarpous. Those are actually not berries. Your raspberry and strawberry, they are not berries at all. Do you know what is the example of berry actually? The example of berry was very funny, which you'll start laughing at this. The example for berry is banana. Banana is the example for berry actually. So with that being said, with that amazing fact in botany, we will start zoology. So can we start, students? Can we start next chapter? Can we start human reproduction? Can we? Show some energy in the chat, students. We will be starting human reproduction now. Yes, good morning, good morning, good morning. How was the fact? How many of you knew that actually? How many of you knew that fact that banana is a berry and not a, other type of fruit which we would have considered? Banana is a type of berry. Okay. So can we start now? Sir, parthenocarpic and parthenogenesis. Parthenogenesis is your honeybee, which we learnt in your principles of inheritance, right? That is parthenogenesis. Parthenocarpy is for fruits. Okay? Yes? Yes, amazing. So let's start with the human reproduction. Now quickly, we'll start with the mind map. Yes? Show some energy in the chat because early morning, everyone should be energetic. So students, first under human reproduction. Under human reproduction, we'll be focusing on the male reproductive system now. Yes, the entire of your male reproductive system is divided into how many parts? Into two parts. One is called as your primary sex organ and the second one is called as your secondary sex organ. Now what is this primary sex organ? The primary sex organ here is the testis. Testis is the primary sex organ here. Now you might be asking, sir, why is it called a primary sex organ? Because it is able to produce the gamete and also it is able to produce a sex hormone. The gamete here is the sperm and the hormone here is the, let me know in the chat, that's going to be a type of androgen, right? Then we have the testis. Now students, tell me in the chat, testis is made up of around 250 small compartment called as your testicular lobules. So testis is made up of what? Testis is made up of around 250 testicular lobules. Now inside each testicular lobules, everyone, inside each testicular lobules, there are 1 to 3 highly coiled structure called as your seminiferous tubule. Around 2 to 3 highly coiled structure called as seminiferous tubule. Now there was a question before. What do you find inside the seminiferous tubule? Yes. So inside the seminiferous tubule, it is lined by two type of cells. Can anyone in the chat tell me which are the two type of cells inside the seminiferous tubule? The two type of cells inside the seminiferous tubule are, number one is your Sertoli cell. Number one is the Sertoli cell. Now the second one is the spermatogonia. The spermatogonia also called as what? Male germ cell. Male germ cell and Sertoli cell. The male germ cell or spermatogonia undergoes your mitosis and meiosis, which later develops into sperm. Why do I say mitosis and meiosis? Because remember, the spermatogonia, starting it undergoes mitosis to produce the primary spermatocyte. Later on it undergoes meiosis. Remember that point? I will be telling you that once again. Then we have Sertoli cell, which is also called as your nurse cell, which is involved in the spermiogenesis, not spermatogenesis, spermiogenesis. Then we have Leydig cell. Leydig cell is also called as what? It is also called as interstitial cell, which is present in the interstitial space. What does it produce? It produces the androgen. So Leydig cell produces what? Androgen. Done. Then we have, that was your primary sex organ. One branch is done. The second branch, that is your secondary sex organ. Secondary sex organ mainly includes ducts and glands. What does it include, students? It includes ducts as well as glands. Now, what are the ducts here? If you notice, the main ducts are your rete testis, vasa efferentia, epididymis, and vas deferens, as well as vas deferens. Now, quickly tell me in the chat. Quickly tell me in the chat, if I tie the vas deferens, right? What is it called as? If I cut and tie the vas deferens, what is it called as? Tell me in the chat. Then we have the different glands. The different glands which are present here are the seminal vesicle gland, which is present as a pair. Yes, that is important. Present as a pair. Then we have prostate gland, which is present as a single entity. Then we also have the bulbourethral gland. Bulbourethral gland is also called as the Cowper gland, right? It is also called as what? Cowper gland. All of you know that. Now tell me quickly in the chat, what is the function of seminal vesicle? Seminal vesicle gland is mainly involved in the releasing of certain enzymes as well as certain amount of fructose, because it provides nutrition for the sperm, right? Then we have prostate gland, which mainly involves in releasing of certain calcium ions, also involved in the releasing of calcium ions, which increase the motility of the sperm. What does it increase? It increases the motility of the sperm. Done. This is also a question. It's a NEET PYQ and it has been asked in your board examination repeatedly. So students know what is the functions of different glands. Okay? Different glands. Now, that was your axillary glands. Any doubt here, students? I believe there should be no doubt here. Diagram is important. They might ask you to draw the diagram, but very rarely they'll ask you, but not much. But you need to know what is primary sex organs and secondary sex organs. Clear? Now students, let's go to the female. Where is it? Let's go to the female reproductive system now. Again, the entire female reproductive system is divided into two parts, right? The entire female reproductive system is again divided into two parts. The first part is what? The first part is called as your primary sex organs again. And the primary sex organs are the ovaries. The primary sex organs are called as what? Ovaries here. See here. The primary sex organs are called as ovaries. 1 second, students. No, why is this happening? 1 second. I don't know why did this start coming up? Yeah, go on. Yes, we are back. I think I clicked on the subtitles. Subtitles started coming. Now, the primary sex organs are the ovaries. Now, again, tell me in the chat, ovaries are responsible for two things. The first one is going to be production of gametes. That is, in the case here is, so what is the function of bulbourethral gland? Very good question. Bulbourethral gland is mainly involved in the secretion of lubricants. During the intercourse, the lubrication is provided by bulbourethral gland. Very good question. Students, ask your doubts. If you have any doubts anywhere, we will take up your doubts. And if we miss out any doubt, we will address it at the end of the video. Or let us know in the comment section if there is any doubt, we will resolve it. We are here for you. Okay? Now, students, the ovaries has main two functions. I told you. That is, the first one is releasing of gametes, that is ovary. And the second one is the releasing of the sex hormones. The sex hormones are here, are the estrogen as well as progesterone are the sex hormones here. Remember that. Now, the entire ovary is divided into two layers. If you look at the structure of ovary, it has an outer structure. If you look at the structure of ovary, the outer part is called as epithelium. Yes, it is called as epithelium. And the inner part is called as stroma. It is called as what? Stroma. Stroma. Now, students, this stroma again can be divided into the outer cortex here. Peripheral cortex or outer cortex. Peripheral cortex. And inside part, can you see inside part? This inside part is called as inner medulla. The inner part is called as what? Inner medulla. Clear? Now, students, then we have the axillary ducts again here. The axillary ducts mainly includes oviduct, which is also called as fallopian tube. Yes. Then we have the uterus, which is present as a single entity. which is a inverted pear shape, correct? Then we have the vagina, which is the external opening. Vagina is the external opening, not the external genitalia. Remember that. Then we have the oviduct. What are the different parts of oviduct? What are the different parts of oviduct? The outermost region or the region which is closer to the ovary, NCERT line. The region which is closer to the ovary is called as infundibulum, and the infundibulum has a finger-like structure called as fimbriae. All of you know that, which helps in picking up the egg. Then we have ampulla region, which is a much more wider region in which fertilization takes place. Important lines, every single line which I am speaking right now can be asked as a separate question, students. Remember that. Ampulla region is the region where the fertilization takes place. Then we have the isthmus region, which is a much more narrow region. Yes, it's much more narrow region. Clear, all of you? Clear? Yes, students, all of you are really amazing because the chat is on fire today. Yes. Now, students, then we come to the external genitalia. That is going to be the okay. Then we have the external genitalia, that's your mons pubis, labia majora, labia minora, hymen and clitoris. Now here, clitoris is the structure which is made up of many nerve endings. Clitoris is made up of many nerve endings, and clitoris also has erectile tissue, which is a hidden line. It has erectile tissues. Then we have the hymen layer. Lot of myths, students. Lot of myth is regarding hymen layer. If hymen layer is broken, does it indicate sexual experience? No, because there are many instances in which a female can undergo, when she's undergoing extreme sports, horseback riding or a sudden fall, the hymen layer can actually break that time. So hymen layer does not indicate sexual experience. Remove that point. Then we have the mammary glands. Mammary glands has a small mammary glands has something called as lactiferous duct, which has alveoli. Alveoli is a site for production of milk and also storage of milk. Very important line. It is very important line. Yes, students, the entire PDF will be given to you on the Telegram channel for free. So don't worry, do not have to buy any book. We'll be giving it for free on the Telegram channel. Okay. Then we have, now let's understand spermatogenesis. Can we start? Students, I'll briefly explain you spermatogenesis. So listen to me very carefully. So under gametogenesis, there are two main parts. That is spermatogenesis as well as oogenesis. Now quickly tell me in the chat, when does the spermatogenesis start? Tell me in the chat quickly. Students, remember the spermatogenesis starts at the puberty. Spermatogenesis starts at the puberty with increased levels of GnRH, not testosterone. It is a NEET PYQ, important for board also. Increased levels of GnRH. Which starts the spermatogenesis. Now students, spermatogenesis starts at the puberty. Increased levels of GnRH. What is the first step? First step is going to be mitosis. Because of mitosis, the particular male germ cell, also called as your spermatogonia, also called as male germ cell, undergoes mitosis and it increases in number. First point, what? It increases in number, not division. In first step, it is not division. It is increasing in number. Can you see? It is increasing in number. Now, next step. Now the primary spermatocyte, when it increases in number, we call it primary spermatocyte. Now remember, primary spermatocyte is still diploid in nature. It is not haploid because it has not undergone meiosis yet. Remember that point. NEET PYQ again. Now this primary spermatocyte undergoes the first meiotic division, which results in the formation of secondary spermatocyte. Now tell me in the chat, secondary spermatocyte, is it haploid or diploid in nature? Second spermatocyte is haploid in nature, which undergoes second meiotic division to produce the spermatids. Produces the spermatids. Clear? Now spermatids, do they look like sperm here? Do they look like sperm? No sir, they do not look like sperm. They look like some gulab jamun. Yes, they don't look like sperm here. Now this spermatid undergoes differentiation now. It undergoes what? It undergoes differentiation, and that process is called as spermiogenesis. It's called as what now? Spermiogenesis. To produce the spermatozoa. Finally produces the spermatozoa. And the spermatozoa head is embedded in the Sertoli cell. Now release of the sperm from the seminiferous tubule is called as spermiation. Last point. So remember the difference between spermatogenesis, spermiogenesis and spermiation. All three difference you should know for your board examination as well as a NEET examination. Remember that. Okay, done. Quick revision, done. Then we have oogenesis. Now tell me quickly in the chat, oogenesis starts. I will explain oogenesis in 1 minute. Listen to my words. In 1 minute. Oogenesis under 1 minute. Listen to me. 3, 2, 1, go. Oogenesis starts at the embryonic stage. Point number 1. In the embryonic stage, there are millions of oogonia. Imagine, millions of oogonia. Now, one such oogonia starts to undergo differentiation, and that produces the primary oocyte. So, one oogonia undergoes differentiation to produce the primary oocyte. Now, primary oocyte is said to be diploid in nature. Yes, it is diploid in nature. The primary oocyte undergoes meiosis 1. But does the meiosis 1 get completed? No. In fact, it gets arrested at the prophase 1 diploid stage. Arresting is happening. Clear? Then, now once the female child reaches puberty, meiosis 1 is completed, which results in the formation of secondary oocyte. Secondary oocyte is haploid in nature after meiosis 1. When? During puberty. Done. Then students, now this secondary oocyte undergoes meiosis 2, because we know meiosis 1 and meiosis 2 stages are there. So this secondary oocyte undergoes meiosis 2. When? But does it finish meiosis 2? It does not finish meiosis 2. Meiosis 2 is completed during fertilization. When the sperm comes in contact with the egg, meiosis 2 is over. And that is during the fertilization, which results in the formation of ovum and second polar body. Ovum and second polar body. Secondary oocyte and first polar body are produced together. The first polar body is produced inside the ovary. Very important point. And the second polar body is produced in the fallopian tube. Fallopian tube. Remember that. Fallopian tube. So entire done. Oogenesis done. Now, menstrual cycle. Students, I don't think I'll be explaining the entire menstrual cycle because it's going to take so much of time. But one thing which you need to note here is the hormones involved. There are two types of events which are happening here. What are two events? One event is happening in the uterus. One event is happening in the ovary. The event which is happening in the ovary is influencing the event in the uterus. That is very important. That one event is influencing the other event. Now, what are the hormones here? The first hormone is increased levels of FSH. Can you see your diagram? Increased levels of FSH, which stimulates development of follicle. Follicle will do what? Increase levels of estrogen. Will increase the level of estrogen. Clear? Now students, then we have and estrogen will do what? Estrogen will help in development of endometrium layer. Estrogen is involved in what? Development of the endometrium layer. Done. Then we have LH. LH, clear? Now we have LH. LH will increase here on the 14th day, called as LH surge. LH surge. Then we have the LH is increasing here. Now LH is influencing the maintaining of corpus luteum. Now where does the corpus luteum come from? Corpus luteum basically came from the Graafian follicle. When the Graafian follicle here, can you see mature follicle, when it ruptures open, it releases the egg. That is called as your ovulation. But whatever the leftover part, can you see the leftover part? The leftover part gets converted to corpus luteum. Corpus luteum increases the level of progesterone. Can you see? Progesterone level is increased. Now progesterone will do what? Progesterone will maintain the endometrium layer. Formation of endometrium layer, estrogen. Maintaining the endometrium layer, progesterone. Difference is very important. Clear? Then once the LH level goes down, FSH level goes down. Why? If there is no fertilization, the level goes down. Now if the level is going down, what happens? Can someone maintain the corpus luteum now? No. No one is maintaining the corpus luteum. If no one is maintaining the corpus luteum, can it produce progesterone? No. So endometrium layer starts to break after this. Endometrium layer starts to break after that. That is your entire menstrual cycle. Done. The next is the, can you see? The structure of the sperm. Again, structure of sperm is very important, divided into head, neck, middle piece and tail. Head region has a cap-like structure called as what? Acrosome, which has a lot of enzymes which is involved in the fertilization. So acrosome helps basically in the fertilization. Clear? Okay, used in the fertilization. Now students, now one question can come in your examination. Why does only one sperm enters the egg? That question can come, right? Why does the one sperm enter the egg? Because as soon as the first sperm here is entering, or as soon as the head of the sperm is touching the plasma membrane, the plasma membrane of the egg gets completely depolarized. Now once the plasma membrane gets depolarized, no more sperm can enter inside. And which leads to the fertilization. Clear? Now once fertilization, what happens? Once the sperm and the egg are meeting, which results in the formation of zygote. Now this zygote starts to undergo cleavage. Now what is the meaning of cleavage? The meaning of cleavage here is the particular zygote is undergoing mitotic division. The zygote is undergoing mitotic division now. That is, the zygote which is unicellular. Starts to undergo division. So 2-cell stage, 4-cell stage, 6-cell, 8-cell. 8 to 16 cell stage is called as what? Morula. Yes. Now later on it gets converted to blastocyst. The zygote gets converted to blastocyst. Now what is this blastocyst? Blastocyst has mainly two parts. If you look at the structure of blastocyst, it has divided into two parts. If this is the blastocyst, it has two parts. The outer part is called as what? Trophoblast cells. And the inner part is called as inner cell mass. This is inner cell mass, and the outside part is called as what? Trophoblast. The outside part is called as what? Trophoblast. All of you are amazing. All of you know this. Now students, this trophoblast will develop, this trophoblast will undergo implantation. Now once implantation happens, listen to my words. Now once implantation is over, on the trophoblast cells there will be something called as chorionic villi. Now what is this chorionic villi? Now chorionic villi plus maternal blood plus uterine tissue. So uterine tissue of the endometrium layer, uterine tissue and the chorionic villi will get what? Interdigitated, and that will result in the formation of placenta. So this trophoblast cell plus uterine tissue will develop into placenta. Very important line. If they ask you the development of placenta, very important line. Now what happens to this inner cell mass? Inner cell mass gets converted to the embryo. Very important point. Done. The last part here is the functions of placenta. Sir, what are the functions of placenta? Students, all of you know, placenta acts as a temporary endocrine tissue. All of you know that. It is a temporary endocrine tissue. The question will come, why? Why is it called as a temporary endocrine tissue? The answer is very simple, students. The answer is very simple. Tell me in the chat quickly. Fire in the chat, all of you. It's called as temporary endocrine tissue because placenta is able to produce hormones. The example is HCG, that is human chorionic gonadotropin. The other one is HPL, just like the IPL. What is HPL? Human placental lactogen. Human placental lactogen will produce a type of carbohydrates which is essential for the development of embryo. Yes. Then we also have estrogen and progesterone. Now there was a NEET question and also a board question. Which are the hormones? A NEET question and also a board question. Which are the hormones released only during pregnancy? Question is important. Which hormones are released only during pregnancy? The answer should be HCG. The answer should be your HCG. First one, HCG. Second one is HPL and third one is relaxin. Third one is relaxin. Done. Now what is the function of placenta? The function of placenta is very simple. The function of placenta is very simple. It will help in exchange of nutrients. See, imagine you are a baby inside the mother's womb. Will you need food? Will you eat food? Yes. Will you need oxygen inside the mother's womb? Yes. Once you eat food and take in oxygen, will you release out waste? Yes, sir, will release out waste. So all this taking of oxygen, giving food to the fetus, removing of the external waste, everything is facilitated with the help of placenta. That is the importance of placenta. Important board question and NEET question as well. Now, the next step is parturition. Now, the baby is ready, fully developed, fully ready, bindas to be sitting there. It is just wearing its glass and it's like, okay, I am ready to get out from here now. I am ready to move out of my womb. Where will the first signal come from? The first signal for the parturition actually comes from the baby itself and also the placenta. It's called as fetal ejection reflex. Very important question for NEET as well as board. What is it? Fetal ejection reflex. And the delivery of the baby. And the delivery of the baby is called as your giving birth to the young one is called as parturition. Parturition is nothing but giving birth to the baby. Clear? Every single point of NCERT I am covering, students. Every single point of human reproduction is getting, jogged in your mind. So every single point it said, one question will definitely come in your board examination. That is my guarantee from whatever I've told. Then we have finally the lactation. The last point of this chapter is going to be lactation. What is lactation? Now once the baby is born, it needs protection. Yes. It is in the outside environment. It doesn't know what kind of bacteria might be there. Yes. The first protection is given to the baby in the form of colostrum, which is the breast milk. And it is very important because it is rich in which antibody? That is your A. Clear? That is your lactation. Lactation is production of milk towards the end of the pregnancy. Students, did you notice that? We covered the entire chapter in less than 30 minutes, right? Yeah. Less than 30 minutes, the entire chapter revision is done. And this is how we'll be doing every single chapter. Detailed explanation. Content, best content. Yes. So with that being said, I will hand over the mic to Gopika Ma'am, who will be doing the next chapter. That's going to be your reproductive health. You'll be happy when she this. Yeah. You can take up. Reproductive health.
How was the Josh, yeah?
Josh is a little going down, ma'am.
Going down?
A little bit going down. So much of.
Drop, no, now? Students, questions are not only going to come from human reproduction, questions are going to come from all the chapters, okay? So do not leave the class as soon as you have seen human reproduction. Human reproduction is not examiner's favorite chapter, even though it is your favorite chapter, okay? Chalo, let's start. Reproductive health, energy is there, okay? Yes. Please don't spam. Please don't spam. Okay. Okay, fine. Look here, students. So, reproductive health. What is reproductive health? So, according to WHO, reproduction means or reproductive health means that a total well-being, not only just physical, mental well-being. Again, in human health, this topic will be, pressurized or concerned. This topic is all about that we'll be talking. So, human health, okay, human health and diseases, here you should know, According to WHO, reproductive health means a total well-being in all aspects of the reproduction, because you know that when a couple is not able to conceive, when they are not able to have a baby, they go through a lot of roller coaster of emotions, right? They go through a lot of depression, they go through a lot of things, okay? And this lot of things is not something that they're happy about, because so many emotions, students, at that point, like, you know, societal questions. So that's why they're saying, according to WHO, reproductive health means the total well-being, okay? Total well-being. Amazing, amazing. Okay, Josh is very high. Awesome. Now, even though they're talking about reproductive health as you know, adolescents, as teenagers, it is very important for you to know, because this is your age where you feel like, I need to have the freedom, I need to have the, you know, I need to be cool in front of my friends. So you people try out a lot of things, you people do many things out of, you know, that moment of curiosity. So that is why this chapter is included to all of you, because it's important for you to know what are the sexually transmitted disease, what are the different types of art technologies and all that. So we'll be doing that. So all of you look here. After the definition, we'll be trying to understand the birth control methods. So students, birth control methods are mainly mentioned natural, barrier, surgical methods, and then your intrauterine devices and oral contraceptives. It's a very, very easy topic. So just keep your mind and everything open and just try to listen to this, okay? You do not need to memorize anything. Students, so first thing is, how do I avoid unwanted pregnancy? Because see, even though a person is going through depression because they're not able to conceive baby, there are also people who are doing abortion every day. There are millions of people who are trying to abort the baby, or there are a lot of people who are undergoing miscarriages. So how do I avoid unnecessary pregnancy? Because unnecessary pregnancy, or the procedure of avoiding that, also causes depression. Now let's see that. First thing is natural. Natural method is divided into three types. First one is coitus interruptus. That means basically withdrawing the penis from the vagina, so before the sperm ejaculation. This way they will be able to avoid pregnancy. But the problem is, students, these techniques are not, are they like reliable? Are they reliable? My tongue is twisting. Okay, so okay, all of you tell me in the chat box, yes or no, ma'am. Yes or no, ma'am. Okay, yes or no, ma'am. Vijan, I'm seeing all your messages. Okay, you are not asking any doubt. That is the reason. This is a very serious class. Okay, and I do not have the time to answer other questions. So if you have any subject related question, please ask me. Now, students, the first thing is coitus interruptus. So first thing is what? Withdrawal of the penis from the vagina to avoid the transmission. That's all. But is it like 100% yes, no matter if the withdrawal happens, the woman is not going to be pregnant? Definitely not, because the pre-ejaculatory sperm, the pre-ejaculatory semen which already got released, okay, already has millions of sperm in it, and the sperm can survive in a human or in a female's body for 4 to 5 days. During this 4 to 5 days, there are chances that ovulation, or there are chances that fertilization will happen. Yes or no? Yes or no? So this is not a very good method. So if the question comes, which is the most reliable method, you should please opt for IUDs and oral contraceptives. Do not use for natural methods. Okay. Now, second one is, students, avoiding coitus from the days, that is 10 to 17 of the menstrual cycle. That means the chance where the woman will be, on the 14th day she will ovulate. But here and there, most of our cycles are 33, 34, 36 days. So in such kind of cases, okay, what is going to happen? The chances, like, okay, it's like astrology. It's like astrology. You throw something and you'll be like, yeah, today is a very lucky day for all of you people. You will all score 100 on 100 in your CBSE exams, your Neet exam, you're going to crack. That is astrology. That is not right? That is just like, might happen. Same way, this is also astrology. If the female ovulates somewhere between 10 to 17 days and they avoid sexual intercourse, maybe the woman will not get pregnant. Again, a failed reason. Next one is, intercourse is avoided at the day of ovulation. That means on the 14th day. How do the man know when the woman is going to ovulate? The woman itself don't know her period day. The woman itself don't know how long her cycle is. Will the man be able to sit and predict? Again, astrology, he'll do an assumption. Okay, maybe this day she's pregnant on 14, because that fellow would have studied human reproduction, and he'll be like, all the women ovulate on the 14th day and all the women have 28 days cycle. Do all of you agree or not? Do all of you agree that every woman have 28 days proper, proper menstrual cycle, and on the 14th day ovulation will happen? Yes or no? Okay. So again, this is a prediction, luck, by luck. This will also fail, this will also fail, this will also fail. Got it? So these techniques are not very reliable. I don't know why I can't say this one. Next one is, students, the most used technique, the most, what to tell, which is used the most time, that is called lactational. That means during the ovulation, if you don't do sexual intercourse, that is called as lactational amenorrhea. Okay, now next one is, students, very easy, barrier method. That means using condoms, contraceptive, diaphragm, cervical caps, wallets, spermicidal. So here, this one is just to lubricate the condoms. Okay, that means to make the protection a little more better, we use spermicidal creams, jellies and foams. Okay, they are not a method to avoid. On the condoms, cervical wallets, caps, these are just like, a foaming or a lubricant kind of a thing. Got it? Did you all smash the like button, students? Come on, please, yeah, don't be confused. Don't be so confused. Don't be so KDs. Don't run away from the class because I asked you to like. What is there in liking? Okay, what is there in liking? It's showing love. Yes, ma'am, I was there in the class and I totally enjoyed it. Okay, so barrier methods clear. Use of condoms, which is the most safest method. But again, lot of people around the world has got pregnant even after using condoms. That is because in the condom packages also, they will definitely write it is only 98%, you can trust it. 2% or 1% failure is always there. Okay, 2%, 1% failure is always there. Now, next one is the most used. Students, the question will come like this. Which is the most used technique in India? IUDs. IUDs is the most used technique in India. That means intrauterine devices. The female will be inserting these devices in her vagina, and this will release copper, this will release some kind of hormones which will make sure the sperm cannot survive in her body. Okay, so it is first non-mediated IUDs, that is your lips loops. Okay, copper releasing IUDs, that is your CUT, CU7 and your multi-load 375. Please keep it in mind, they will directly ask you a question. They will directly ask you a question. Okay, next question, student, hormone releasing IUDs. That means, see, we have to control the progesterone and estrogen level. To manipulate these levels, we will be using hormone-releasing IUDs, which can be a combination of P plus E, that is progesterone and estrogen, or P, or just E, one among this. Okay, and your, yes or no, got it? And your LNG20. These two examples are there. So, progesterone and LNG20. Okay, LNG20. That is your levonorgestrone. Levonorgestrone. Okay, now students, no use of writing because it will anyway go. Now, contraceptive. Now, students, what are these three people doing? That is your lips loops, IUDs, that is your CUTs and your hormone releasing. Okay, what are they doing? They are going to make sure that the sperm's motility is reduced. That means sperm has to be very motile to reach the ovum, right? Just now you studied that with Baswasa. Now the question comes, how do I make sure that unwanted pregnancy does not happen? I reduce the motility, and then this continuous release of copper will reduce the motility. The hormonal IUDs will change the, the hospitality that the vagina provides for the sperm will completely change. Okay, so it will make sure that we get a non-hospitable, environment for the sperm, so that the sperm dies. Then they will increase the mucus thickness so that the sperm cannot travel through the mucus. Got it? Clear? Clear? Now, contraceptive pills also work on the same hormonal changes only. That is, contains progesterone and estrogen. It prevents implants of the blastula. Implants will be kept under the skin. Okay, implants will be kept. Norplants, example is Norplants, you can remember that also I have made you write. So, implants will be kept under the skin, which will again avoid the unnecessary pregnancy. Now, the next one is again the most used, but this method, the question will come in such a way, when a family or when the male and the female is done with their family planning. Question to all of you, when a male and female is done with the family planning, which is the technique that they use? First question. Second question. if they want a break from the first baby and the second baby, what is the method that they use? Two questions, I want answers together. First question, if the family planning is done, which kind of method they will use to avoid pregnancy? Second thing, if they want a break from the first child and the second child, or the second child and the third child, which is the method they will use? All the answers, come on, in the chats. In the chats. Now, okay? Yes, awesome. Twinkle, twinkle. She's twinkling. Okay? Yes, come on, all of you. I'm waiting. Like Vijay's dialogue, no? I'm waiting. Yes, awesome, awesome. Dishti, Meg, Kia, Saumya, Kota, Twinkle, Darshini, Vaishnavi, Rinin. Okay, all of you, awesome. Janisha, awesome, awesome. What is it? First one, you can use the surgical method. That means you have already done the family planning. You do vasectomy and tubectomy and completely avoid any kind of pregnancy in the future, because you have already decided that, okay, I want only 2 kids. So we are going to stop the pregnancy right here. Okay. Then the next thing is what? I want a delay from the first baby and the second baby. I can definitely use IUDs. I can also use your oral pills, which are there for 3 years, 5 years, or implants. Okay, implants is an option. IUDs are an option. Most of the people use implants because implants are there for sometimes a period of 2 years to 3 years. Okay, IUDs you will have to keep changing, but 3 years to 2 years, that option is given by oral contraceptives and oral pills and your implants. So implants also is an answer. Okay, awesome, good job, good job. So vasectomy you have in males and tubectomy in females. In vasectomy, what is happening? Vas difference is cut and tied. Okay. Tubectomy, what is happening? Fallopian tube or oviduct is cut and tied. Okay. Now, students, actually, how did they start this gender revealing, all this? How did this test happen? Very simple. The process was called as amniocentesis. Okay. Amniocentesis is a thing that was done before to know if the baby has any abnormalities, if the baby is going through any genetic disorder, so that so that it does not become a burden later for the parents as well as for the child. If the life is going to be difficult and miserable later, they had an option to, abort the child. This was only done in extreme cases. But what happened was later, parents started using it, or people started using it to understand the gender of the baby, and they started female homicide. That means if the baby was a female, we will just kill the baby because I do not want to educate her, I do not want to give the dowry, and lot of misconception about women. Okay, clear? So amniocentesis became a negative thing because amniocentesis turned out to be a thing to gender reveal and to lead to female homicide. Got it, all of you? So here you can see negative application, positive application. Positive application is to detect any genetic disorder, any kind of diseases in the baby. Is the baby normal? Technically speaking, just to know the health of the baby. Okay. Next thing is, yes, correct, awesome. Used to detect genetic disorders. Awesome. Okay. Now what has happened? The negative thing was that we understand the gender of the baby and we do female homicide. Okay. So now amniocentesis is completely stopped, even though in abroad people do it, but they do not lead to female homicide. They are very happy even if it's a boy or a girl. You would have seen a lot of gender revealing videos, but in India, amniocentesis is completely stopped. Okay. Now, students, after understanding this, we need to understand what are the problems if I do not use, proper contraceptives, and what if I have, what if a person has multiple partners, and what are the problems they will cause? Very, very simple thing is sexually transmitted disease, or your STDs, right? STDs, your AIDS. And we also understood HIV life cycle. You know how dangerous HIV is, and a person who has AIDS will not die just because HIV is inside them. They will die because even a normal fever will become very extreme for them because their T cells will be continuously damaged during this process. Yes or no? So look here, you get hepatitis B, genital herpes, HIV, diseases transmitted through sexual intercourse, or you happen to use graft, that is your implantation or anything, and there is a small mix of the blood sample, that time also it will happen. Okay, and if the mother has it, it will get transferred to the baby as well. Now, what is the importance? So look here, to avoid unwanted pregnancies and in cases where continuation of pregnancy is harmful to the mother and fetus. All of you know erythroblastosis fetalis, a condition where because of the blood group, the second baby is in problem. Now, such a condition come and Rogue is the vaccine which we use to avoid this. Okay. Now, in case nothing works for the mother, okay, nothing works for the mother, what do? We can definitely use your MTP, that is medical termination of pregnancy. This is only applied when the woman is in the first trimester. Agreed, all of you? Which is the most safest time to do MTP? All of you in the chats. Which is the most safest time to do MTP? Okay, come on, all of you. And tell me the full form of MTP. Tell me the full form of MTP and which is the safest time to do MTP. Okay, done. Now next one. All of you in the chats. Next question to all of you. When is ZIFT? Okay, when do I do ZIFT or embryo transfer? Is it in the above 8 blastomere stage or below 8 blastomere stage? Awesome. First trimester, right? First trimester, that is up to 12 weeks. Okay, up to 12 weeks. Yes. Next question to all of you. When can I do ZIFT or when can I do embryo transfer? When can I do ZIFT and IUT? This two. ZIFT, when can I do? When can I do IUTs? Is it above 8 blastomere stage or below 8 blastomere stage? Yes, medical termination of pregnancy. Yes, awesome. Students, full form, very, very important. Even though this chapter will be like, oh, such a jujube chapter, I do not have to study. Lot of questions can come from it. Jujube means, yeah, why did you ask? Okay. Yes, awesome. ZIFT means below 8 blastomere stage. Yes, yes. Okay, and IUT is above 8 blastomere stage. Okay, that is 8 to 32 blastomere stage. IUTs will be done. ZIFT will be done below 8 blastomere stage. Very, very important question. And both of these people come under embryo transfer. Okay, embryo transfer. Fine. Students, very simple. Full form you have to know. ZIFT, GIFT, IUTs, everything. Zygote intra-fallopian transfer. That means once the zygote is formed or the embryo is formed, it will be transferred to the fallopian tube of the mother and later the rest of the development will happen there. Okay. Next we have ICSI. That means intracytoplasmic sperm injection. That means the female does not have a problem but the male sperm has some motility issue or the number is not okay, then in these cases we will be injecting the sperm. Okay, they will be injecting the sperm. That is your ICSI. This is only when the couple has failed IVF. Okay, ART or your assisted reproductive technologies is when a person is not able to conceive normally. We do a different use of technology. Okay, now very, very common one is IVF. Now in case IVF does not work, we go for your ICSI. Okay, good job, understood? Fine. Okay, now ZIFT is your gamete intra-fallopian transfer. That means we transfer the male and the female gamete into the woman. Okay, and we let the fertilization to happen inside. This is only cases again when the male has a problem and then even the female ovum is not ovulating, some kind of issue, both their timings are not matching. That time what is going to happen, we are going to collect this and we are going to fuse it and we are going to add it, or if the egg does not have a problem, we only inject the sperm into it. Got it? This is done. This is a gift. Okay, gift. Now AI is artificial insemination. This is used by females only. Okay, used by females only. So students, this is I think all about it. Let me see once again everywhere. Okay, so is this clear? 8 blastomere and above 8 blastomere stage, very important. You should know that IVF is the most common art technology which is used. Okay, in vitro fertilization, that means we make all these procedure which is supposed to happen in the body inside the lab. Okay, in vivo and in vitro. Okay, that is what we are going to do. That is IVF is the most common. In case IVF does not work, we go for other kinds of techniques. Okay, and then there are a lot of people who freeze the egg and sperm also. If they feel like they're not ready to have a baby, they do that. Okay, and did we miss anything? Did we? Students, Saheli. Saheli is a non-hormonal pill. Saheli is a non-hormonal. That means it does not have any side effects. That is the only pill in India which does not have any side effect, and it comes, the entire strip comes for I think 24 rupees, which is also very affordable. Very affordable. This was done in your Central Drug Research Institute in Lucknow. Okay. Yes, I think that's all. Importance, MTP done. Are we done with everything? Amniocentesis, birth control, surgical, barriers. Yes. Done, done, done. Yes, done. Done? Are we good? Yes, once in a week is a non-steroidal pill or your non-hormonal pill. That means basically it does not do any side effect. All other things have side effect. Okay, all other things have side effect. Okay, yes, CDRI, Central Drug Research Institute in Lucknow. Got it done? Done, 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 done. Chalo, let's give the mic. Hand over the mic to our respective guest of the day.
Yes. Thank you so much, ma'am. Thank you so much. Thank you so much.
Please give a vote of thanks.
Thank you so much.
Thanks so much for providing the speech.
Thank you so much for giving me the mic.
Okay.
Ma'am, how is the Josh now? Is everyone ready for the next chapter?
Up and down,
no?
Yeah, up and down.
Okay. If you start singing, I have to run away from here.
I will do that if you don't come up.
Okay. Okay, students, are you ready for principles of inheritance? Ma'am, you took the. Okay, ma'am took the pen. Ma'am, ma'am went out also. How will I write now, students? How will I write? I have to some extra. I have to use some other one. So, hello, students. Are you ready for principles of inheritance? Quickly, let me know in the chat if you are ready for principles of inheritance. Ma'am took away the pen. I don't have any pen. I have to use some extra pen now. This is what happens. She gave the mic and she took the pen. Students, okay. First, principles of inheritance. The first important point we are going to talk about here is, all of you know, I'm not going to tell you what the difference between a character and trait. All of you should know by now what is the difference between a dominant allele and recessive allele. What is the difference between a character and a trait. What is the difference between a homozygous and heterozygous. That basic information everyone should know by now. But addition to that, we'll be talking about Gregor Mendel first. We'll be talking about Gregor Mendel. Now quickly tell me in the chat, Gregor Mendel, what was his experiment? Like, what did he work on? Not the plant. Plant is a separate question. What was his experiment called as? Did he do recombination? Did he do linkage? What did he try to achieve? So students, Gregor Mendel experiment is called as hybridization. What is it called? Plant hybridization. The main plant which Mendel used was called as Pisum sativum. Now one question which can be asked in your board examination is, why? Why did he take pea plant? All of you eat peas, right? In palau and everything. All of you eat peas. So what was the reason he used pea plant? Is there any speciality about it? Yes. There are a lot of speciality about your pea plant. Life cycle is less, contrasting traits, bisexual in condition, easily can undergo self-fertilization to produce pure lines. Yes. So that can be asked as a question, why the pea plant? Now students, Mendel used pea plant which had seven different characters. Now each character is equally important here because they can ask you in your examination. In your board examination they can give you the entire list, they can ask you. Or in your NEET examination they can ask you. In your NEET examination they ask you which is the dominant trait and which is the recessive trait. They can ask you which is the dominant trait and which is the recessive trait. Very important. And I have told you a trick to remember this. Yes, the trick is gravity. All of you remember the trick for your dominant as well as recessive trait. Now apart from this, Mendel used something called as 14 true breeding lines. Now what are these true breeding lines? True breeding lines are the lines, they are not a line, they are basically a set of plants. They are basically a set of plants which are homozygous in nature. They are what? They are homozygous in nature. They can be homozygous dominant or they can be homozygous recessive in nature. Now, true breeding lines are the lines which have undergone self-pollination repeatedly. Now, what is true breeding lines? True breeding lines are the lines which have undergone repeated cell division. Very important point, students. Very important point. Now, here they can ask you what is different characters. They can ask you a question. So, please be prepared for that. Now, next, students. What are the laws? What are the laws did Mendel give? Now, all of you know monohybrid clause. Now, quickly tell me in the chat, what is the phenotypic ratio in the case of monohybrid? And also tell me what is the genotypic ratio in the case of monohybrid. That is, phenotypic ratio is your 3:1 and the genotypic ratio is going to be 1:2:1. Basic class 10th level? Yes. Now students, what did he understand from monohybrid cross? He did monohybrid cross and he obtained something from it, right? What were the different laws? What were the different laws he obtained from monohybrid cross? So students, from monohybrid cross, the main laws which was obtained is law of dominance as well as law of segregation. Students, the two laws which were obtained from monohybrid cross was law of dominance as well as law of segregation. Now, what is this law of dominance? Law of dominance basically tells you, shall I read it? I'll tell it as such. Law of dominance basically tells you, out of the two alleles or factors which Mendel used, out of the two factors, one factor is, I know, I realized it. Pass me the pen, ma'am. Out of the two factors, one factor is dominant, one factor is recessive in nature. The dominant factor will express. It is like the talkative person in the class. He will always keep on talking even if others are present. The dominant allele will keep on expressing even in the presence of the alternative allele. The other one is your recessive allele. A recessive allele is a silent kid in the class. He will not talk when others are present. Yes. Tell me in the chat, how many of you are extrovert or introvert? I'm a big extrovert, right? So students, introverts are the people who do not talk when other people are talking. They're like, nah, my work, my life. So that is your recessive allele. Recessive allele will not express in the presence of dominant allele. So that is law of dominance. Done. Now what is this law of segregation? Law of segregation is very simple. Law of segregation basically tells you that this is your F1 generation. Yes, in the F1 generation, one second students. In F1 generation, we have two type of alleles. In F1 generation, we have two alleles. Now students, these two alleles will segregate into gametes. These two alleles will segregate into gametes, and each gamete here is pure for that particular trait. For example, this gamete is pure for tallness. This particular gamete is pure for dwarfness. That is why law of segregation is also called as what? Law of purity of gametes. That is, the two different alleles segregate during gamete formation. Clear? Is that clear to all of you? Is that point clear to all of you? Yes, that is clear. That is called as your law of segregation. That is, the two alleles are segregating. And the other main point here is, the law of segregation also tells you that alleles do not blend. It tells you what? Alleles do not blend. That is, the two different alleles might be present here together, but are they blending? Are they in a relationship? No, they are not. They might look like they are together. You have in your class, right? One girl and guy will be, or you might see them always walking together. You might think they're a relationship, but they are not. That is what is happening here. You might think these two alleles are blending. They are not blending. They are not blending. Because even in F1, the small t may not be expressing. The small t may not be expressing right now. But in the F2, will the small t express itself? Yes. Why? Because it did not undergo blending. In F2 generation, we will obtain one dwarf plant. One dwarf plant will be obtained in the F2 generation. Clear? That is your law of segregation. Then we have dihybrid cross. Then we have what? Dihybrid cross. Now what is this dihybrid cross? Can anyone tell me, in dihybrid cross, do we take a single character or do we take two characters? Tell me in the chat. So students, in your NCERT example, they have given you for dihybrid cross, shape of the seed and color of the seed. What have they given you? Shape of the seed and color of the seed. Now quickly tell me in the chat. Dihybrid cross, what is the phenotypic ratio and what is the genotypic ratio? Tell me in the chat quickly. In the case of your dihybrid cross, the phenotypic ratio, it means how the plant looks from the outside. How the plant looks from the outside is called as phenotypic ratio. The phenotypic ratio in the case of your law independent, in your dihybrid cross, the phenotypic ratio is going to be 9:3:3:1. In here, the 9 is going to be what? The 9 here is going to be what? Round and yellow. 3 is going to be your round and green. Yes. So 3 is going to be your 9 is going to be your round and yellow. This 3 is going to be round and green. Yes, I told correctly. The next 3 is going to be what? Wrinkled and yellow. And this 1, can you see this 1? That is your wrinkled and green. Its last one is what? Wrinkled and green. Now, out of these 16 combinations here, the first 9 1 and this 1, both of these are called as what? Both of these are called as your parental combination. What are they called? They are called as parental combination. Very important. Now, these two, 3, they are called as what? These two are called as what? Recombinant types. They are called as what? Recombinant types. Because round and yellow will look like one of the parents. Yes, that is a dominant parent. Wrinkled green will look like the other parent. That is going to be your recessive parent. Clear, students? Are you understanding? Now, this is your phenotypic ratio. Now, what is genotypic ratio? Genotypic ratio is going to be your 1:2:2:1:2:4:2:1:1. Yes, that is going to be your genotypic ratio. And I've already told you the trick to remember it. All of you remember the trick, right? Do all of you remember the trick to solve this? If you do not know the trick, ask me right now. I'll tell you the trick again. Okay? Now done. That is your dihybrid cross, students. Now from your, see, they have given you dihybrid cross. Dihybrid cross, clear? Now students, which law do you obtain from dihybrid cross? Now what is that law? From your dihybrid cross, we obtain law of independent assortment. Which one do you obtain, students? Law of independent assortment. Now, what does the law of independent assortment say? Law of independent assortment is very simple. Law of independent assortment is very simple. It is telling you that the two pair of characters, right, they will assort independently. They will not merge with each other. They will not interfere with each other. That is, when two pairs of contrasting traits, that is going to be your round and yellow and wrinkled and green. Two pairs of contrasting traits, they are combined in a hybrid. They might be combined in a hybrid. Segregation of one pair of characters, that is segregation of round and yellow, is going to be different from segregation of wrinkled and green. The two different characters are going to get separated independently. Clear? Is independent of other pair of characters. Independent of other pair of characters. Some of you are asking me, sir, you do not know the trick. Can you please tell the trick? Very simple, students. All of you know multiplication. All of you know multiplication, right? Does all my students know multiplication? Yes, sir, we know multiplication. Students, 1:2:1 into 1:2:1. Is this your genotypic ratio in your monohybrid? Is this genotypic ratio in the case of your monohybrid? Yes, sir, that is genotypic ratio in monohybrid. So when you multiply two different monohybrid, we get a dihybrid. How, sir? 1 into 1 is 1, 1 into 2 is 2, 1 into 1 is 1, 2 into 2 is 2 into 1 is 2, 2 into 2 is 4, 2 into 1 is 2. Then 1 into 1 is 1, 1 into 2 is 2, and 1 into 1 is 1. That is your genotypic ratio. That is your genotypic ratio. Similarly, you can do for phenotypic ratio also. How do you do, sir? 3:1 into 3:1. 3 3s are 9. 3 1s are 3. 1 3s are 3. 1 1s are 1. Done. That is a small trick to remember phenotypic and genotypic ratio. Clear? Now, that is also done. Now, students, apart from 3 Mendel's law, are there any exceptions to Mendel's law? Tell me in the chat, are there any exceptions to Mendel's law? Can anyone tell me? Yes, sir. All of us have heard something called as incomplete dominance. Now, what is the incomplete dominance? Students, in the case of your incomplete dominance, one allele is incompletely dominant over the other allele. That is, if I am the dominant allele, I am not doing my work properly. I'm very lazy. I'm like, even though I'm dominant allele, I'm so lazy that I'm not suppressing the other one. That is called as your incomplete dominance. Where is it? Where is it? Here, students, here. Incomplete dominance. One of the two alleles of a gene is incompletely dominant over the other allele. For example, listen to the example. We have the flower color in the case of your Antrium or Mirabilis jalapa. Antrium or snapdragon, also called as your dog flower, also called as Mirabilis, is called as what? 4 o'clock plant. In this case, we see something called as incomplete dominance. Example, important. Example is very simple. That is, they have given something here. Okay. Example is the flower color. For example, students, we have two type of flowers here. One is your capital R. One is your capital R, which is the dominant color. Then we have small r and small r. That is your recessive color. Okay. When they two have a baby after forming gametes, we get a result that is capital R, small r. Now this capital R, small r, does it resemble any of the parent? Yes or no in the chat? Tell me. This capital R, small r, which we kept in incomplete dominance, does it resemble any of the parents? The answer should be no, sir. It does not look like red. It does not look like pink. So it doesn't look like white. What is it? The color here is pink color. It means the child is adopted. Here. That is pink color flower. Now students, the most important part here is going to be the phenotypic ratio and genotypic ratio for the incomplete dominance. Now what is the phenotypic and genotypic ratio here, students? So in the case of, it's not given here. It's given here. Okay, sorry, it is given here. Notes are notes. Everything should be given, but now yes, students, listen to me very carefully. In the case of your incomplete dominance, the phenotypic ratio and genotypic ratio are going to be same to same. They're going to be what? Same to same. Because why? Because blending happened. So phenotypic ratio is going to be 1:2:1. 1 is going to be red, 2 are going to be pink, and 1 is going to be white. And the genotypic ratio will remain the same. That is, 1 is going to be homozygous dominant, 1 is going to be homozygous recessive, and 2 are going to be heterozygous dominant. Okay? Clear? That is your incomplete dominance. Now students, the other one is called as your codominance. Now what is this codominance? What exactly happens in codominance, students? In codominance, tell me in the chat, what happens in codominance? In the case of codominance, students, the child, the offspring will look like both the parents. It will look like what? It will look like both the parents. And example is your ABO blood grouping. Example is what? ABO blood grouping. Yes, blending is happening. Exactly, blending is happening. ABO blood grouping. Now what exactly is happening here? What is the concept here, students? The concept here is called as multiple allelism. What is the concept here? Multiple allelism. Now what is this multiple allelism? Multiple allelism is very simple. All of us know till now, one gene has two alleles. One gene will have two alleles. But in the case of multiple allelism, one gene will have many alleles. One gene will have what? One gene will have many alleles. That is going to be here. That is your multiple allelism. A gene exhibits in more than two alleles. Example is your ABO blood grouping. It's called as what? ABO blood grouping, which is seen in the case of your ABO blood grouping. It's called multiple allelism. It is called as multiple allelism. Example here is very simple. We have here the dominant allele is what here? So in the case of multiple allelism, okay, what happened? In the case of multiple allelism, we have three different alleles. IA, IB, and I0. Now out of these three alleles, The two are dominant, one is recessive. One is recessive. Now students, out of these three alleles, two alleles will be present in the case of humans. Two alleles will be present in the case of humans. So every single time we have IA and small capital small I naught, A will be dominant. IB and I will be present, IB will be dominant. So when both are present, IA and IB, when both are present, it will result in AB blood group. AB blood group. That is your codominance. Clear? Next we do something called as pleiotropy. Now what is this pleiotropy? Pleiotropy is very important. Pleiotropy is very important. What is pleiotropy? Ability of a gene to have multiple phenotypes. So one gene. So in the case of pleiotropy, what is happening? In the case of pleiotropy, one gene, multiple phenotypes. One gene and multiple phenotypes is your pleiotropy. Clear? One gene, multiple phenotypes. Ability of a gene to have multiple phenotypic effects as it influences a number of characteristics simultaneously. That is your pleiotropy. Now one question comes in. Now what is polygenic inheritance? What is this polygenic inheritance? That is, polygenetic inheritance is ulta of this. In the case of your polygenetic inheritance, we have many gene. Gene 1, second gene, third gene. Many genes are influencing one character. That is, example is what? Example is skin color. Human skin color. Human skin color is the example, or the human height. And remember students, polygenetic inheritance is what? Additive in nature. It is additive in nature. More number of genes, more darker the color in the case of human skin color. So more genes, more effect. You write it. More genes, more effect. The more you like the video, more will get energy to teach. Right? That is your polygenetic inheritance. Done. Then students, then we have, one second students. This is done, multipluralism is done, pleiotropy is done. Now students, next thing we understand is mutation. Next thing, let's go to mutation. Okay. Now what is this mutation? What exactly happens in mutation now students? Mutation, all of you know. What is mutation? Mutation is nothing but sudden inheritable change. Mutation is what? Sudden inheritable change. Now mutation can be of multiple types. That is frame shift mutation, point mutation, or there can be a complete change in the genome itself. That is aneuploidy and polyploidy. Aneuploidy and euploidy. Yes, it can be aneuploidy and euploidy. That is complete change in chromosome. One chromosome number can increase. That is in the case of your trisomy of 21. We'll come to that later. Or there can be a deletion of chromosome. That is in the case of your Turner syndrome. Yes, students. Now there can be a chromosomal aberration also. Now what is chromosomal aberration? There can be a chromosome can be damaged. A structure of chromosome can be changed. All of those causes the mutation. The next concept is your sex determination. Very easy again. Direct question. From the sex determination, you can get a direct question. Students, from your sex determination, a direct question. Now what happens in sex determination? We have something called as male heterogamety and female heterogamety. Now what is this male heterogamety? Male heterogamety is very simple. That is, the male gametes will decide the offspring. That is, in the case of male heterogamety, there are two different male gametes. That is, two different chromosomes can be there. Two different chromosomes. For example, in the case of your humans, not given here. In the case of humans, it can be male can be XX or XY. Or in the case of your grasshopper also is male heterogamety. That is X over X XO is the male heterogamety. Now what about the female heterogamety? In the case of female heterogamety, it's going to be for birds and butterfly. That is, the female is going to be different. The female chromosomes are going to be different here. Example is here birds, ZW are the two different type of chromosome in the case of birds. Very simple, direct question, students. Expect direct question here. No changes will come here. No changes. Direct question. Now, next is your chromosomal theory of inheritance. Next one is chromosomal theory of inheritance. Very important. Very important. Tell me how many of you know, how many of you are still confused? Tell me in the chat how many of you are still confused about chromosomal theory of inheritance. Tell me. How many of you are still confused about chromosomal theory of inheritance? Or if you know but you might have forgotten. Anyone tell me in the chat? Yes, yes. Insect, yes. See, in the case of your grasshopper and humans, it's male heterogamety, right? In the case of your female heterogamety is seen in the case of your butterfly as well as your birds. You have forgotten? Let me remind you. Let me remind you with one simple line. Chromosomal theory inheritance was given by Certain and Boveri. First point. It was given by what? Certain and Boveri in the year 1902. 1902. Now, but what does it say? Chromosomal theory of inheritance basically tells that there is a parallelism between chromosome and gene. That is, all they are trying to tell you is chromosome and gene are basically like brother and sister, or they are like twins. They are like twins. That is, chromosome behavior and gene behavior is same to same. The way chromosome is behaving and the gene is behaving is same. If the way chromosomes are segregating, gene is also segregating the same way. So the behavior is same to same. That is all is chromosome inheritance. Students, and Certain and Boveri basically explained the Mendelian traits. They explained Mendelian traits with the help of chromosome separation during the gamete formation. Clear? That is your chromosome theory of inheritance. That is, chromosome behavior and gene behavior are same to same. Clear? Now, students, Certain what did he do? Certain basically did what? He compiled information. He compiled information on chromosomal separation plus Mendelian inheritance. In Mendelian inheritance, and together they call it as chromosomal theory of inheritance. Chromosomal theory of inheritance. CTI. Chromosomal theory of inheritance. Done. Do all of you know now? Yes. That is your chromosomal theory of inheritance. But who gave the proof? The proof of chromosomal theory of inheritance was given by none other than your T.H. Morgan. Thomas Hunt Morgan gave the experimental proof. Experimental proof was given by in the form of experiment was done on your Drosophila melanogaster. Was done on Drosophila melanogaster. That is, Morgan coined the term linkage as well as recombination. Linkage as well as recombination also was given by your Morgan. Now, all of you should know one point here, that is between linkage and recombination. That is, this is very, very important. Linkage is inversely proportional to recombinance. Clear? It is linkage is inversely proportional to recombinance. That is, if linkage is increasing, that is, if linkage is more, recombination will be less. Recombination will be less. Inversely proportional relationship you should know. Okay, that is very important here. Done. Numericals you can expect on that. You can expect numericals, and if you want to know the formula of recombinant frequency, number of gamete formation or the offspring formation, please watch the principles of inheritance in one shot and you will get the concept very clear. Okay, please watch the session. And students, if you are new to the channel, like the video and share the session, because you will not find anywhere on the YouTube who's going to teach the entire biology with the help of mind map, every single chapter in 30 minutes. Okay. Next one is your pedigree analysis. Next one is pedigree analysis. Now students, all of you should know this. All of you should know this. Pedigree, you can expect a direct question. Oops. Pedigree, you can expect a direct question here. Okay. Pedigree, you can expect a... One second. Molecule bus came off. Why is it not zooming? Oh, no, it came to some much. In pedigree analysis, they can directly ask you a pedigree. That is, they can either give you this entire symbols, or they can ask you which symbol represents what. That question they can ask you. And they can also ask you, they can give you the two different pedigree analysis they have given you. That is, one is your autosomal dominant myotonic dystrophy or autosomal recessive, that is sickle cell anemia. Any of those two, they can give you the pedigree, and they can ask you to identify which one is what. That is very important, both for NEET and as well as for boards. Then students, why are we doing pedigree? Pedigree study is to utilize trace the inheritance of species traits and abnormality of diseases. Basically to find out if in your family, if there is any abnormality is happening or not. That is pedigree analysis. Clear? Any doubt in pedigree, students? And students, if you want to know how to do pedigree analysis, I have done the video on it. Please go check it out in Principles of Inheritance. What are the different characters of your autosomal dominant? What is the character for your autosomal recessive? What is the character for your X-linked dominant? What is the character for your X-linked recessive? What are the characters for your Y inheritance? All of them is done and dusted. So please watch it once. The last part of this chapter is going to be diseases. That is genetic diseases. The last part is going to be genetic diseases. Students, the entire genetic diseases is divided into two parts. That is chromosomal disease as well as your Mendelian disease. Chromosomal disease as well as Mendelian disease. Now under Mendelian disease, we mainly have autosomal dominant, that is going to be your muscular or myotonic dystrophy. Autosomal recessive, we have sickle cell anemia, right? We have sickle cell anemia. Then we have your sex link, that is hemophilia. Also your other one example is going to be your color blindness. Other one is going to be color blindness. The one example is not given here. Let me know in the chat the other example for your autosomal recessive. One example is missing here, which is from your NCERT. Tell me that also. Then students, we have chromosomal observation. In chromosomal, we mainly have three diseases. How many? We have three different diseases. The first one is called as your Down syndrome. In Down syndrome, there is an extra chromosome number 21. That is also called as trisomy of 21. What is what? Trisomy of 21. Exactly. Thalassemia is missing there. Chromosomal 21. Then we have Klinefelter syndrome. That is extra X chromosome. In the Klinefelter syndrome, a particular male will develop breast, and that is called as gynecomastia. That is gynecomastia. Extra X chromosome. The last one is called as your Turner syndrome. That is one X chromosome is missing. One X chromosome is missing in the case of female, which will result in the formation of a female of very short stages and also rudimentary ovaries. Students, with that, the entire principles of inheritance is over in 30 minutes. Do you notice that? So with that being said, now we'll be doing the next chapter, that's going to be molecular base of inheritance, and let's invite Gopika Ma'am for that. Guest of honor again.
Right.
Okay.
All of you vanished. They think question will only come from reproduction chapters. It's a reproduction unit. Okay. Yes. Okay, students, now we are going to start molecular basis of inheritance. Where's the pen? Yeah, the Soli. Where's the other one? Oh. Okay. Yes, students, molecular basis ready? Okay. Chalo, let's start. Okay, molecular basis of inheritance, my students' nightmare. Every day I get at least one message telling, ma'am, molecular basis of inheritance, can you do it again? Okay, I don't know what is so difficult about this chapter, but let's try breaking it down as much as possible. But students, with this alone, like if you're studying this chapter for the first time, you will not understand. Okay, if you're studying it for the second, third time, definitely it's going to be easy. Okay. When we talk about molecular basis of inheritance, you only have to think about 2-3 people. That is DNA, RNA, protein. But mainly we are focusing on DNA, which is the genetic material for most of the organism. Now the question arises, such an important thing, DNA, how is it formed? It is simply formed with something called as a nucleotide. Is nucleotide alone? No. Nucleotide has 2 more or 3 components that join together to form the DNA structure. So when many nucleotides come together, what is that called, students? Many nucleotide comes together. What is it called? What is that chain called as? So see, what is the chain called as? Here, molecular basis, it is. Who is telling the answer? Come on, fast, 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 fast. Okay. So, it is called as polynucleotide, right? So, in your textbook, you will see structure of polynucleotide chain. That means many nucleotide together forms the DNA. That nucleotide chain is called as polynucleotide. Simple. Now the question comes, what is this nucleotide made up of? Ma'am, you told 3 people are making it. Yes, 3 people are making it. Who are they? First one is a phosphate group, then a sugar and a nitrogenous base. Now, I told you that you have only few people to remember in this chapter. DNA, RNA. Now, DNA and RNA, there is some different. RNA has extra something that DNA does not have. All of you will tell me in the charts. Echoing? Is it echoing? Fine? Okay, something is echoing. Now it's better? I think now it's better, students. It's just the, yeah, okay. Okay, look here, all of you. It is made up of, yes, all of you told me, that is phosphate group, sugar and nitrogenous base. Yes, definitely thymine and uracil is different in DNA and RNA, but the first ever thing is, DNA does not have an oxy group or an oxygen group that RNA has, which makes DNA so much, so much stable, so much chemically, less reactive, so many things, okay, so many things. So here you can see sugar is of 2 types, ribose and deoxyribose, okay. Now, phosphate group is there, that is, so we have phosphate group, can I move this? So I'll write it here, students. This is your phosphate group, okay, this is your phosphate group. Now, sugar is there, that is your ribose sugar and deoxyribose sugar. Here we have an absence of oxygen, okay, we have absence of one oxygen group, because of which DNA becomes the most stable genetic material and it is the most accepted genetic material for most of the organism. Now, nitrogenous bases. Nitrogenous bases are divided into 2 people, that is your purines and pyrimidines. Okay, purines and pyrimidines. All of you tell me in the chat, how do you remember purines? How did I tell you how to remember purines? How to remember pyrimidines? All of you in the chat, quickly. Okay, how are you going to remember them? Purines and pyrimidines. What is the trick that I told you? Yes, phosphate group is the reason for negative charge of DNA. Very good. That is the reason why it is able to wind around the positively charged histones, right? Because such a long 2.2 meters long thread, we have to keep it inside the nucleus safely. We have to keep it inside the nucleus. For that to happen, we are going to take some histones, proteins that will wind around and form the nucleosome. Now the question comes to all of you is, how do you remember purines and pyrimidines? Okay, how do you remember purines and pyrimidines? All of you in the chat, quickly. What did I tell you? To remember purines, you will remember this, pure as gold. Okay, pure as gold. Pure as gold means what? P stands for purine, A stands for adenine, G stands for guanine. For pyrimidine, how will you remember? Cut you will remember. That is cytosine, uracil and thymine. This is how you are going to remember these. Okay. Now, one thing we know that, see, cytosine and thymine is only present in DNA, uracil is only present in RNA. That means thymine from DNA is replaced with uracil in the RNA. Okay, that's it. Then, here if you see, I told you that we have to make sure that this 2.2 meter DNA gets into the nucleus. For that, we need a complete packaging system. Who is going to give that packaging system? That is going to be our set of proteins called as histone proteins. Now, these histone proteins are positively charged. DNA is negatively charged. So, easy. Positive, negative will attract. Now, the question comes, how many histone proteins have been using? Students, 5 histone proteins are helping, out of which only 4 are helping in the histone octamer. What are they? What are they? All of your charts should keep moving. Don't sleep. Okay. These are the 4 histones. These are, red is not that visible, no? Yeah, these are the 4 histones that are going to help in the formation of an histone octamer. Okay, in the formation of an histone octamer. Each of them will double. Now, to keep all of these people in position, we have our H1 protein here that is also called as your linker protein. Okay. Now, DNA will go around the histone protein with the linker protein. Together, this entire thing is called what? It is called your nucleosome. Okay. And many nucleosomes together will form a bead of string structure under the microscope. That is why in your textbook there is a black and white image and down it is written bead of string. Okay, bead on a string structure. Okay, this is how the entire 2.2 meter DNA is packed into our nucleus. Now, why are we studying this chapter? What is the main thing about this chapter? The main thing, like how our earth revolves around the sun. Sun is the ultimate person, right? So, like that, around the sun also, all the planets are moving. Same way, the entire chapter or entire our, molecular biology, if you tell, the entire biology revolves around central dogma. Central dogma is nothing but DNA gets converted to mRNA, mRNA gets converted to protein. DNA to mRNA is called transcription, mRNA to protein is called translation. If DNA wants to make its own copies, it is called as replication. Okay, replication. Nuclein was the thing which was first found. Okay, first found by Francis. He was doing an experiment on the pus cells and by fluke he happened to find something called as nuclein, and then a scientist called as Altman found that this nuclein has some acidic nature, because of which we found out that it is called as nucleic acid. And the nucleic acids you are studying in this chapter is DNA and RNA. Okay, that is the starting of the chapter. Okay. Now, students, look here. This DNA forming its own copy is called as replication. Okay, and DNA does semi-conservative replication, and the experiment to prove what? Messerson and Stahl experiment. Yesterday I did that in MCQs. Okay. Now, look here all of you. First ever finding. Okay, first ever finding of like who is the genetic material, or DNA was the genetic material, definitely was by Francis and Altman. They found nucleic acid, but who is the exact genetic material, who is actually doing something, was found by Griffith's experiment. Okay, Griffith's experiment, he took a mice, he took 2 strains of Streptococcus pneumoniae, he took 2 strains of Streptococcus pneumoniae. He was working on Streptococcus pneumoniae and he took 2 strains, S and R strain. This is called the rough strain, this is called the smooth strain. Okay, rough strain did not have a polysaccharide layer or a mucus covering layer. Smooth strain had it. Now, simple students, S strain injected with the mice died because S strain was virulent, and they believe that because of the polysaccharide layer, the S strain was virulent. Then the R strain he injected with the mice, what happened? The mice lived. Okay, no, here the mice lived. Then he thought, let me do one thing. S strain when I put alone, the mice died. R strain when I put alone, the mice did not die. Let me do one thing. Let me heat kill the S strain. So he heat killed the S strain and he injected into the mice. The mice lived. Heat killed S strain, the mice lived. What did he do again? He took the heat killed S strain, he took the R strain and he combined together and he injected into the mice. He injected into the mice. Now he was like, what do you expect? If you were Griffith, what would you expect? If I was Griffith, I would have expected that the mice would live. Why? Because when I used heat killed S strain alone, the mice lived. When I used R strain alone also, the mice lived. So obviously, when I mixed this both, the mice were supposed to live. Griffith got shocked. Why? Because the mice ended up dying. He was like, what? The mice died? But how come? Now the question arises that there was something that transformed from the heat killed S strain to the R strain that made the R strain virulent, and that was termed as transforming principle. Because he did not know who got transferred, he did not know if it was RNA, DNA, protein, carbohydrates, lipids, he did not know anything. So he was like, let me do one thing, let me be on the safer side, let me call it transforming principle. And then he called it the transforming principle. Okay, transforming principle. Now definitely if one scientist come, then many scientists will come. After that we had your Avery, MacLeod and McCarthy, who tried to find the biochemical nature of this transforming principle. They tried, but they failed. Okay, I'll tell you why they failed, even though they had an answer for, whatever the questions were there, but most of the biologists and zoologists did not agree. Simple thing. So here you can see Avery, MacLeod and McCarthy did an experiment that DNA is the genetic material. So basically they took some enzymes, protease, DNase and RNase. They injected that into this combination, that is your heat killed S strain and R strain together. They put this. Now imagine if protein is supposed to be the genetic material, when I use protease, protein will be digested. Then the mice were supposed to live. This was what the explanation was. If protein was it, if I use protease, protein will go, then the mice should live. Same thing for RNA, DNA. He did the experiment, he used protease, protein got died, but then the mice died. Okay, the mice died. Now the second one is, I think I'll write it because it's very confusing experiment. Okay, students, simple thing, let's write it here. What happened? He took heat killed S strain and he took the R strain. Okay, first he used protease. In protease, what would have happened? Protein got digested. What is expected? The mice would, here the mice died, because again DNA is still there. Next one is he used the same combination. He used RNase. Here also the mice died. Now the twist comes. He used the same thing with DNase and he noticed that the mice lived. See, if DNA is gone, if DNA is already digested, then definitely the mice will live, no? Because this was a person who got transformed from the S strain to R strain, okay? So, in your textbook, it is given in a very different way, no transformation, no transforming principle and all is given, but this makes more sense. Why? Because whenever we are doing this, in all of these cases, DNA is still there. So the mice will obviously die, right? But in this, what happened? DNA got digested. Now why will the mice die? Because there is nothing that is transforming. There is nothing to get transformed. Protein will not get transformed. RNA will not get transformed. So this was the experiment. Now see, it is very ambiguous. It is not very clear. So this experiment failed. Then the most accepted experiment was your Hershey and Chase experiment. Hershey and Chase experiment was with the use of bacteriophages, E. coli. Infection was done, blending was done, centrifugation was done. What did they do? They took radioactive phosphor, radioactive sulfur. Okay, phosphor is only present in DNA. So if DNA is the genetic material, we will definitely be able to see in the cell if radioactivity got transformed or not. Same thing they did for protein as well. Because protein was in the genetic material, they noticed that the protein was in the supernatant and it was not in the pellet during the centrifugation. We all know genetic material is heavier. Genetic material would have got transformed into the cell, because all this bacteriophages are virus that infects bacteria. The most special character of virus is the moment they infect something, they will leave their genetic material inside the host cell. Yes or no? We all know this about virus. So the same thing happened here. Okay, but the catch was that protein did not, the radioactivity was not seen in the cell which was infected with the protein. Okay, so the same, if you look here, what happened here in this, we got the pellet. In the pellet, we saw the phosphorus. We saw the phosphorus content, and we proved that DNA is the genetic material and DNA is the one that got transformed. Is this clear, students? Is this clear? Might sound a little confusing, but it's not that confusing. Okay? Clear? Now, next one was, our DNA does replication. Now, we proved that DNA is the genetic material. There are some criteria for being the genetic material which you would have seen in your NCERT. Now, the question comes, okay? The question comes, DNA wants to do replication. What type of replication will you do? It can do conservative, disruptive or semi-conservative. So, it went up doing semi-conservative replication. Now, how did I know all of this? I know all of this because of the experiment called as Mendelian and Stahl experiment. Okay, because of Mendelian and Stahl experiment. Stahl experiment. Now, this experiment proved that our DNA, they knew, they were hypothesis that DNA replicates with semi-conservative method. Mendelian and Stahl proved it with the help of this experiment. Okay, this experiment I have done in your diagram based properly. So, I am not going to do in depth, but just understand, first they use N15 heavy nitrogen, then they use N14, they got a hybrid, okay, and they used E. coli because E. coli replicates every 20 minutes and it is easy to take E. coli. If we take some other cells, it would have become more difficult, okay. So, here they take 20 minutes replication, again they did 40 minutes replication, and they saw that they got 2 strands of light and 2 strands of hybrid. This way they were able to prove that our DNA does replicate in a semi-conservative manner because they were one strand of N14, one strand of N15. Hybrid. Semi-conservative means what? One parent DNA, one daughter DNA. One parent DNA, one daughter DNA. Here also same thing happened. Parent was N15, daughter was N14, N14, N15, N14, N15. Like that they tried to do the combination. Easy? Easy? Now, ma'am, yes, you told what type of replication happens. Now the question is, how does replication happens, right? So students, DNA replication happens at your ORI site, that is your origin of replication, and the enzymes used are helicase, DNA polymerase, ligase and primase, okay. This also I have done in the diagram. Please check out the diagram thing. Now look here, what are the things that you need to know from this diagram is that you can see from the 5' to the 3' strand, okay, or your 3' to 5' this way, okay, you will get a continuous synthesis. Why is this continuous synthesis? Because your DNA polymerase will only work on a strand which is from 3' to 5'. Okay, this condition also I mentioned last time, okay. So, this strand became continuous. Here if you see it is a discontinuous strand. What happened? DNA will not work on a strand which is from 3' to which is from 5' to 3'. So primase will get an idea that let's do one thing, let's do replication reverse. Okay, so there are breaks or there are fragments or they are lagging strand. Okay, so you can call it as lagging strand, discontinuous strand, and the other word is Okazaki fragments. Those are fragments, and that is found by Okazaki. So it is called as Okazaki fragments. Got it, got it. And they are in DNA replication, the strand which is used is called the template strand or the parent strand, and the strand which is formed is called the daughter strand. Okay, daughter strand. Clear? Yes, very good. DNA dependent, DNA polymerase, and again, replication happens, students. After replication, what we have to do? Transcription, right? So, transcription happens in a very simple way. Again, all this full chapter is diagrams. Again, I finished the diagrams. Here we have in transcription, promoter initiation, in promoter region, initiation happens. In structural gene region, elongation happens. And in terminator region, termination will happen. Only thing that you need to remember, there is a transcriptional unit. What is this transcriptional unit? With the help of promoter, structural gene and terminator, they form a transcriptional unit. All you need to remember is this much. In transcription, you have to remember about sigma factor. Okay, you have to remember that DNA is getting converted to mRNA. Okay, and the enzyme or the person who's going to help is your DNA polymerase. Sorry, DNA dependent RNA polymerase. Okay, and what you need to know is the rho factor. Rho factor. Now, all of you look here, these are the people who are going to help in transcription. That means you have sigma factor, which will be in the promoter region, which will help the RNA polymerase to move or to initiate the reaction. And structural gene region is the region where your actual mRNA formation will happen. I already told you, 2.2 meters of DNA, I do not want to make mRNA. I only want a small portion to form the mRNA, so that mRNA can form the protein. Yes or no? Now, that portion which will form the mRNA is called as the structural gene portion. In that structural gene portion, elongation will happen. Okay, then we have termination. Termination means we need to end this because RNA polymerase does not know where to end. For that, we are going to take the help of rho factor. Rho factor will go and bind with RNA polymerase and terminate the reaction. So, who and all are we supposed to remember in transcription? DNA gets converted to mRNA. The process is called transcription. We have a transcriptional unit. That means we have promoter, structural gene and terminator. In the promoter region, initiation happens with the help of sigma factor. In the structural gene region, elongation will happen where the actual formation of mRNA happens. In the termination region, there will be rho factor which will bind with RNA polymerase and terminate the reaction. This is all you need to know. This is all you need to know. Do not break your head, okay? UTRs and release factor, all this is there. That is very small. Just go through your NCERT once, okay? Now, look here, after transcription, sometime a problem can happen. The problem is because, students, our gene is not a pure gene. Pure gene means what? They are mixed with something. Okay? They are mixed with something. Got it? Now, mixed with something means what? They have coding and non-coding sequence. Okay? The gene which we have is split gene. They have coding and non-coding sequence. Now, I need something which is only going to help me in coding. What do I mean by coding? Genetic codes have to be read to form the protein. I need coding sequence. To remove the non-coding sequence or to remove the introns, I will do a process called as splicing. I will do a process called as splicing. Splicing is the process by which removal of introns happen. Okay, removal of introns happen. Now, introns are the non-coding sequence. Okay, introns are the non-coding sequence. I do not need introns. I only need exons. So, I will use exons only for my next process. Yes. So, we have polycistronic and monocistronic. Monocistronic means we can only form from one DNA, one mRNA to one protein. Polycistronic means one mRNA can code for multiple proteins, okay, which is not possible in eukaryotes. It's only possible in prokaryotes, okay. That's all. Yes or no? Yes or no? Okay, fine. Then, students, after splicing, okay, after removal of your introns, we do something called as tailing and capping. Capping and tailing. This is basically to protect it from any restriction endonuclease, also to protect it from any introns coming and joining again. So, for that we are going to do capping with the help of methyl guanosine triphosphate. What are we going to do? We are going to use methyl guanosine triphosphate. And students, this is also a technique to understand the 5' end and the 3' end. Okay, this is also a way for us to mark our mRNA 5' to 3' end. Okay, that is what this technique is used. Now, students, look here, capping is done, tailing is done. Tailing is done with the help of poly-A-tain. Now, what is happening? Because of the split gene, we got introns, exons together, no? We got a heteronuclear mRNA. Now, that we made into a pure exon. Only exons are there. Only coding sequences are there. Okay. Now, to this only coding sequenced, beautiful, newly formed mRNA, we will put a head and we will put a tail. Okay. We will put a head and tail. So, we know which is the 5' end, which is the 3' end. On this mRNA, there will be genetic codes. That's why I have kept telling coding sequence, right? AUG will code for methionine, AUG is a start codon. For that, we have a set of codes. To read these codes, we will take the help of tRNA and ribosome. We will take the help of tRNA and ribosome and do our translation. Okay, look here, to do our translation. All of you, this image also we did. Look here, so this is your mRNA, newly formed mRNA. On this you can see some letters written, no? What are those letters? That is your genetic code. How many genetic code we have? 4 into 3, you had 64 genetic code to code for 20 amino acids. Initial plan was to make 20 for 20 amino acids, and this was the most correct number, okay? Correct number. Now, we have 64 codes. Now what we have to do, we are taking the help of a tRNA and a ribosome. This one which you are seeing here is a large unit of ribosome. This one you are seeing here is a small unit of ribosome. This one you are seeing here is your mRNA. Now what happens on the tRNA? There are anti-codon regions. Anti-codon regions will read like this. Okay, AUG is there. It will form the complementary strand to this. Okay, and on the top who will be there? Amino acid acceptor site. Okay, so think of this way, students. tRNA. Here there is your code, how to read the code with the help of an anti-codon. Okay, so here some codes are there. AUG is there. This will form complementary to that. If you look here in the diagram, you will be able to see that. See, look here, it is GCA. Okay, complementary CGU they have made. Okay, after that what is going to happen? On the top head portion of the tRNA, there is an amino acid acceptor site. So in that amino acid acceptor site, one amino acids will come and accept. After accepting, they will form a completely, you can see here, polypeptide chain will be formed. Many proteins will be formed. Now, how do they stop? When do they know that they need to stop? They will stop when they see a stop codon in the end of the mRNA. Okay, so at the end of the mRNA, there will be a stop codon when polypeptide chain should stop forming. Why proteins are formed? To give us different characteristics, right? To give us different phenotype. So all that is done because of protein. So proteins are necessary and protein synthesis should happen. That is the only way by which we did RNA processing. RNA processing means your splicing we did know, that is called as RNA processing. It is not there in prokaryotes, it's only there in eukaryotes. Now here, look here, genetic code, there are some points which you need to remember, very important from board point of view. They are always triplet, that means AUG, triplet, right? UUU, okay, UAG, all triplets. And it is non-ambiguous, degenerate, okay, they comma-less, universal, and again comma-less, they given, universal. Universal means phenylalanine which will code for bacteria will be the same phenylalanine which will, now imagine UUU codes for phenylalanine. Okay, phenylalanine. This will code, this UUU will code phenylalanine in bacteria as well as in human. That means it's universal. Be it the smallest organism, be it the largest organism, UUU will code for the same amino acid from bacteria till the largest organism. That is why it is called. Okay. Now, comma-less means you will not read AUG, comma, UUU. You will read AUG, UUU, UAG, okay, like that it will go. Now, the last concept is lac-operon concept. Again, yesterday I mentioned lac-operon concept. Just understand, when lactose is present and when lactose is absent, the genes can regulate themselves. That means they are feeling like, okay, I don't want to work about this, so they will just switch off. That means during the transcription, translation, gene processing or your RNA processing, any of this time, the gene can decide to stop. They can switch off. Now, how do they switch off? Because of some conditions. One of the things that you have to study in prokaryotes, it's your lac-operon concept. That means when lactose is present and when lactose is absent. When lactose is present, that means when inducer is present, what is going to happen? Inducer will bind with the repressor and not allow this repressor molecule to go and bind with the operator. This way, the RNA polymerase which is coming to do transcription will be happily doing transcription. Okay. When lactose is absent, that means inducer is not there. Repressor will be there. Repressor will go and block the operator and the RNA polymerase which is coming will be very disappointed and it will not be able to do transcription. That means the gene has decided to get switch off before transcription. That's all. Okay. Then LAC-Z, LAC-Y and LAC-A. LAC-Z will cause release beta-galactosidase which will break down lactose to galactose and glucose. Y will release permease which will help in increasing the permeability of the E. coli cells to absorb more lactose. Then we have transacetylase which one's function is still not known, but they definitely help the Z and Y to release their enzymes and do that kind of function. Okay, is this clear? Now, Yes. So students, we have structural gene, operator. O stands for operator gene, P stands for promoter gene, repressor is there and inducer is there. That's all. Now, Human Genome Project is a very in-depth topic. You have to know the goals and the salient features. Okay, you have to know the goals and the salient features. 1 second, students. Did we miss something? No, we did not miss anything. No? Okay, look here. Your Human Genome Project is to identify all the estimated genes in the human DNA to determine the sequence of 3 million base pairs. To understand, basically to study what is this fellow doing, what is this fellow doing, will they become chromosome, will they become sex chromosome, will they become normal chromosome. To do all of this, we need a system. Okay, we cannot carry books and walk around. Oh, my sickle cell anemia happened in this, here point mutation happened. We cannot walk around like that. For that, we need a complete system. That system is given by our HGP. Okay, other than HGP, we also have DNA fingerprinting. Yesterday, I explained it with the help of a diagram. Okay, DNA fingerprinting. So keep that in mind. Keep that in mind. Okay. Students, that's all. This chapter is very simple and easy. Do not have that fear to the chapter. If you have the fear, then all the chapters are difficult. Evolution also will be difficult. Principles of inheritance also will be difficult. Human health also will be difficult. Do not think the chapter is difficult. It's just concepts. Learn from the diagrams. That's all. Okay. Yes. I'm tired.
No, ma'am. The Josh will be high because students.
No, students are very low. Low Josh.
Low Josh, no?
That's why my Josh also.
This is what happens, students. When your Josh is going to be low, even our Josh is going to be low. It directly correlates. Direct relation. So can we start? This is my phone. Let's go to the chat here. So can we start, students? Can we start with the next chapter? Ma'am to the pen. No, ma'am didn't take the pen. That's going to be your biotechnology principles and processes. Can we start? Biotechnology principles and processes. Can we? Yes. Welcome, 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 welcome. Welcome to me. Yes. So students, what is so difficult about this chapter? Tell me in the chat. What do you not understand in this chapter? Like there are a few concepts in this chapter where students actually struggle and the question actually comes. That is going to be your students still don't understand PCR. PCR is such a very easy concept. I don't know why students still struggle in PCR. There is concept called as your recognition sequence. What is recognition sequence? You should know. You should also know what is the meaning of a vector. You should know what is the meaning of restriction enzymes. Very, very easy. Briefly, I'll explain you every single point here. Yes, briefly I'll explain you every single point here. And in detail for NEET students, we'll be doing later. Unfortunately, we could not complete only this chapter from your entire of your class 12th before your boards. But it's fine. I know because I know our students would have studied this. But we'll do a complete small cutie revision now. Okay, one small revision we'll do. Okay. Now, what is the first thing which comes to your mind? Students, the entire of your biotechnology can be divided into two parts. Yes. The first part of your biotechnology is going to be genetic engineering. And the second part is going to be bioprocess engineering. The second part is going to be bioprocess engineering. Now, students, what is the main thing which comes to your genetic engineering? What do you do in genetic engineering? That one question comes in, sir. What are the main processes in the case of genetic engineering? So, students, there are three main processes in genetic engineering. The first one is your rDNA technology. The first one is your rDNA technology, also called as your recombinant DNA technology. Also called as what? Recombinant DNA technology. The second one is called as your gene cloning. The third one is your gene transfer. Now, what do you do in recombinant DNA technology or rDNA technology? In recombinant DNA technology, we basically take a foreign DNA, we take a vector, we take the foreign DNA and we add it inside the vector. Then we take this vector and we send it inside the host. I'll repeat again. Remember when you were in class 6th, 7th or 2nd, 3rd, you are getting ready for the school. You are the foreign DNA. You are the foreign DNA that is getting ready for the school. How do you go to the school? You will take a taxi and you go to the school. The taxi here is going to be your vector. Vector is your taxi now. So you are the foreign DNA. You are sitting inside the taxi. That is vector. Take the foreign DNA, added inside the vector. Now where are you going? You are going to the school. Now you take this vector, you send it to school. Now what is going to be the school here? The school is going to be the host here. So you take a foreign DNA, add it inside the vector, now send the vector to the host. That is the entire process of your rDNA technology, also called as recombinant DNA. That's how you make the recombinant DNA. Yes, sir. Now students, can we build a house without the tools? Can we build a big house without using bricks, without using cement, without using manual labor? Can you build a house? No, you cannot. You cannot, right? So to build a house, do you need certain tools to build a house? Yes. So students, if you want to do the rDNA technology, if you want to do recombinant DNA technology, you will need some tools. You will need some tools. Now, what are these tools? That is what we're going to study here. So, what the first main tool, students? The first main tool is going to be your restriction enzymes. What is going to be? The first one is going to be your restriction enzyme. Now, students, what is a restriction enzyme? The first thing, briefly I'll explain. Listen to me very carefully. Restriction enzyme is also called as your molecular scissor. Restriction enzyme is also called as what? Molecular scissor, which is mainly obtained from bacteria. Now, why does the bacteria has this scissor? Bacteria has a scissor against for protection. For protection. So basically, we take this restriction enzyme, also called as molecular scissor, to cut the DNA. What is the use? To cut the DNA. We have something called as endo restriction enzymes and also exo restriction enzymes. Endo and exo. Endo means within, exo means outside cutting. Clear? Now what happens here? Listen to this. Look at this diagram very carefully. The enzyme cuts both DNA strands at the same site. That is, we are using the same enzyme to cut two different fragments of DNA. One DNA is going to be your vector DNA. The other DNA is going to be your foreign DNA. Remember, the foreign DNA is you. Basically, we are cutting you. We are cutting you and we are also cutting the taxi. Why? To make sure you and the taxi join together. To make sure we are cutting the two DNAs, to make sure the vector DNA and foreign DNA join together. How do they join? Listen to me very carefully. Students, if you look at the vector DNA and the host DNA, there is a small site here called as your palindromic sequence. Yes, there is something called as what? Palindromic sequence. Now tell me in the chat, what is this palindromic sequence? Tell me in the chat quickly. What is this palindromic sequence? Palindromic sequence is a sequence which can be read on both the sides. It will result the same. Example, Malayalam. If you read from the front or the back, the meaning is going to be the same. Now here students, we have a palindromic sequence also called as your recognition sequence. That is GAATTC. Now students, this particular recognition sequence is for specifically for one enzyme. That is, it is very specific to one restriction enzyme. That is EcoR1. This example is for EcoR1. Example is for what? EcoR1. Now this EcoR1 is very special. It will exactly cut here. Can you see? Between G and A. Between G and C here. Between G and C it will cut here. It's cutting here. Two cuts are made. Now students, one question arises. Do we take the same restriction enzyme to cut foreign DNA and also to cut your vector DNA? The answer is going to be yes. We have to take the same restriction enzyme. Why? Because in order to obtain the sticky ends, these are called as what? Sticky ends. Now, what if we use a different types of restriction enzyme imagine we're using one for your foreign DNA and some other restriction enzyme for vector DNA. If you use different ones, you will not get what? You will not get complementary strands. You will not get complementary strands. So in order to get the complementary strands like this, you should use a same type of restriction enzyme. So if they are complementary to each other, if these two sticky ends are complementary to each other, can they join? Just tell me in the chat, can they join? The answer would be yes, sir. They can join very easily since they are very complementary, which are also called as sticky ends or overhangs. So this will join with this. That is, the joining is done by DNA ligase. DNA ligase will fuse them together. DNA ligase will fuse them together, and that's how you are getting inside the taxi. That is how your alien DNA is fusing with vector DNA. Done. The first part of the restriction enzyme is done. Clear? That is your restriction enzyme. Clear? Different fragments. Now, how do you separate these restriction enzymes? Is there a process? That is, the cut DNA fragments are separated by gel electrophoresis. What is it? The separation is done with the help of gel electrophoresis. Now, how are we separating them? The separation is done by electrophoresis. In gel electrophoresis, the principle behind gel electrophoresis is very simple. The principle here is the lighter the weight, more distance it will travel. That is, DNA is said to be negatively charged. DNA is negatively charged. It will move towards the anode. And the smaller fragments will be moved front. The heavy fragments will be left back. So that's how you obtain the DNA fragments. The dye it is used here is ethidium bromide because it will help us to visualize the DNA later on. And if you are in my Telegram channel, I have uploaded a video. Actual image of a gel electrophoresis. That is, after radiography. That is, after autoradiography, you will get one image. So on my Telegram channel, I uploaded an image which was done by me only. So I have done this experiment in my MSc. The experiment I have added a photo. So if you can go check it out also. Okay. So that was your gel electrophoresis. Now, students, I spoke about vector, students. I spoke about vector from that time. But students, what is a vector? Vector is also a type of your plasmid. Now, what is a plasmid? Plasmid is nothing but a self-replicating extra chromosomal DNA. Yes, plasmid is a extra chromosomal self-replicating DNA. The example which is given your NCERT is your PBR 322. The example given here is PBR 322. Now, what is this P here? The P stands for plasmid, the B stands for Bolivar, R stands for Rodriguez, and 322 stands for your order of discovery. Now, what is so special about your PBR 322? The speciality about your PBR 322 is it has in total of 8 cloning sites. Now, this is not 8 cloning sites. There are in total of 8 cloning sites. 2, 3, 4, 5, 6, 7, 8. 8 cloning sites. Now, apart from 8 cloning sites, it also has 2 selectable markers. It has what? 2 selectable markers. The selectable markers are basically your tetracycline resistance gene and also ampicillin resistance gene. Now, why are they called selectable markers? The reason they are called as selectable markers is because they help in segregation from transformant and non-transformant. Now, what is this transformant? Transformant is basically a type of type of your organism in which a recombinant DNA is present. It is transformant. Non-transformant is not required, which does not have a gene of interest. Non-transformant does not have a gene of interest. Clear? So we have ampicillin and tetracycline selectable markers here. Now, what are the features of a vector? Now, they can ask you a question. What are the features of a vector? There are 3 main features. It should have a ori site. Yes, students, it should have a origin of replication. Can you see here? Ori site is here. Origin of replication, which will basically help in replication, starting of replication. Then we also have something called as your ROP here. What is ROP? ROP is basically repressor of protein. What is it? Repressor of protein, which basically helps in replication. Producer type of protein. Then it should have a selectable marker. We learned here. Tetracycline as well as ampicillin selectable markers. Then it should also have few cloning sites. Does it have cloning sites? Yes, sir, it has cloning sites. So will you be able to write all these points? Will you be able to remember all these points? Yes, sir, you will be able to remember all these points. Very important. Now students, the next step is you have your recombinant DNA is there. Recombinant DNA is there. Now you need to take this recombinant DNA and we need to add it inside the host, right? You are sitting in a taxi. Now you will be like, nah, I will not go to school today. Will you make the tantrum? Will you make the tantrum that no, I don't want to go to school today. I will fall sick. Our bacterial DNA, our recombinant DNA will also show some tantrum. The DNA will be like, no, not today. I will not go inside the bacterial, I will not go inside the plasmid. They will make sure some tantrum. It will not become transformant. So how do you make it transformant? How do you make sure it enters inside the host? Yes, how do you make it? You will make tantrums. How will it make? So basically your DNA is said to be what? Hydrophilic in nature. Where's the pen? It is said to be hydrophilic in nature. Yes, DNA is hydrophilic in nature. It will not cross the barrier and go. It will not cross the barrier and go. But you have to make it, you have to force it. Like, go today, you have to go. You have to force it. Basically, you're supposed to force the bacteria to take up our DNA. How are you forcing? The first method of forcing is by using divalent cations. How are you forcing? You are like, take this cation, divalent cation. Yes, we are giving it not selectable markers. Kaushal, selectable markers is for differentiating. How are we making it host specific? You're using divalent cations like your calcium, Ca2+. So as soon as you add Ca2+, will there be holes in the cell wall layer? If there are holes in the cell wall layer, can the DNA now quietly go inside the holes and get into the host? That is the first method. That is the first method, that is, you are making the host is made compatible. The first method is you are using divalent cations and the host is made compatible. Now, how are you making it compatible? Basically, you are giving it a shock. Boom. Imagine your mother is telling you, I am not taking you to school. I am taking you to a park. Will you happily go? Yes, you will happily go. Suddenly, as soon as you reach the school, you will be like, oh, you will become shocked suddenly. No, suddenly the school will come, you will become shocked. That is what we are doing. We are shocked. We are doing something called as heat shock method. We are doing something called as what? Heat shock method. Now, what are we doing? We are making a bacteria shock. How are we making it shock? Basically, you take the bacteria, you take your DNA, and you keep it in a cold condition, ice cold condition. You keep it in ice cold condition. From ice cold condition, you're taking it, keeping it in heat condition suddenly. Full heat. It is a shock now. What is happening? Now, again, you take from the heat condition, again back to cold condition. Yes, again back to cold condition. Your mother rocked, you shocked. You became shocked, right? That is what you're doing. Here, bacteria is shocked. DNA is rocking now. Okay, DNA is going inside now, becoming host compatible. That is first method. The other method is very simple. Take one gun and shoot it. The other method is take the gun and shoot it. That is your gene gun. That is your gene gun. That is gene gun, also called as your, and also ballistics. Gene gun as well as ballistic. Take your piece of DNA and shoot it inside the host. Direct. The other one is your ballistic method. That is, cells are bombarded with high velocity microparticles of gold and tungsten coated with DNA. Basically, you have a chamber like this. You have a chamber like this in which we have your DNA here. We have your host plant. Look at the leaf. High velocity particles are sent here. High velocity particles are sent. In that high velocity particles, we coat DNA also with it. We coat the DNA also with it, and as soon as the particles are coming here, this one barrier here, the particles will be left here, but the DNA will go inside. The DNA will go inside. That is your ballistic method. That is your ballistic method. Don't worry, students. Gopika Ma'am or I, any one of us will be teaching this in detail for all the NEET students with a diagram. With a diagram, the concept will be clear. Right now, just the basics. Any one of us can teach this chapter. Both of us are experienced in biology. That is not just zoology or botany. Okay. So any one of us will be teaching this chapter. Remember it. Then we have the microinjection, which I already told you. Our DNA is directly injected into the nucleus of the animal cell. Nucleus of the animal cell. The other method is going to be your using a particular virus. That is, vectors like agrobacterium 2, not bacteria, Agrobacterium tumefaciens, when infect the cell, transform the recombinant DNA into the host. Basically, what we are doing, we are taking a bacteria, we are putting a gene of interest inside it, and we are infecting it. Now, once the infection is happening, our DNA is also replicating. That is your infection method. Basically, you take your bacteria, add the DNA to it, make the bacteria enter inside the host. Once it enters into the host, it will start to replicate there. Now, once the bacteria is replicating, DNA is replicating. Simple. Yes. Agrobacterium tumefaciens. Now, students, did we leave any point here? Are you noticing? In a much more fun way, we are actually finishing the chapter. We have one more last point to go. That is going to be your process of your RDN technology. What are the processes in your RDN technology, students? What are different processes in your RDN technology? Clear? Now listen to me very carefully. Oh, my phone is dying. The first step, can anyone tell me which is the first step in your RDN technology? Students, clear, right? Are you understanding? This is how we learn biotech. Biotech, see students, the reason I am able to explain biotech so fluently and how I know this, because I have actually done all of these experiments, all the way from isolation of DNA, PCR, every single thing we used to do in the lab. I used to sit in the lab and actually sit and do PCR. I used to sit in front of the PCR machine and wait for my DNA to amplify. When you do the experiments, it gets stuck in your mind. Okay, when you do the experiments, it gets stuck in your mind. So the first step, students, the first step in your RDNA technology, the first step in your RDNA technology is isolation of DNA. What is it? Isolation of DNA. Now, how do you isolate DNA? Tell me, where do you get the DNA from? You can take the DNA from a bacteria. You can take a DNA from the plant. You can take a DNA from an animal cell. You take the DNA. Now, once you take the DNA, does the DNA has a bacteria? Will have all the things. Take a bacterial cell. It will have DNA, RNA, proteins, and cell wall. Yes, sir, it will have cell wall. So, how do you break the cell wall now? Even in the case of plants, will it have a cell wall? The plant cell will obviously have a cell wall. So, how do you break the cell wall? How do you break it? Can anyone tell me in the chat? Anyone? Students, remember, in the case of your bacteria, you use something called as lysozyme. Which will break the cell wall. Clear? In the case of your plants, we use cellulose to break the cell wall. And in the case of animals, we use chitinase to break the plasma membrane and other layers. Okay. Now, students, the next question is, after your outer layer is broken, now we need to remove all the RNA. What do you use? We use something called as RNases to remove all the RNA. Done. Then we have to remove all the protein. We use proteinases to remove all the protein. Protein also removed. Now, what is left over? The leftover is DNA. So, how do you extract the DNA? We use something called as chilled ethanol. You add chilled ethanol. You add, and you spool out the DNA. So, the first step is what? The first step is going to be your isolation of DNA. Now, you have your DNA, students. Done. Now, what would you do? Take the DNA. Imagine it's like cooking. This entire process is like cooking. Get your chicken. Get your chicken piece. Cut the chicken piece now. How do you cut the chicken piece now? With a knife or a scissor. Now, what do you do now? Second step. The second step is going to be your using restriction enzymes to cut the piece of DNA. Cut. Done. That is your restriction enzyme is done. Now, students, once you cut the DNA, imagine you only have around 1 or 2 pieces of DNA. Is that 1 piece and 2 pieces of DNA enough for your experiment? Tell me, if you have only 1 piece of chicken, are you happy with that chicken piece? No. Imagine you have 10 people in your house. 1 piece will be enough for everyone? No. So, what do you do now? You bring more piece of chicken. Yes. So, in the case of your processes, we take that fragments of DNA and we amplify the DNA. What do? We amplify the DNA. How do we amplify? With the help of PCR, polymerase chain reaction. Students, are you understanding? This is all like a story. Remember like a story and you will not forget. Okay. Next one is amplification. How do you do amplification, students? No, separation is later on. Twinkle, separation is later on. First, separation has to be done. Okay, suppression is done. After suppression, we have amplification. Now, what do we do in amplification? After suppression, we are doing amplification now. Amplification, we have something called as PCR. Can anyone tell me the steps in PCR? Not quickly tell me the 3 steps in PCR. The 3 steps in PCR is denaturation, annealing and extension. Denaturation, annealing, extension. Students, in my diagram video, I have explained the entire steps by steps. So please go watch it. Now I won't be explaining the entire thing. I'll tell you what you need to remember for examination. What you need to remember here is what? Polymerase chain reaction. The only point you need to remember here is Taq polymerase. Remember the Taq polymerase, you're set to go. Taq polymerase is an enzyme which is obtained from a bacteria called as Thermus aquaticus. Obtained from Thermus aquaticus. Basically, this enzyme does not denature in the high temperature, because we know denaturation, annealing, everything is done in high temperatures. Taq polymerase, remember that point. Okay. Now, once you have this cutting is done, amplification is done, what do we do? Recombination, RDNA technology. We learned here, right? Process in your RDNA technology. Now you do RDNA technology now. What do you do? RDNA technology. All of you know the steps in RDNA technology. That is, take your foreign DNA, take your vector, join together. Then take the vector, send it to the host, that is called as transformation. Next step you do what? Transformation. Transfer of RDNA into the host, that is called as what? Transformation. Now students, once transformation is over, you wait now. What do you wait? You wait for your result. Now once you give your NEET examination, you wait for result? No. Once you give a board examination, you have result. Oh my God, when is the result coming? You wait like that? No. Now also you are waiting now for the result to happen. That is called as you are obtaining foreign gene products by bioreactors. So students, you can obtain the result in a much more small scale in a lab. But remember, the entire biotechnology is divided into 2 parts. One is your genetic engineering, which we just learned. The second step is what? Commercialization, selling the product, making the product. That is going to be your bioprocess engineering. In bioprocess engineering, we mainly have the bioreactors. What do we have? We have the bioreactors. That is, whatever you do, right? Whatever you do in your lab to field, if it has to go, we need more quantity. Imagine you are making chicken now, chicken biryani for 1,000 people, or 10,000 people. Will you make the chicken biryani in a small cooker? No. You will take a big vessel, right? That is your big vessel. Chicken biryani vessel is your bioreactors. Bioreactors are the big vessels. They are basically big vessels. They basically maintain in the optimum temperature, pressure, and everything. And you add all your raw material to it, and it will nicely make your products. Nicely make your products here. So students, in total we have 2 types of bioreactors. One is called as your stir tank bioreactor. The other one is called your sparged bioreactor. Now what is the difference? The main difference between 2 of them is, in sparged, can you see, oxygen is sent from the bottom. Oxygen is sent from the bottom. In the case of your stir tank bioreactor, we have fans. Fans are nicely rotating here. So bioreactors will make sure we get the product in a much more larger quantity. So with that being said, students, the entire biotechnology chapter is over with the help of a small story. Okay. That is the end of bioreactor. Did you notice? We did every single, see, this is a very interesting chapter. And if you really know what to study here, everything becomes very, very easy here. Okay. Clear? Students, do you need a break or do you want us to continue? Which bioreactor is better, students? If you ask me, it is going to be your sparged, because bubbles dramatically increase the oxygen transfer area. Yes, more important. Do you need a break? The entire they need a break, right? So let's take a 15 minutes break. We also eat something in them. I will faint. I'm also very hungry. My stomach is like growling already.
Students, we came early morning, so
yeah, we came here at 9 o'clock. So, see, students, you think all this can happen in one day? No, students. in order for us to teach continuously like this, right? It takes a lot of effort for us to go through so much of notes and remember all of these points so our students can get the benefits the best, right? For that very reason, we make sure that the content is on point and all of you can benefit from it. Okay? I was very hungry now. So students, we'll take a small break. After that small break until 2:15, ma'am? 2:15? That's 2:15. Break till. Oh, lessons back. I didn't read. Nice handwriting. Okay, students. So with that being said, students, like the video. If you're not liking the video, it's going to be very sad for us because all the effort we see, right? All the effort which we are doing, we are teaching you the entire chapter in 30 minutes. Every chapter 30 minutes. All that will go in vain because the more you like the video, the more it will go reach. That's your YouTube algorithm. Yeah. That's the reason we tell you to like the video is not because we want to see the like there. We want to see, but the more you like the video, the more students will benefit from this because the more you share the knowledge, more it grows. So we are sharing the knowledge here for you to understand. So you also should share this video as much as possible. So go on. Once the break is over, I want more students to attend the class, more share the video, like the video, and we'll be back at 2:15. Okay?
Sure, let's run.
Let's run, ma'am. Order the food now. We order. Okay. Friends, we'll be back in 2:15. Okay? Bye-bye. Evolution. After that, principles, then application part. I'll teach application. After application. Break. Friends, break.
Okay, first students, in evolution, you have to know the theories of evolution, right? So in theories of evolution, I made you study all the theories of evolution, and today I noticed that the notes that have been shared on the Telegram does not have the written part, so I'll share another notes to all of you, okay? Okay, another notes to all of you. Is the energy still there or all of you are a little sleepy? Students, we will complete it in another 1 hour, max to max 1 hour, we'll be able to complete it. Okay, so all of you stay energetic, be like, yes, I'm completing the entire 12th portion revision with this. Okay, now all of you look here, there are some theories of evolution like theory of biogenesis, that means life comes from pre-existing life. Okay, that means life comes from pre-existing life, that's all. Okay, that is what the theory of biogenesis. Then we have theory of chemical evolution, which was given by Oparin and Haldane. This was the most revolutionary change in the term of evolution. Okay, in the term of evolution. Yes or no? Yes, okay. So, this theory told that life comes from organic components which are made up of inorganic constituents like your RNA, protein, amino acids, all this. And to prove this, there was an experiment done by S.L. Miller's experiment in the year 1953, which proved this, that actually chemical evolution happens. That means life happened because of some changes in the chemicals, some inorganic organic changes. Okay, next thing is, students, the theory of panspermia. This is still believed by most of the astronomers that there were some seeds, okay, seeds of life that came from another planet, okay, that came from another planet, or that is in the universe, that came from one planet to our earth. Okay, and that is called as theory of panspermia, which is also said that seed of life. Okay, seed of life, and it is still believed by most of the astronomers, most of the Greek thinkers, because they believe that there was seed of life in the universe and it got transported from one planet to another planet. Okay, one planet to another planet, that is from some planet it has come to earth, and that is how first life happened. Okay, now theory of spontaneous generation or abiogenesis, this was proven again by your Reddy and Splancy. They proved that, they showed that, you know, life came from when they kept the dead yeast and they, you know, opened it and kept, they saw some moths and flies on it. When they closed it, they noticed that flies and moths disappeared. And when they kept a muslin cloth or a, you know, like a cotton cloth on top, they noticed that moths or flies were on top of that. So they understood that some kind of spontaneous, you know, generation is happening, and it just, life is just happening like that, okay? And then you also have theory of special creations, okay, which you shouldn't forget. Theory of catatropism, that also you should remember. That means whenever there is a death of a full generation, there is a space created and a new generation or a new species will come into that place, okay. Same way, theory of special creation, that God created the entire universe in 6 days, and on the 7th day, okay, 7th day, human happened, or man, life happened, okay. Yes. Then students, this was disapproved by your Louis Pasteur. Now, theory of pre-existing, that means life comes from pre-existing things, came from your Louis Pasteur. He told that life is coming from pre-existing thing. And during this chemical evolution also, Oparin and Halden also made the statement that life comes from pre-existing organic, inorganic components, okay. Now, let's look at S.L. Miller's experiment. So, students, what did they do? They knew that the atmosphere had reducing power. That is because of the gases like CH3, NH3, methane, ammonia, water vapor and hydrogen, okay. This was an experimental setup, and this was a fake lightning that was created with the help of electrodes to prove that chemical evolution is with which life happens. At the end of this experiment, they were able to find out amino acids like alanine, okay, aspartic acid. Students, this I have explained in the diagrams, okay, aspartic acid and glycine. So, like this, many people did this similar setup experiment, and they got sugars, they got fats, they got nitrogen bases, and our DNA is also made up of nitrogen bases. Protein is made up of amino acids. So, now it makes sense, no, that life came from some inorganic components, organic components, simple get broken down to complex and much more, okay, clear? Yes, awesome, okay, at 800 degree Celsius, okay, and the reducing atmosphere was created by which 4 gases? All of you. Then, students, some evidences that prove that, you know, evolution happened was first your paleontological evidence. That means we got some fossils that showed that yes, evolution happened, our ancestors look like this, that kind of an evolution. That means from fossils we understood paleontological, okay, and it could be in the form of impressions, or it could be in the form of like if the dead or the, you know, body was there in very cold temperature, then decomposition will not happen. That kind of ideas also we were able to understand. Amazing, amazing. CH4, H2, your methane, ammonia, water vapor, hydrogen cause the reducing atmospheric temperature, okay. We want an oxidizing temperature. We need oxygen for survival, okay. Now, students, look here. Paleontological evidence, that means with the help of fossils. Then we got some morphological and anatomical evidence, that is the presence of homologous and analogous organ. I already gave a trick to remember. Homologous you will remember with HOCDF, no, HOCFD. One second. You remember this with HOCFD, and here analogous you will remember with AC. Oscar did a fantastic show, okay. Oscar did a fantastic show. What is it? Hi. So, students, this is hi, Oscar called Friends director. So, the reply is Oscar did a fantastic show. This is what H stands for homologous, O stands for origin, origin is common, function is different, okay. Here Oscar did a fantastic show. Analogous, origin different, function same. Okay, please write it down somewhere in one side. This is the only way you can remember it, because in exam you will have so many things in your mind you will not be able to remember. So, they came up with some morphological and anatomical, that is your analogous and homologous. Then, biogeographical, that was proved by adaptive radiation, that even though organisms are maybe a part of Australia, there were marsupials in Australia, they went to different place and they got modified. Okay, they went to different place and got modified. Okay. Then adaptive radiation also proved your Darwin's finches, marsupial radiation and placental mammals. So what did they do, students? Basically, Okay, basically Darwin's finches was that he noticed that in the Galapagos Island, when he was roaming around with, which was the ship that he used? Tell me the name of the ship. Okay, name of the ship which he used. Darwin noticed that even though all of these finches were seed eating, when they moved here and there, one became fruit eating, one became insectivorous, one became, sharp eaters. So their beaks got modified differently, and this proved that even though if they are of the same species, if they have moved in the same geographical area, they will get adapted to the condition. Okay, very good. HMS Beagle was the ship in which your Darwin roamed around the South America, and he proved Darwin's finches as well as your natural selection. Okay, then student, marsupial radiation, we know that it migrated to different place, it moved to different place and became different forms. Okay, this you have to remember by yourself, and I give you a trick to remember placental mammals, so that same trick you can use for marsupials. What is the trick? Ma'am, trick was ma'am likes sweets and burgers on weekend. Okay, on weekend. What is this? Your M stands for mole, other A stands for anteater, M stands for mouse, L stands for lemur, S stands for your, what is that in this one? Okay, you can remember sugar gliders if you want, or else here B stands for, I forgot the names of them, but then you can remember these both. All of you tell me what is B stands for? Yes, very good. Ma'am likes sweets and burgers on weekend. Yes, S stands for squirrel. Very good. This is how you should help a teacher. Okay. Okay, B stands for, and W stands for wombat. Okay, wombat. B, all of you tell me quickly. B. Yes, bobcat. Awesome. Bobcat. Okay. This way you will be able to remember half of this. Okay, ma'am likes sweets and burgers on weekend. You can remember these and you can connect it with these. And placental mammals and your marsupial or Australian marsupials show convergent evolution. That means they would have started off differently, but they come to the same point. Like yesterday I gave you an example. You're all NEET students. You're from different states, different hometowns, different cities, different lifestyles. But you all come to our Vedantu NEET English with the same motto. Right, that is to crack NEET, to clear both. So that is your convergent evolution. Okay, very important question. Now, look here students, embryological we did. Now, here if you see theories of biological evidences, that is your Lamarckism and dwarfism. Lamarckism has two main points. Who can tell me the two main points? Okay, two main things in Lamarckism is your use and deuse. Use and disuse, okay, and acquired characters. These were the two main points of Lamarckism. Use and disuse means if I keep using my right hand becomes more stronger. If I stop using my left hand becomes weaker. So whatever organ or whichever thing that you keep using, it becomes stronger, and which you don't use, it becomes weaker. This was the first, postulate of Lamarckism. Second thing is, acquired characters get inherited, okay, from generation to generation. Now, there is a catch about it. What is the catch, students? All of you tell me, can acquired character get transferred from one generation to other generation? Can it get transferred, or is there a condition? All of you tell me, is there a condition or they can get transferred? That is a question to all of you. Then, Darwinism, which was basically natural selection. Okay, natural selection. This also had two things, that is branching descent. Okay, this one I think dwarfism, sorry, why am I telling dwarfism? Darwinism, sorry students, Darwinism had two things, that is branching descent and natural selection. Okay, branching descent and natural selection. Yes, let's see who. Very good, awesome. Drishti, sweetie, Twinkle, amazing. Students, what has happened? Only if the germ plasm changes, then only acquired inheritance will be carried to the next generation. This is exactly what happened in giraffe. Giraffe was eating from down, like from shrubs and herbs. Later, when the height of the trees kept increasing, the giraffe kept stretching his neck and now it has a long neck. That got transferred to the next generation because it was a change in the germplasm and not in the somatic cell. Okay, awesome, very good. Branching descent and natural selection were the two main points of your Darwinism. That means the strongest will survive. That's all. The person who does variation will survive. The person who is going to fight all the environmental condition, they will survive. This is what is natural selection. Explanation is with the help of a moth, dark-winged and white-winged moth, that is industrial melanism. Okay, industrial melanism. Now, single-step mutation, students, what is happening? When there is a sudden change, okay, when there is a sudden change from, say, one generation or to a next generation, we call this as saltation. What do we call it as? Saltation by Hugo de Varies. So, even though Darwin told about natural selection, even though Darwin told about variation, he did not prove variation. Who proved variation? Variation was proved by Hugo de Varies with the help of mutation. He told variation, something is not there, it is mutation. It is a directionless random change. It is directionless and it can be random. Sometimes it can be even one step. That means a jump can happen and that is called as saltation. Are we good to go? Are we good to go? Okay, that is called as saltation. Then the most important topic of your evolution is your Hardy-Weinberg principle. Simply says P square plus 2PQ plus Q square. This is the formula. And how did they find this P square and 2P square? That is, if the allelic frequency does not change, if the allelic frequency is constant. How is that possible? That means they assumed if evolution does not happen, ideal condition, evolution does not happen, then P square plus 2PQ plus Q square is equal to 1. This is ideal condition. So, what were the factors that could affect Hardy-Weinberg principle? Simple. Gene migration, evolution happens. Genetic drift, change in allelic frequency. Mutation, variation happens. Genetic recombination, variation happens. Natural selection, again it happens. So, Hardy-Weinberg principle is a very ideal condition. When evolution does not happen, then we will get P square plus 2PQ plus Q square is equal to 1. But if evolution happens, then Hardy-Weinberg principle does not stand. Why? Because he says that if allelic frequency is constant, how can gene pool of a place be the same? Migrations happen, immigrations happen, genetic drift happen, all this happens, right? Every time same set of people will be there? No. Things will keep changing. So, this is a very ideal situation. So, the question can be factors that change the Hardy-Weinberg principle: gene migration, genetic drift, mutation, gene recombination and natural selection. Students, please keep this in mind. Then, human evolution, okay? Human evolution is super, sorry. Human evolution is super easy. That is, how are you going to remember this? Draw hen. You are going to remember draw hen. This is a type of hen. Okay. So, we have Dryopithecus, Ramapithecus, Australopithecus, Homo habilis, Homo erectus, Homo sapiens. That is your primitive man who thought that bodies have to be covered and the dead bodies have to be buried. Okay. Draw hen. This is how you are going to remember. Okay. And students, can you all tell me the brain capacity? Very important question. Brain capacity. Yes. What is the brain capacity here? So, it starts from Homo habilis. Yes. Homo erectus and this one. So, here it starts from 650 cc to 800 cc. Here it is 900 cc. Here it is 1400 cc. Okay, this is what you have to remember. This is the brain capacity. Are we good? Awesome. 650 cc. Yes. Okay, Homo habilis 650 to 800. Java man, it was found in Java. Okay, and 900 cc, and your primitive man was around 1400 cc. Understood? Easy? Done? Done with the chapter? Let's check if you missed something. Reduce, bro, reduce. We did everything, right? Yes, we did. Yes, good to go.
Yeah, it was too hot. It was the bomb. The thing is in the fridge, there was nothing.
Yeah, so we switched it off and
No, nothing's there in the fridge. Yes, students, welcome back. This is Basparazzo, welcoming back to all of you to Vedantu Neet English. Huh? No, only one.
then after
Human health. Where's the pen, ma'am?
I left it there. See on the sofa.
Where? This pen?
That only I'm using.
Where's the other pen? Okay. Welcome. Sam, ma'am, are you Tamilian? Ma'am is not Tamilian, ma'am is Malayali, ma'am is from Kerala. Well, and I'm Kannadiga. Let's got it. It's fine, keep it. And I'm from Bengaluru. So can we start, students? Can we start with the evolution now? Oh, what evolution? The evolution is over. Wait. Can we start with the application chapter? Can we start with the biotech and its application? Can we start? Biotechnology and its application. Hello. Can we start with, can you hear me? Can we start? That is biotechnology and its application. Can we start? Yes. So students, we learnt about principles and process, done. Now we need to apply whatever we learned in the previous chapter to this chapter. So what are the different applications of your different biotechnological applications? For example, the first and the foremost is going to be in agriculture. So the first application which we will be studying is the agriculture. Now student, tell me, all of you know Green Revolution? Yes, all of you know Green Revolution, right? Green Revolution was revolutionized by M.S. Swaminathan, which basically did tripling of the food. So whatever food was made in India, three times the food increased. That is, if you imagine we are making 100 tons of rice, now we were making 300 tons of rice, and that rice was also not enough for Indians, especially South Indians, because we love rice, we love curd rice. I love curd rice. I do love curd rice. So student, here even after the Green Revolution, it was not enough. So what do we do? Later came the technology of your biotech. So we were like, instead of just Green Revolution, why don't we use technology? That is biotechnology in the case of your agriculture. Now, what was the main use of this biotechnology in agriculture? The first main was it made crops more tolerant to earth body stresses. That could be drought, salinity, or any kind of earth body stresses. Our plants became tolerant. Our plants are like, no, I will not move, I will not die, I will not do anything, whatever happens. And that is because of the biotechnology. Then we have reduced resilience on chemical pesticide. Yes, that is reduced resilience on chemicals. We started using chemical pesticide less. Then we have helps to reduce post-harvest losses. That is in threshing, winnowing. All of you know that, right? Threshing and winnowing and storage in silos and miles, right? Storage. All the post-harvest loss also became less. Example, I'll tell you, flavor saver tomato. How many of you heard flavor saver tomato? Tomato. How many of you heard flavor saver tomato? So flavor saver tomato is a biotechnological invention which has a higher shelf life. For example, if you take one tomato and you keep it in your open, after 2 to 3 days, can you see the tomatoes becoming a little black? Yes, it's becoming a little black. But flavor tomato is like not that. Flavor saver tomato is like I have higher shelf life. So that is one of the applications, high applications of your genetic engineering. Then you have increased efficiency of mineral usage by the plants. Best example I'll tell you here, that is your golden rice. Golden rice has high value of vitamin A also. Remember vitamin A, golden rice, high vitamin A. All the again an example. Then we have enriched. Here we have golden rice. Enriched nutritional value of the food, that is golden rice, which is rich in your vitamin A. Yes, vitamin A. Very good. Now students, one specific example. One specific example is going to be cotton. See, all of you know, ever since Gandhiji, India is famous for cotton. Remember, Gandhiji was doing that charka. I forgot what is it called. Okay, so Gandhiji was doing that, remember. So basically, India is very famous for cotton. Especially, we have so much of black soil in India, we grow cotton a lot. And especially, India is really good exporter of cotton also. But so it becomes extremely mandatory for us to, like, make something for cotton, because most of the cotton which we have in the nature was getting destroyed by certain bacteria or certain pesticide, nematode and everything, right? So we made something called as, not we, our entire group of scientists and everything, they made something called as BT cotton. Can anyone tell me what is this BT cotton now? Yes, what is this BT cotton? BT stands for Bacillus thuringiensis. The BT stands for Bacillus thuringiensis. So basically we have a bacteria, yes, BT toxin, BT. So what did we do is we isolated the gene responsible for BT. Yes, we took the spinning wheel, yes, we took the gene responsible for BT and we incorporated that gene into our host plant. So when we took that gene and incorporated the host plant, our host plant was producing a particular type of gene called as crygene. It is called as crygene. It was releasing a certain type of toxin. It was producing some type of toxin. Now this toxin is very special. That is, it will not harm humans. This toxin will not harm humans. In fact, when the insect takes up the toxin in its alkaline gut, the toxin becomes activated and the insect is going to die. Yes, that is, Bacillus thuringiensis contains BT toxin which is in inactive form. That's why it does not affect us. It becomes active toxin in the alkaline pH of the insect. The activated toxin binds to the surface of the midgut epithelium cells in the insect and creates the holes, and that's how a particular insect will not be able to survive. That is, this causes cell swelling and lysis and eventually the death of the insect. Now students, we have two types of crygenes. In total, three types of crygenes. In total, we have three types of crygenes. That is, Cry1 AC. Let me write down. We have something called as your we also have Cry1 AB. Now Cry1 AC as well as Cry2 AB, they both of them are extremely efficient against your cotton bollworm. Extremely against cotton bollworm. And your Cry1 AB is against cotton borer. Now what does this tell you? It basically tells you that every single gene is specific to one nematode, and that's how these plants are also developed. So similarly, apart from BT cotton, we also have BT brinjal, yes, and we have other BT corn. But BT brinjal and BT corn are not very famous in India because of certain ethical issues. But in countries like US and everything, BT cotton, BT brinjal is very, very famous. Okay. Then we have something called as RNA interference technology, or also called as RNA silencing. So here what happens is we have a particular DNA which is producing two types of RNA and anti-RNA. This RNA or antisense RNA, they join together, they form the double-stranded RNA. Now this double-stranded RNA will actually silence the mRNA of the particular insect, and because of that, that particular insect will not be able to survive. The example here given in your NCERT is, that is, RNA interference strategy is used to prevent the infestation of tobacco. In the infestation of tobacco mosaic in the tobacco plant by a nematode, that is Melanogene incognita. Melanogene or something, Melanogene incognita. So basically this particular mRNA of this particular nematode was silenced, and hence the tobacco plant was able to grow properly. Now this usually infects the roots of the tobacco plant, and hence it was able to grow properly. Okay. Then we have the advantages done, right? The next one is going to be in medicine. See, that was in the case of your agriculture. Now, what about in medicine, students? The medical application is very simple. That is, all of you know diabetes, right? All of you know diabetes. Diabetes is a disease caused by the lack of insulin in the blood. If insulin is less in the blood, what will happen? The blood glucose level is not regulated. The blood glucose level can increase or decrease drastically. So, to make sure the blood glucose level does not increase or decrease, insulin is artificially added in a diabetic patient. Yes. Now, but this insulin, where does it come from? Technically, our body should be able to produce the insulin. But if our body is not able to produce the insulin, we have to take the insulin from the external needs. Yes. Now, initially in the olden days, insulin was mainly procured or obtained from your cattle and pigs. But there was one major problem. The one major problem was very simple. That is, some of the patients become very allergic to it. They became what? They became very allergic to it. So they later on decided, no, we cannot take insulin from your cattle and pigs. Now we need to obtain insulin from bacteria. But when we obtained insulin from the bacteria, there was one major problem again. Because the insulin which we obtained from bacteria was in the form of proinsulin. What was it? It was in the form of proinsulin initially. But what is this proinsulin, students? Proinsulin is very simple. That is, proinsulin will have 3 chains. How many chains? It will have 3 chains. That is, chain A, chain B and chain C. But do we need all the 3 chains? The answer is no. Because in your mature insulin, we only have chain A and chain B. Chain C is removed out in the mature insulin. So how was this problem solved? The problem was solved in a very simple manner. That is, we grew 2 different types of bacteria. One bacteria was producing chain A, one bacteria was producing chain B. So later on, when we took chain A from this bacteria, chain B from this bacteria, and then we compiled it together with the help of disulfide linkage. That's how the problem was solved about insulin. Amazing story, I told you. I should become a storyteller now. Yeah, storyteller I'm becoming now. The next application is a gene therapy. Very important. Therapy is very important, students. Mental therapy is also very important. Now here we have gene therapy. For example, usually how do we cure a disease? All of you. If you get cold, what do? If you get a cold, you take a paracetamol or Dolo, you sleep off. Right? Or you take something else. Okay. Now students, but what if there is a disease which is heritable? Like we inherit the disease from the parents. How do you cure such a disease now? One such disease is your ADA. Yes, adenosine deaminase. That is, adenosine deaminase is a disease which is basically hereditary. Now, how do you cure such diseases, students? The actual cure right now which is happening is we take bone marrow cells and we implantation of transfer of bone marrow cells. But the transferring of bone marrow cells has to be done in a very periodic manner. Is it sustainable? Just like how dialysis cannot run for a very long time, even transportation of bone marrow cells also becomes very difficult. Yes, becomes very difficult. In that case, what do we do? In that case, there is something called as gene therapy. That is, we identify the gene responsible for your ADA cDNA, identify the gene and implement the gene at the embryonic stage. And the first case was done where? The first case was done a 4-year-old girl in the 1990, old girl with a ADA deficiency. The first clinical gene therapy was given to in a 1992 a 4-year-old girl with ADA deficiency. That is your gene therapy, students. That is your gene therapy. Why is not going well? That is your gene therapy. The other one is your molecular diagnosis. Now, what is this molecular diagnosis? Listen to my story again, you will understand the concept. After this, just read the NCERT once and it will be stuck in your mind, I'm telling you, students. Just read once, listen to me what I'm saying, and then read the NCERT once. Even though I'm not teaching this chapter in detail, this chapter will be in your mind. Okay? Now, what is happening in molecular diagnosis? Very simple again, very simple. So basically, when you have a cold, so when you have a disease, until the disease spreads big time, will you be able to see the symptoms? Will you be able to see the symptoms until the disease spreads completely? The answer is no. Right? Once the disease is spread enough, then you have symptoms like you'll get cold, headache, everything will come. So if we wait until the symptoms are arrived, sometimes it can be too late to cure the disease. Example is HIV. If you wait for the symptoms for the HIV, it becomes very difficult to cure. Yes, very difficult to cure. So in that case, what do you do? In that case, there is something called as molecular diagnosis. In molecular diagnosis, you basically detect the viral load or the enzymatic load in a very minute manner. Even the viral load is so less, you will be able to detect it. That is your molecular diagnosis. That is, you use methods like PCR. Remember, in COVID cases, RT-PCR we are doing, RT-PCR. That is basically detection. Yes, RT-PCR was basically detection of your COVID virus. Yes. Similarly, we have something called as ELISA. That is enzyme-linked immunosorbent assay. Right now, I will not teach you the whole process of ELISA because that will take around 15 minutes to teach. But I'll tell you one simple line which you need to write in your board and NEET examination. That is the principle of ELISA. Now, what is the principle of ELISA? The principle of ELISA is your antigen-antibody connection. Antigen-antibody connection. Remember that very much. Because in ELISA, if I teach you ELISA, it will take a lot of time. That is going to be, I'll tell you, we have two types of genes. So we take a probe, I attach the probe clone. Not understand. I need to actually draw the entire thing of ELISA, then only show you. That I'll be doing in the detailed class how ELISA works. Okay. Now, students, that is molecular diagnosis. Then we have something called as transgenic animals. Now, what are these transgenic animals? How are they different from normal animals? Do they have, instead of having 1 or 2 horns, does it have 5 horns? Do their morphology look different? No. Not morphology different. They can have difference inside internal structure. That is, animals that have DNA manipulated to possess or express an extra gene. Basically, we are altering the DNA to make sure we have a particular gene, and expression of gene is different. That is, transgenic rat, rabbit, pigs, sheep, rosy cow. Very important example. Rosy cow is the first transgenic cow which produce a lot of a high rich milk, right? Your lactobulum. Yes. Then we have something called as your advantages. Some here, students, board examination or NEET examination. One question will come from here: advantages of your transgenic animals. Advantage of transgenic animals, one question directly will come. That is going to be to study the normal physiology and development. That is, how a particular gene is expressing normally. We usually take a particular animal and we test it on it. Then we have to study the contribution of genes in the development of a disease. Yes, how a disease is working. We take mouse here, poor rat or mouse. Every single time we have to do an experiment, kill one mouse. That's what even Griffin did, right? Griffin experiment. To know how the disease was working, pneumonia. To know how pneumonia is working, he used a rat. Yes, he'd killed rat and everything, right? So that's what we are doing still. Biological product. This is where you learn rosy cow. Very important. What is rich in the milk? That is also very important here. Then we have vaccine safety testing. Every single time there is a vaccine which needs to be tested out before giving out to humans. So whom do you test it on? Poor animals. Poor transgenic animals. You test all the different vaccines and you get the result. Then we have the chemical safety training. That is basically testing the toxicity. Basically testing the how particular product is toxic. So we take a transgenic animal which is very sensitive, poor sensitive one. So we do all the study on that sensitive one and see how it is reacting to a toxicity or a particular product. The last one is your GEAC, that is Genetic Engineering Approval Committee. Genetic Engineering Approval Committee. Now this committee will basically look over all the genetic engineering projects and it will make sure some of them sent, some are not sent. That is approval, they basically give approval. If there is no approval, we might have such, weird combination of genetic engineering can happen. It can lead to a mass extinction also. Imagine we create a superior breed and what if we introduce it? Alien species invasion. The other native breed will it be able to survive? No. So we can have a large mass extinction if the control is not there on the genetic engineering. That is very important. That is making decisions about the validity of GM research, safety of GM organism for public services. And apart from this, there is one more two concepts called as your biopiracy. That is also very important. Learn biopiracy and also patenting. For example, basmati rice. These US people, they took our basmati rice and we crossed with something else and they called their basmati rice. You're noticing what they're doing. Similarly, if you don't take our turmeric, Indian turmeric or neem turmeric paste, they are patenting it. They're using it. Why? When you patent something, basically you're telling that it is mine. You're telling the house, right? This is my bottle, this is my book. That's what patenting is. This is my product. So patenting is very important along with biopiracy. Okay, students? So with that being said, this chapter I've told you in a much more story manner. Don't worry. I'll make you write every single point of this. See, I've told almost all lines of NCERT I've told verbally. I didn't make you write anything. Basically from these points I've told you the entire chapter. Right? Entire chapter has been done. Now don't worry, after your examination for your NEET, we'll be making you write every point and making you understand even more detail, like detailed explanation. We'll go much more detailed explanation. For example, I'll be teaching you how ELISA works, how does your antisense works, how does the BT cotton works in detail. So with that, I'll give the mic to Gopika Ma'am now. Yes, ma'am. Next big chapter. Healthy, are you healthy?
No. Yes.
So, we'll welcome Gopika Ma'am and I will scoot now.
Okay, students, welcome to Vedantu NEET English. I am Miss Gopika, your biology master teacher. Those who are new to this channel, do not forget to subscribe, and if you want these mind maps for free, join us on Vedantu NEET English Telegram channel. That is the only way to access these notes. There is no other way. We will not be giving a downloading option here. Okay, so quickly join Vedantu NEET English Telegram channel and take all these notes for free. Do not pay money and buy the same mind maps anywhere. Okay, so yes, students, we are going to do human health and diseases. I know all of you are tired. Even we are tired because it's a continuous lecture. But yes, we will do it faster. Okay, do not worry. I will not talk as much as sir talks. Okay, so yes, we will continue. Okay, human health and diseases. The first thing that you need to know is, yes, the definition of health. Okay, the definition of health. State of complete physical, mental and social well-being. Okay, that's what I told you. If you're physically healthy, does not mean you're healthy. Okay, you need to be really good with your surrounding people. You need to be really good with your parents. You need to be good with your friends, teachers, everyone. That means that is what is mean by well-being. Okay, social well-being. How are you socially? If someone has some problem and they want to come and talk to you, if you're always grumpy, irritated, frustrated, they will be like, okay, this person is not mentally healthy, right? So you have to be physically, mentally, socially healthy. And if you still haven't smashed the like button, then this healthy relationship will break. Okay, so make quickly like the session. Okay, students, so in this chapter, this mind map is more than enough if you're doing like a quick revision. I will not go in depth because this chapter is very long. Okay, you know that I took it in I took it almost 8 hours separately, 4 hours, 2 hours, 2 hours. So I took around 8 hours to complete this chapter in detail. So I hope most of you saw that. But if you haven't seen and you want some topic detailedly, just go check my video. Okay. So look here, we have first diseases. So diseases are basically something that is caused by a foreign particle. It could be a bacteria, it could be a protozoan, it could be a helminth, it could be a virus, it could be any among this. Okay. So what you need to remember, typhoid is caused by Salmonella typhi, which is caused by a bacteria. It's a bacterial disease. Then we have pneumonia, which is caused by Streptococcus pneumonia, again a bacterial disease. This notes also is in the Telegram. I will, if you want, I will reshare it once again. If you ask me, you can just put a comment, ma'am, I want the notes. I will reshare it again. Okay. Then we have here, students, look here, Salmonella typhi, pneumonia. Then we have filariasis or your elephantiasis. Okay, filariasis or your elephantiasis, which is caused by your worms. Okay, which is caused by your worms, and the person who is responsible is your Culex mosquito. Culex mosquito is the vector of this disease, which is elephantiasis. There is an image in your textbook where a person has a very bulged leg, right? Have you seen that? That is called as elephantiasis. Okay. Then we have amoebiasis, which is caused by Entamoeba histolytica. This is again caused because of contaminated food and fecal matter mixing with our vegetable soil and etc. Okay. Yes, then students, we have common cold, which is my favorite, my best friend, my soulmate, everything. Okay, whatever I am today is because of rhinovirus, because cold never leaves me. And you know, most of you will be there who is like has a very close relationship with rhinovirus, that is your cold, okay, caused by virus, okay, virus. Now, malaria, which is caused by Plasmodium. Plasmodium vivax life cycle we studied yesterday and day before yesterday. So, revise it quickly. Tell me the enzyme, or tell me the chemical that is, not enzyme, chemical that is released in this Plasmodium vivax that causes fever and chills, also promotes the formation of gametophyte. In the chat, fast, all of you, tell me the chemical which is released by Plasmodium vivax that helps in causing fever and chills. At the same time, it also causes gametophyte formation. Let me see who is the one who is going to answer. Second question to all of you, tell me which is the Plasmodium that causes malignant malaria. Two questions. First one, which is the chemical that will be released, okay, by the RBC that causes fever and chills. Second question, which is the one? Awesome. Claps for Lakshmi. Lakshmi, good job. Drishti, good job. And Darshini, good job. Okay. Yes, malignant malaria is caused by Plasmodium falciparum. Very good, sweetie. Hemozoin is the chemical release that causes fever and chills, because of which even it also facilitates the gametophyte formation. Okay. Yes. Students, then we have Ascaris. This is Plasmodium vivax. Okay. Ascaris, which causes ascariasis. Okay. These are the diseases. So, typhoid, pneumonia, malaria, common cold, elephantiasis, ascaris, amoebiasis, and your, that's all, no? Pneumonia. Okay. These are the diseases caused, and the causative agents are mentioned here. The symptoms I have mentioned in my notes. Okay. I'll share it with you all after the class. Okay. Now, now here you have to know about the types of immunity. We know that there are a lot of diseases, but there is a way that our body will also be, you know, responsible, or the body will protect, okay, protect from all these things, right? That is your immunity. So, we have two types of immunity. So, look here, students, we have acquired immunity and innate immunity. Innate immunity is the immunity which we have from birth, and it is not specific. Like any fellow comes, we will react type, that kind of a innate immunity. Acquired immunity is no, I will only target the student who is not listening in the class. I will only target them, right? I'll only target them. Yes, ringworm caused by Microsporum tetraphyton and your epidermophyton. Okay, that is your ringworm allergy. That allergy picture which is given in your textbook, you would have seen one white color patches. Yes, yes, two types of immunity, innate and acquired. Innate by birth, acquired after birth, right? After birth, we get this acquired immunity. Innate immunity is non-specific, and which are the types of barriers in innate immunity is physical barrier, physiological barrier, cellular barriers, and cytokine barriers. Physical barriers means whom, students? Simple, our skin and mucus. Skin and mucus comes under physical barrier. Skin is a protection, mucus also is a protection, right? Second one is physiological barriers. This is your tears, okay, tears and your HCl which is released in the stomach. These are physiological barriers, okay. Then we have cellular barriers and cytokine barriers. Cellular barriers is your T cells, B cells, all your cell mediated immunity part. Cytokine barriers are where you will focus on your interferons. Yesterday also I told you with the questions, interferons. That means I am in problem because of a viral infection. Baswa sir is next to me, Shreyas sir is next to me, Diksha ma'am is next to me. What will I do? I'll be like, I am getting affected by a viral infection. I will not be able to, that's what, I will not be able to do anything about it. So, I will release interferons to give the signals to these people and tell them, you people escape, or you people start forming something against this virus. I will sacrifice my life. Students, I'm the good person. Okay. So, I will sacrifice my life. So, this is the cytokine barriers. Okay. Fine. Now, acquired immunity. Acquired immunity produces primary and secondary response. That means what? Your primary, there is primary, secondary lymphoid organ, no, like that only. There is a primary and a secondary response. Primary response is, first time if something comes to my body will use 10,000 soldiers to fight this. Second time the same thing comes to my body, 10 of them will be more than enough. That means memory is there. Body always holds a memory of what has come. Okay, but the problem is diseases like cold, memory will not work because they always come in new variants. Now, acquired immunity is very specific, okay, and they form or they help in the formation of your T lymphocytes and B lymphocytes, okay. T lymphocytes and your B. Very emotional story I told you. Don't worry, students. Don't be emotional. I sacrificed my life and I send them signals and they all saved themselves from viral infection. Okay. So, yes, produces primary and secondary lymphocytes, that is T cells and B cells. T cells and B cells, where is it formed? Where is it formed, all of you? Where is the formation happening? Where is the maturation happening? Okay. Formation, you will tell. Maturation, I will tell. Okay. So, B cells will be matured in the bone marrow. T cells will be matured in the thymus gland. Okay. Thymus gland. That is why the name T cells. You can remember like that. Now, B cells produces antibodies. Who are antibodies? Why is the answer not mentioned? Students, tell me the answer. Where is B cells and T cells formed? Manufacturing, where is it happening? Where is the manufacture happening? Okay, it produces antibodies. I am not going to do this diagram again because you already know the diagram. Paratopes are there, epitopes are there, 2 heavy chains, 2 light chains, 16 disulfide bonds. 16 disulfide bond. 1 antibody, 2 paratopes are there. Okay, this is the paratope where the antigen will come and bind. No one is telling the answer. Where is B cells and T cells manufactured? No one knows the answer. Sorry, no one knows the answer. Okay. Something, you know. Where is it? Very good, sweetie. Awesome, awesome. It is manufactured in your bone marrow, but maturation is happening. B cells will mature in the bone marrow. B cells will mature in the bone marrow. T cells will mature in the thymus. Okay, T cells will mature in the thymus. Now, acquired immunity is again divided into 2 types. One is active own antibodies, ready-made antibodies. What is this? Active and passive. Okay, active and passive. Now, look here. Natural exposure to infectious agents. That is natural. That means now you got a disease. Okay, your body is countering it for the first time. It will make antibodies against that. First time when antigen came into your body will start making antibodies against it. That is called natural immunity. Natural active immunity. Same thing we will do artificially. When COVID happened, what did we do? We gave a weakened vaccine. Okay, weakened vaccine means what? We gave a weakened antigen. Our body realized that this was something foreign particle. It started forming antibodies against it. This is artificial because we are injecting it from outside. Is this clear? Okay, artificial, that is immunization or vaccination. Now, natural ready-made, we are giving ready-made like your colostrum from your mother's first milk. Okay, first milk, right? That is your natural. That means the mother was not injected with anything. The mother made it naturally, but it is ready-made for the baby. Right? It is ready-made for the baby. So, it is colostrum and what are we getting? IgA is given. Okay. Artificial antibodies like your snake. Now, if you get a snake bite, they will give you a snake venom. Right, with the help of snake venom. What these people will do? They will inject this into the horse. Why do I tell? Horse. Okay, they inject it to the horse and the horse will make antibodies and that antibodies will be taken for the treatment. So, this is completely ready-made, completely artificial. We are injecting something into the horse. The horse does not have snake venom in its body. Is this clear, students? So, we have active own, ready-made. Okay, ready-made. Active we have natural and artificial. Natural is when we are already exposed and our body is making. Artificial means we are giving weakened antigen and our body is making. Okay. Now, ready-made means what? Ready-made and it is passive. First one is the milk that is given from the mother, colostrum. It has antibodies which are necessary for the baby. It is made naturally in the mother's body, but it is given from outside, ready-made. Next is artificial. Whenever there is a rabies, snake bite, we give artificial antibodies directly. We do not wait for them to make antibodies. We just give them directly. That is why it's called passive ready-made. Okay. You go to a clothes shop, you tell, give me skirt or give me a dress. That is ready-made. You do not have to stitch. But if you go to another shop and you take the cloth and come, that means you are taking the cloth, you are making it into your own style. So, that becomes what? It is active, but it is still artificial, but it is coming under your active kind of immunity. Okay, because you are still going to stitch the dress. Ready-made means what? It's already made by someone, you just have to wear it. Okay, got it? Fine. Now, look here students, drug abuse, one second. Here, cancer, types of cancer. Okay, types of cancer is your benign tumor, malignant tumor and metastasis. Metastasis is not a type of cancer, it is the continuous replication. That means they have lost the ability of contact inhibition and they do not understand that if I touch a cell, I should stop. They does not know that. They have lost that ability. So even if they are passing through a cell, they keep replicating and keep multiplying. That's why cancer is very difficult to cure. So we have two types, benign tumor and malignant tumor. Benign tumor means wherever it goes, it just stays there. Now your hair, right, hair when you take bath, it goes and clogs near the sink hole. It will stay there. Until you pour water, it will not go, it will stay there. There only it will keep accumulating and accumulating. Malignant tumor is not like that. Imagine there is a chunk of hair in a river. As water moves, wherever it goes and keeps multiplying. Same way, malignant tumor, wherever it goes and stays, there it multiplies, again moves and goes and multiplies. It keeps spreading and spreading and spreading. Benign tumor, wherever it stays there. So that is easier to remove. Okay, easier to remove. Okay, cancer cells have lost the property of contact inhibition. Now, how do I detect it? I can detect it with biopsy, radiography, CT and MRI scans. Most better one is MRI scan because magnetic radiations are sent. All other, there is UV and X-rays sent, which is again very harmful. How do you cure? There is no proper cure for cancer, but you still do chemotherapy. So in chemotherapy and radiotherapy and immunotherapy, we basically kill the cancer cells. Okay, we kill the cancer cells, but chances are there that they would have done the job of spreading very soon, and most of us or most of the people get to know it in the third and the fourth stage. Okay, because sometimes symptoms will not be visible. Okay, so radiotherapy, chemotherapy and immunotherapy is the method by which we can at least, many people have got saved. It's not like survival rate is less, but sometimes relapse happen. Okay, sometimes relapse happen. Now look here all of you, lymphoid organs. We saw their immunity. Here we are seeing lymphoid organs. Students, in this mind map, you will not see antibodies and all. IgA, IgM and all that you won't see. Understand IgG, okay? IgG is the one which is most abundant. Just remember only this much. IgG is the most abundant, okay? And IgM is the largest, okay? Largest in size and more in number. IgG, more in number, abundance around 75%. And IgM is the largest size-wise, okay? Yes. So here you can see, students, lymphoid organs are of two types, primary and secondary. Primary, who and all are there? Bone marrow and thymus. Bone marrow, B cells will be matured and, formed, and here T cells will be matured, okay? Now, secondary, we have spleen, lymph node, tonsils, Peyer's patch, right? And here only we will be discussing about malt. What is it? Mucosal associated. What is the full form? Mucosal associated lymphoid tissue, which covers 50% of the lymphoid organs. Yes, very good. IgM, IgA, IgE, IgD. Correct. Okay. Gamed. How do you remember the antibodies? I told you gamed. Gamed. Okay. This is the antibodies. Out of this, G and M are one is most abundant, one is largest. Yes, Peyer's patch, tonsils, spleen, lymph. Okay. Here, Peyer's patch and malt also, MALT, that is your mucosal associated lymphoid tissue. Now, drugs. Drugs, students, symptoms prevention is not our tension, but before the drugs, let's do this also. Let's do autoimmune. Okay, look here, pathogens we understood. Autoimmunity, that means what? Our body definitely loves our cells definitely love our self cells, yes. But what happens? No, sometimes our cells will harm the self cells. That means I am there, my twin sister is there, we both come from the same mother. I will harm her or she harms me. That means what? We both are attacking each other. We are from the same family, from the same womb. So we are attacking each other. Now what is going to happen? We will get completely, okay, we will have a problem. Yes. Yes, it is there in the tracheal lining. Very good. In the tract lining. Now, students, when our cells start killing the cells, we get autoimmune diseases. Okay, give me an example of an autoimmune disease. It starts from M, it starts from A, different names. I mean, it starts from R. These are three different. Give me an example of an autoimmune disease. Okay, students, look here. Memory-based acquired immunity, which is able to distinguish foreign molecules or cells from the self cells. But when our cells start killing the self cells, it will lead to autoimmune disease. Autoimmune disease, the example is rheumatoid arthritis. That is the example of your autoimmune disease. Okay, is this clear? Now, what is an allergy? We all have got allergy once at least in our lifetime. Students, what is happening? Basically, one foreign particle is entering our body, and that foreign particle is causing some kind of problems in our body. During that time, our WBC will do something. Can any of you tell me what our WBC will do? Our WBC will squeeze out and go to the tissue and help the, it does phagocytosis. I want the name. It starts with D. It starts with D. I've given the clue to all of you. Okay. Now, exaggerated response of the immune system to allergens. Allergy, when an allergy comes or when a foreign particles enter, our body, the blood vessel vasodilates, more blood enters, causing inflammation, redness, itching. That is to show our body that something has gotten, or you're eating something that is you're allergic to. How do people know that I have walnut allergy, I have peanut allergy? How do they know? That's because our body is causing an allergic reaction. And that is with the help of mast cells. Mast cells, okay? MAST cells. Awesome, Drishti, diapedicis. That means WBC squeeze out to the pores and go to the tissue which is affected, and they do phagocytosis. Okay? Yes, and it is, yes, serotonin. Histamine is the biggest fellow. Histamine is the reason why we are able to see that kind of an allergic reaction. Okay, and who is mast cells are the ones who are going to help in this. Now, antihistamine, adrenaline and steroids quickly reduce the symptoms. So, histamine is the one who is getting released, okay, who is getting released and causing that allergy. So, how can we control it? By taking antihistamine, okay, antihistamine. Is this clear? Okay, students, should I do retrovirus again? Yeah, yesterday also I did retrovirus. Just understand, your HIV or your AIDS is caused by human immunodeficiency disease, and that only attacks three types of cells: T cells, macrophages, dendritic cells, which has CD4 receptors in them. This much you know, and you know that they have integrase, protease and reverse transcriptase, and they will manipulate our human genome. They will manipulate our human genome. Okay. Now, drugs, the most important thing here is your opioids, cannabinoids and coca alkaloids. You only have to remember these three: opioids, cannabinoids, coca alkaloids. Opioids is taken. Okay, opioids is taken. Done, no? Yeah, I will not do HIV again. Opioids is taken from Papaver somniferum, or simply called as poppy. Okay, poppy. Coca alkaloids is from Erythroxylum coca. Erythroxylum coca. Cannabinoids is from Cannabis sativa, which looks like a palm. That leaf you saw yesterday, no? I showed you in the diagrams. It looks like a palm. Cannabis sativa. Okay? Yeah, coca. It is called, this one, your cocaine, crack, coke. All this is for your coca alkaloids. Okay? Then, students, prevention causes symptoms. All of you know. It is just, you have to take care and you have to reduce the curiosity, unnecessary curiosity in trying drugs and not be addicted to it. And sporozoids done. Okay, HIV done. Should I do HIV and malaria again? No, no. I've done diagrams. Yeah, diagrams I'm not doing again. Because this is revision. You should have watched the diagram video. Okay. Don't do diagrams, sir. Yeah, you have to watch. Okay, handing over.
Thank you, thank you, thank you, thank you, thank you, thank you.
I'm going Students, do you want microbes? Tell no, ma'am. I will not do microbes.
Exactly. Thank you so much, ma'am, for the warm regards. Warm regards. Warm regards for this.
I want students to listen to talk too much.
Yeah.
Yeah.
Yeah, I know.
You have too much story then.
Okay, should I reduce my story then, students?
Yes.
Should I reduce my story time? I thought stories were interesting because that's how I learned it, because I learned biology with the help of stories, walking in the park, learning botany in the jungle. Yes?
No. Yes. No.
Everyone is sleeping. Students, can we start with organism and...
Sorry, one second.
What happened?
One second. Give me the mic. No, 1 second. Just respond to this. Okay, students. Lakshmi, chikungunya and dengue is not part of your board syllabus, dear. This is part of Neet syllabus. But this is a revision. So, chikungunya and dengue, please go watch that video. I think it's only 2 hours. Okay, you can watch it in 2x. It will become 30 minutes. Okay?
Watch that. Hello. So, are we ready for ecology? Are we ready for 2 chapters of your ecology first? Ready for ecology? Ma'am, teach dengue and chikungunya how many times? Nothing. So, students, are you ready for ecology now?
Yes, they are ready. So, please come.
They are ready? Okay. So, students, organism and population, students, you should know by now, the organism and environment part has been removed from your NCERT, right? This part has been removed from your NCERT, so you do not have to study the organism and environment for your boards. For Neet, yes. So you need to study for all of these for Neet. That you should be knowing what is the meaning of your regulators, conformers, migrators and suspenders, and also what is conformers, regulators and partial regulators for Neet students. Board students, no. This part has been removed for you. For Neet students, I'll be telling you that is, regulators are the organisms which can regulate the internal body temperature. Conformers are the organisms which cannot regulate the body temperature. Regulators are organisms like cold-blooded animals. Have you seen crocodile? Have you seen crocodile? But it comes out of the water and sits there and open its mouth. It is opening its mouth and sitting in the sunlight. Why? Because it cannot regulate the body temperature. That is why it's opening its mouth and it's sitting in the sunlight, so it can take more heat inside. Then we have partial regulators, which is between the conformers and regulators. But let's focus on what is important here. Let's focus on this part here. That is population part. Students, you will be getting a lot of questions from your population attributes. Let's focus on population attributes. First point. See here. In population attributes, normally question will not come from population size or natality, mortality, sex ratio. Question will not come. The main question which you can see in your 1 mark question or 5 mark question is going to be your age pyramid. It is going to be what? Age pyramid. And I have explained age pyramid in day for yesterday video complete. I have explained. Briefly, I'll tell you right now. Students, age pyramid is basically a pyramid which tells you that distribution of different types of age group in a pictorial form. That is, we have expanding, stable and declining. In expanding, it's a pyramid shape, and expanding is mostly seen in the case of your developing countries. Then we have stable. In stable, can you see your pre-reproductive as well as post-reproductive. Pre-reproductive and reproductive are almost equal in your nature, and this is stable is seen in the case of your developed countries. Seen in the case of developed countries. Then we have urn shape, which is declining, where reproductive stage might be high, but post-reproductive and pre-reproductive are very low. That is in the case of Japan. In Japan, they don't want to get married anymore. They don't want to have kids at all. That is, pre-reproductive is less. And in Japan, there are a lot of old people. Post-reproductive is also very low. Even it may be high, but they are also dying very much. And also declining can also be seen in the case of your war countries. Now students, here I want all of you to understand growth models. Question, if they ask you, they might ask you from your growth models. That is, in total there are 2 types of growths. That is, one is logistic growth, one is your exponential growth. In your exponential growth, what happens in exponential growth? That is, you are given unlimited money. So your organism has been given unlimited resources. Will it keep on growing? Yes. That is your exponential growth, which you can see a J-shaped curve. And here formula is, equation is dN by dt is equal to Rn. R is your rate of intrinsic factor, that is internal factor. Clear? Then we have a more sigmoid curve. Sigmoid curve basically represents the, here, this is your sigmoid curve. Sigmoid curve represents this one. Sigmoid curve represents the logistic growth in which resources are very limited. Very limited resources in the case of your sigmoid curve. The equation is your dm is equal to Rn k minus n by k. Now what is this k now? K is called as your carrying capacity. It is called as what? Carrying capacity. Carrying capacity basically means the same. Imagine you have a scooty. All of you ride a scooty, right? On the scooty, can more than 10 people sit? Imagine more than 5 ones they can't sit. Imagine there are 5 people sitting on a scooty. Will the scooty be able to go further, or will it collapse? The scooty will collapse, right? That is called as carrying capacity. That earth or a particular environment has a carrying capacity. If the carrying capacity exceeds, the entire ecosystem or the biosphere becomes destabilized and population becomes reduced. And the entire sigmoid curve can be divided into this is your lag phase, this is your log phase, and this part is your asymptotic curve, in which the population is not increasing after this, Clear? Then we have, this is equation for your population density, that is for nt to the plus 1, that is nt is plus birth plus immigration minus death plus emigration. Now what is the difference between immigration and emigration? Immigration means they are coming into the population. Emigration means they are going outside. See here. Immigration, they are coming into the population. Immigration, they are coming into the population. And emigration, they are going out of the population. Now here, immigration will increase the population, as well as natality will increase the population. Whereas mortality will decrease, as well as emigration will also decrease the population. That is just the point here. Okay. Then we have, now if you ask me, where does the question come from here? Since this adaptation was a place where the question used to be asked repeatedly, it is also removed from your syllabus right now. The place where the question is going to be keep on asking is going to be this table. Did you see this table? In this table, they will not only ask you what is mutualism, they will also ask you what are the examples of mutualism, or what are different examples of commensalism. For example, if I tell you, first one is your, let's take predation. Let's take predation, all of you. Let's take predation and let's talk about predation first. Predation is a phenomena. Oh, I'm clicking on parasitism. Let's talk about your let's talk about predation first. In predation, when you think of predation, what comes to your mind first of all? When you think of predation, what comes to your mind? National Geography. Yes, National Geography comes to your mind. That is, one deer is trying to hunt, one lion or a tiger is trying to hunt a deer. In that case, a tiger is benefited, a deer is under loss. That is plus and minus. But if I ask you, a particular bird eating a seed, is that predation? Is that predation? You will be like, no sir, how is the cute bird eating? That also comes under predation, students. A bird eating an insect also comes under predation. Clear? Now, if I ask you, apart from this, in your NCERT, if you read your NCERT actually, and apart from this table, they have given something called as roles of predators. What are the different roles of a predator? Now, that question is very important. It is a very hidden line and a lot of students miss out. The first important role of a predator is to do what? Is to transfer the energy. The first point of your predator is transfer of energy. Transfer of energy. Yes. That is, when the predator is eating the, don't think of lion right now, that is the top carnivores. Think of a cow. Cow is also a predator. Cow is eating the grass, right? Cow is eating grass, it is predator. So cow is eating the grass and all the energy which is present in the grass is sent to the cow. Now from the cow, the energy will go to the next trophic level. Are you noticing? So the main role of a predator is transferring of energy. That is the first one. That is the first one, right? The second one is keep prey under the control. Now what is this, they keep prey under the control? It's very simple. Imagine a particular frog is not eating little bit of grasshoppers. Will the grasshopper population increase dramatically? Yes. Now, if grasshopper population increases dramatically, will it cause an ecological disbalance? Yes. Imagine the grasshopper is eating on the plants. If there are more number of grasshoppers, will there be more damage to the plants? Yes. So primary productivity will be coming down. So are you noticing? If there is no proper predator, if you remove a predator, the entire ecological disbalance happens. Entire ecological disbalance happens. That is, imagine there is a frog. It is eating a insect. Yes, frog is eating an insect. Insect is eating a plant. Now imagine if this insect is removed, what happens? Frog population will come down and plant population will increase. Yes. So is that good? No, that was not good. Are you noticing? If you remove one species from a population, major discourses happens. Clear? The third here best example. The best example is the prickly pear cactus. You know prickly pear cactus. It was an alien species invasion. It was introduced. Prickly pear cactus was introduced just because there was no predator for it. Just because there was no predator, natural predator for prickly pear cactus, the cactus kept on dividing and increasing so much of number, it became a nuisance. Do you know how they controlled the prickly pear cactus in the end? It's a tongue twister, yeah, I know. They controlled the prickly pear cactus by introducing a moth later on. And a moth came and it fed on the cactus and reduced the population. Example, that's how the question will be asked. That's how the question will be asked. The next one is, they help in maintaining maintain species diversity. Now how do they help in maintaining the species diversity? Now you tell me, imagine there is one big boss in your area. There is one big boss in your area. When that big boss is there, will the small crimes will be happening? No. The big boss will take over the entire thing. The small thieves will not get much of chance. Yes, sir. Similarly, if there is a major predator, in your forest, the small computation between the two different species will not happen. But as soon as the main boss is gone, will the two small organisms, will they fight among each other for the top position? Yes, they will fight among each other for the top position. So similarly, it maintains the species diversity. The main predator is making sure that there is no fight between small species. Clear? The best example is your starfish. That Pleiotropy, Pleiotropy is a type of starfish. Example is very important. Pleiotropy is a type of fish which once you remove that starfish, there was an experiment. Once you remove the starfish, within a year, complete 10 invertebrates went extinct. Example, NCERT example, very important. Okay, 10 invertebrates were completely extinct. Okay, now you tell me, what happens if the tiger, will the tiger ever kill all the deer at once? No. The tiger will never kill all the tiger at once. He will do something called as sustainable eating. That is very important for a predator. Sustainable eating. So this is about predation. Now if the deer is running from a tiger, why is it running? It is protecting itself. Now will any organism in the world be like, okay, come on, you eat me now, I am ready, happily die. Will I happily die? No. So every single organism has something called as sneeze coming, wait a bit. So every single organism has something called as a defense mechanism. They have a defense mechanism. The best example for defense mechanism is going to be your plants. That is, since they can't move, right? They cannot move. So they need to have special mechanisms. Example is cactus. They have thorns on cactus. The other example is your calotropis, which has milky weed on top of it. Yes, calotropis. Then there's other cases. In your case of your butterfly, there's monarchy butterfly. Yes, that butterfly which feeds on your, when it's in larvae stage, will feed on a milkweed. So your butterfly is deadly. Butterfly is so deadly that herbivore, other insects will not eat the butterfly. So every single organism has a defense mechanism. The other example is your frog and octopus and certain chameleons. What do? They camouflage themselves. That is, they completely become invisible. Yes. All of these are different mechanisms in which they protect themselves from predators. Students, are you noticing? In just one topic, we spoke about so many factors in which question can be asked. So students, remember, don't just read this plus minus, remember the examples, because that's where the question will be asked. Similarly, we know mutualism, that is plus plus we say, plus plus we say. Now, what are the examples of mutualism? The best example, which is the first example which is given in your NCERT for mutualism, is mango tree and orchid. Mango tree and orchid. Orchid is growing on a mango tree. Mango tree is not bothered. It is also benefiting. Yes. Mango tree and orchid. No, that is for your commensalism. Sorry, that is for commensalism. The example for your mutualism is given your fig. Fig as well as wasp. Both are benefited. That is mutualism only, right? Yeah, fig and wasp. Mutualism. Both are benefited there. Right? Both are benefited. Mutualism. Now students, you tell me in the chat, what is the example for parasitism? Any example for parasitism in which one is benefited, other is lost? The best example of parasitism is going to be human lice. Yes, in the hair, the human lice. Example for your parasitism. The other thing which I want all of you to notice in parasitism is something called as brood parasitism. Brood parasitism. In the case of your brood parasitism, a particular bird called as quail bird is laying its egg inside the crow's nest. And once what happens, once the quail egg and crow's egg, they look so alike, you cannot differentiate which is which. So what happens then? What happens then is the quail egg will grow, the hatch, and the quail will be born. If it hatch earlier, it will try to kill the other crow eggs also. Yes, that is a brood parasitism which everyone needs to remember. Clear? Then we have commensalism and amensalism. In amensalism are like toxic friends in which one is lost, other one is not benefited, not losing anything. Then we have commensalism. Commensalism example is very simple. That is your first one is a mango tree and orchids. The mango tree is not bothered if orchid is growing or not. The second example is your whale and barnacle. Whale does not get bothered, but barnacle is benefited. The other example is your sea anemone and clownfish. Clownfish is benefited, the sea anemone is not bothered. The other one is egret. Egret and cattle. Egret is eating the insects, cattle is not bothered. All of these examples can be asked in your board examination and NEET examination. They'll ask you for sure. They will not ask you like this, plus minus. They'll give you an example and they will ask you which is which. Clear? I've told you literally, I told you all the examples. And with that, this chapter is done. With that, this chapter thing is there. See, in this chapter, you should know what this graph, this particular graph, and you should know what is natality, mortality, immigration and emigration. That's it. Very easy chapter. Since this part is removed from your NCERT, this is the main part. This was your physical adaptation has been removed. But for NEET students, it's going to be there. And later on when I'm teaching this chapter, I'll teach you again. And if you want to learn the adaptations, you can watch. Gopika Ma'am has done a separate video on organism and population. You can watch that also for adaptations. Okay. The next chapter is ecosystem. Again students, in ecosystem, this part has been removed. Yes, that is, this part has been removed for your NCERT. Succession, entire succession part has been removed. So don't worry about this. And also nutrient cycle has been removed. And also ecosystem service has been removed. So this part of the cycle is not there. Let's focus on this part of the cycle now. Now what is the first thing here? Listen to me very carefully. No. Can we start ecosystem now, students? Can we start with ecosystem now? Can we start with ecosystem now? Yes. Yes. Students, ecosystem, what is the meaning of system first? Tell me. How many of you know what is the meaning of system? System is basically a multiple group of components. System is a group of components working together. Example, computer system. In computer system, we have mouse, keyboard, monitor, CPU, everything. All of them are working together for one function. Similarly, we have something called as ecosystem. Ecosystem is a system in which we have biotic factors as well as abiotic factors. Now, biotic factors and abiotic factors are interacting among each other and also within each other, between and within each other. Okay, that is, ecosystem mainly has your biotic factors and abiotic factors. Biotic factors mainly includes your autotrophs, herbivores and carnivores, and abiotic factors mainly includes ponds, lakes and river system. Now, there is something called as distribution of distribution. Now, what is this distribution of different species? The first one is called as your stratification. Now, what is this stratification? Stratification is very simple. That is, it is a vertical distribution of species. What is stratification? Stratification is basically vertical distribution of species. Remember that. That is, in a particular forest we have grasses, herbs, shrubs. Then we have this is not in NCERT, don't worry about this, shrubs and trees and tall trees. That's all. That is your vertical distribution called as stratification. Called as what? Stratification. Then we have something called as productivity. Now, what is the productivity? Productivity is very simple. It is amount of biomass produced per unit area in a given amount of time. What is productivity? Repeat after me, students. What is productivity? Productivity is nothing but the amount of biomass or organic matter produced per unit area in a given time is called as your productivity. Now, productivity can be represented in two ways. That is, productivity can be represented in the form of weight as well as energy. Yes, primary productivity can be represented in the form of weight as well as energy. When it is weight, it can be represented by weight as well as energy. Weight is going to be your grams per meter square. Yes, and in terms of energy, then energy can be represented. The energy can be represented by your kilocalorie. Energy can be represented by kilocalorie per meter square. That is energy presentation. Yes. Then there is something called as productivity. See, primary production is different. Productivity is different. Primary production is different. Now, what is this productivity now? Productivity is nothing but rate of formation. Productivity is what? Rate of formation. Primary production is this one. That is formation of organic matter or biomass in a given area in a given time. But when you add rate to it, rate of production, that has become productivity. That is, and it can be represented by what? It can be represented by NPP is equal to GPP minus R. Now, what is this GPP? GPP is called as what? Gross primary productivity. Now, what is gross primary productivity? Gross primary productivity is productivity done by the green plants. That is the total photosynthate. The amount of photosynthesis happened, right? That is your GPP. Now, whenever plants are doing some work, will they eat some of it? Yes, they will utilize some energy. Now, whatever plants are using, that is called as R. That is respiration loss. So, GPP is the total amount of photosynthesis produced by the plants, and R is your respiration loss. GPP minus R is going to give you NPP. NPP is nothing but net primary productivity, and NPP is available for next tropic level. NPP is available for herbivores or secondary productivity. That is rate of formation of new organic matter by consumers. That is your secondary productivity. The next important part for your NEET as well as your board is going to be, that is decomposition. Students, I've already told you yesterday with the help of diagram, decomposition mainly involves 5 different steps. The first one is called as fragmentation, in which earthworm makes fragments of a detritus, detrite. Then we have leaching, in which the particular inorganic salts are precipitated and it is unavailable. Then we have catabolism. In catabolism, a lot of fungus and bacteria and enzymes play a role in breaking down. Then we have humification, in which there is formation of humus. Yes, And the characters of humus are very important. That is, it is colloidal in nature, a lot of insecticidal bacterial, against bacteria it is very persistent. Then what about, it is rich in nutrients and decomposition rate is very slow. All those points are important here. Clear? Then we have mineralization, in which the humus is degraded into mineral formation, which is taken up by the plants later on. Taken up by plants later on. Then we have something called as 10% law. What is 10% law? That is, when we go from one tropic level to the next tropic level, 10% of energy is being lost as respiration. Lindeman's law or 10% law. Clear? Then we have food chain. Now what is the food chain? Food chain is basically a order, unidirectional order, in which one organism is eaten by other organism. Then what is the food web then? Food web is very simple. That is, when two food chains combine, that is, grazing food chain is crossing over with, it is like a Power Rangers crossing over. That is, grazing food chain is crossing over with detritus food chain. When they cross over, that is called as your food web. That is called as food web. Then we have the tropic levels, all of you know. That is, a specific place occupied by an organism in a tropic level is in a food chain is called as tropic level. That is, specific place occupied by organism in a food chain is called as a tropic level. The last part of this chapter where the questions are going to come, definitely, if they don't come from this part here, see, they might come from this, see, main question for boards will come from either here, one more question from here, one more question from here, or they can get you one more question from here. But five more questions is definitely from this. 5 more question is going to be from decomposition, or a long question can be coming from the pyramids. Now pyramids can also be giving you 1 mark also, very important pyramids. That is, pyramid of number, first one. Pyramid of number is upright in the case of your grassland ecosystem. Pyramid is upright in the case of grassland ecosystem, but pyramid is again, number is inverted in the case of a large tree. In the case of a large tree, a tree ecosystem, it is inverted number. Clear? Then we have pyramid of biomass. Again, pyramid of biomass can be upright. Pyramid of biomass can be upright in the case of your grassland ecosystem. But same pyramid of biomass can also be inverted in the case of your aquatic ecosystem. That is, there are more number of, the biomass of zooplankton is higher compared to the biomass of the phytoplanktons. Clear? Then we have the pyramid of energy. Now remember, pyramid of energy is always and always upright. Pyramid of energy is always upright. It is always unidirectional. It is always upright and it is always unidirectional. And students, remember, in pyramid of energy, when we go from one tropic level to the next tropic level, some amount of energy is being lost as respiration. That is also your 10% law. That is also your 10% law. You notice we finished the entire chapter, students. In just this part is removed. Yeah, it's removed, right? Ecological succession has been removed. No. Clear, students? That is the end of the. Yeah. Yeah. What do you think? Ecology is.
Baby.
Very good, it is. Next is microbes. After microbes, we'll be doing biodiversity conservation. Last chapter.
Microbes will finish in 10 minutes.
I know, of course.
The thing is like, you sit here only. Yes, students? Not tired, right? Not tired, right? We are tired, but you are not tired, I hope. Yes.
Well, syllabus delivered for 1 year, we're doing it in 5 hours.
Yes, we are doing it 1 year syllabus, we are doing it in 5 hours. Okay? Yes. One second, students. Yes, one. Sorry students. Sorry students, some emergency. Okay. Yes, we are continuing. Microbes will not take us any time, right? Easy peasy, lemon squeezy is who? Microbes. Okay, chalo, ready? Some fire emojis. Yeah, my energy is draining. You people are not giving me any energy. You are like the glucose that I need now. So, put energy fast. Fast, fast. Yeah, this we will do. First we'll do this. Okay. Okay, students, we'll start with industry, microbes in industry, then we will go that side, then we'll come this side, okay? That side, this side? Yes? Why can't I see any fire emojis? Don't want me to take? Okay, look here. So, microbes in industry. So, what is happening? See, microbes are not always useless. There are useful microbes as well. And who taught us this? NCERT, okay? The God NCERT taught us that microbes are useful as well, and microbes are used in many firms and they are of lot of help to us, and we do not know. We always think that microbes, oh my God, causes me some kind of infection, but that's not the truth. So look here, we have fermented beverages. We are using Saccharomyces cerevisiae, that is your yeast for fermentation, right? Your beer, whiskey, rum, all of this alcohol percentages keep changing. One thing to remember, if the percentage of alcohol increases 13%, more than 13%, the yeast will commit suicide, right? Yeast will die, okay? So that's why sometimes we use your distillated drinks. Now, look here, then we have antibiotic penicillium, which is Penicillium notatum. This was also by fluke found. That is actually when the scientist was working on a petri dish, they noticed that in one portion there is no growth at all. Okay, the bacteria are not growing, and they realized that this is something that can actually eat the bacteria. That is when they came to a recognition that there is something called as Penicillium notatum, which is actually able to resist our antigens. Okay, that's how it became an antibiotic. Then we have organic acids. Students, very, very important. Yesterday also you would have seen 1 mark question. Match the following question from this. So, how you have to memorize this, okay? So, Aspergillus niger, okay, that is a fungus which will release citric acid, okay, forms citric acid. Then we have Acetobacter aceti, which will cause acetic acid, or you know, common name is vinegar. Okay, acetic acid. Okay, now Clostridium buticulum, that is your butyric acid. Okay, buticulum, butyric acid. Connect like that. Only this Aspergillus niger, you might have a confusion, like you have to think citric acid, right? That will not come very fast to your mind, but other than that, everything else will come. Because Acetobacter aceti, acetic acid. Clostridium butylatum, butyric acid. Okay, then Lactobacillus, lactic acid. This is the one that is present in milk and curd. So that is the reason why your mom adds some amount of inoculum in the boiled milk, so that next day it will become curd. Right, next day it will become curd. Fine. Now look here, there are some enzymes also which are actually helping us, like lipases, which will actually help us to remove the oil, right, which has a detergent effect, removes the oil. Then we have pectinase and protease. So this is lipases which removes fats, okay, removes fats. Now pectinase and protease to clarify bottle juices. If you notice, apple juice is never, students, I don't know how apple juice or your appy fizz is called as apple juice, but that's not the truth, because apple juice will be so whitish creamy color if you just normally grind the apple, but we always get very clarified juices from you know, like packed ones from our supermarkets. How is that possible? Because of the presence of pectinase and protease. They consume the pectin and protein from the juices to give us a clarified juice. Then we have streptin kinase, that is streptococcus, used as a clot buster. Okay, we use this as a clot buster where during surgeries, during any kind of like you know, when we are doing any kind of procedures, we need to make sure, and in blood banks these are used to avoid any kind of clot, okay, to avoid any kind of clot. Now bioactive molecules, very important, that is your cyclosporin A, which is used, cyclosporin A is used during graft rejection, that means as an immunosuppressant. Okay, it is used as an immunosuppressant. It is used as an immunosuppressant. Now, statins are Monoscus purpureus, which is used as a blood cholesterol lowering agent. Okay, purpuris, Monoscus purpuris is used as a blood cholesterol lowering agent. Suddenly a person comes with heart attack or a person got myocardial infarction. Okay. So doctor will give first your statins or your Monoscus purpuris, which is used as a blood cholesterol lowering agent, because the doctor understood that this person has a lot of cholesterol and that bad cholesterol led to the cause of heart attack or reduction of your lumen or thickening of your depositions on the blood vessels. Okay, got it? Now cyclosporin A, which is released by trichoderma. Trichoderma is a very important concept. Why, students? Because this question, they will mix it up and give. Trichoderma polysporum, which is act as an immunosuppressive drug. That means during any graft or any organ transplantation, our body should not reject it, right? During that time, we give cyclosporin A until the entire procedure gets over. So that will be like our body will not feel it like a foreign particle, but instead it will feel like, okay, it's my cells and not damage it. Okay. Then here, okay, microbes in your biofertilizers. We are only studying this one, scientific ones. So here, first we have bacteria, that is rhizobium, which are symbiotic bacteria. That means they are doing a very happy relationship with wherever they are. That means what? They are converting your atmospheric nitrogen into nitrates, fixing it in the soil and increasing the soil fertility. Now this way what will happen? Soil fertility will increase and the crops will grow better. Right? And here we have azospirillum and azotobacter, which are again free living bacteria, but they are of lot of help. Even we have Anabina, Oscillatoria, Nostoc, Professor will teach. Okay, all these are also of help. Okay, all these are also of help. Then we have fungi. We always think about the job. Also students, they are present in higher plants. They also do this nitrogen fixation. Okay, now they are associated with glomus with plants. Okay, so remember glomus word, they'll do match the following and give again. You should not be going wrong in that. Now symbiosis, I told you, Anabina and Azola. Free living, we have Nostoc, Oscillatoria, that is given under cyanobacteria, but no problem, you can write it under bacteria only as a general thing, no problem. Okay, fine. Then microbes in household, this was supposed to come first, but no problem. Now curd, I told you, there is an inoculum which is added by your mother without even knowing. Your mother just adds 1 spoon of curd into the boiled milk without even knowing that she is doing inoculum. Okay, if you tell her, Mama, are you adding inoculum? She'll throw the spoon on you. Okay, but it is just that scientifically they do not know what happens, but they definitely know that tomorrow morning it will become curd. That is because of the presence of lactobacillus. Okay, then bread, again yeast is there. So baker's yeast is there. Okay, baker's yeast is there, which is your Saccharomyces cerevisiae. Then toddy, which is from palm wine. In Kerala and all, you will see, a lot of toddy, that is from Caryota, okay, palm. This is the type of palm in which we collect toddy from. Okay, then Swiss cheese. Swiss cheese, the holes in the Swiss cheese, the texture, all this keeps varying depending on which type of person, like which type of microbe is there. So here in your textbook you have Propionibacterium Shermani, shyful bacteria, microbes. Okay, now microbes as biocontrol agents. Just now biotechnology started, Bacillus thuringiensis, which is your BT cotton. So this way what we are doing is, students, we are reducing the amount of fertilizers and pesticides and weedicides in the plant, that because we are ultimately we are consuming it. We are the ones who are doing. So because of anthropogenic activities, we end up getting cancer, multiple diseases. So to avoid that, yes, because of the carbon dioxide production is by which the holes are formed on the cheese. Okay, Bacillus thuringiensis, which control the butterfly caterpillars. What do? They go to the larva of this caterpillars and they kill the caterpillars. Okay, they release chemicals and they kill this caterpillar, but they are very specific. They will not kill any other insects. They will only attack caterpillars. Okay, same thing goes with Trichoderma species and your baculovirus. Especially genus Nucleopolyhedrovirus, which controls insects and arthropod. The question can come, which is the one that controls the insect? Your answer should be baculovirus. Okay, fine. Then the last topic of microbes is your sewage. Okay. student sewage, basically 2 steps happen, your primary and biological or primary and secondary. Anyways, you can tell. Primary means I'm basically doing sequential filtration and sedimentation. I'm just like, there's a lot of paper, cigarettes, unnecessary things. I just need to filter all this physical, you know, bigger particles separately. For that, first I'll do sequential filtration. I will use sieves of different sizes and filter it out. How you have this to filter your chai patti or your tea leaf. What do you tell for Kannada, in Kannada? Tea leaf only, no? Yele. Tea yele. Chai, So all this, to filter all this, we use different sizes of sieves, no? Like that we keep filtering, that is called as sequential filtration. At this end of this filtration, all the larger particles would have got filtered. Next we do sedimentation. In sedimentation, we'll be able to get a primary sludge. That primary sludge will be later going to your biological treatment, where we use your bacteria. We call that as flocks, right? We call it as flocks. Now, what do? They do, so by the percentage of BOD and DO level, we will be able to understand how much organic matter is there in this water. Okay, so if the biological oxygen demand is more, okay, and dissolved oxygen is less, that means we have a lot of bacteria in them, lot of, you know, organic content in them. Okay, so BOD has to reduce and DO has to increase. Now, secondary sewage treatment involves the action of microbes. This is the most important thing. These microbes will be sent back again to the aeration tanks. Some of them will be put into your anaerobic digester, anaerobic bacterial digester, where this will be consumed. Okay, they will be completely destroyed. Some amount will go again back as a to use for your biological treatment again. So please check out that cycle from my chapter. I've given the notes there. Okay, then last we have biogas. This we explained yesterday also. Now, microbes in production of biogas, we have your methanobacterium. Okay. Now, methanogens are present in the ruminant, in the gut of the ruminant cattle, okay, cows, and my brain stopped braining, students. So, yes, okay, ruminant cattle. So, what do? They actually help. This cow dung and water will mix together to help in the formation of biogas. These biogases are used by, you know, village people, even some industries, to actually produce the amount of fuel. Okay. Yes. So, we have methane, carbon dioxide, water, sulfide, hydrogen sulfide, and much more. Okay, much more. Any doubts from this chapter? Let's see if we finish the whole chapter.
Really good, ma'am.
Yeah, everything is there. You do not even have to sit and write. Look here, everything is written. Bread, toddy, Swiss cheese. Sir, this kind of notes, no, is so difficult to get. Everything is written, and this is a revision. So, this is how much they should know. They don't need to know the unnecessary part. Because see, secondary sewage treatment, if they would have attended my class, they would have understood the detail. Now, other than that, they only need to know these names. So, students, everything is there, okay? All your job is to now sit and study. And we are coming to the end of this session. That is one more chapter. And I appreciate ourselves and I appreciate the students who stayed from the starting.
I know,
especially,
they were, especially
we,
from starting, because it's not easy to attend entire class 12 biology. Yes. So we had different plans in the mind. How will we finish the entire biology, ma'am? So then we multiple plans. Like, how do we finish the entire biology in one video?
telling what tomorrow?
We'll tell them.
Yeah, shouldn't we?
Okay, students. So your exam is close by, and exam is day after tomorrow, and we thought that students, some of your students are studying in the morning. Yes, some of the students who are studying in the morning, we have a special surprise for you. That is tomorrow, 5 a.m. Tomorrow, 5 a.m. we'll be taking the class. That's right. So we are waking up here early in the morning. We are waking early up in the morning and coming to the studio. Traveling and coming to the studio. We would want you to also sit with us in the morning, 5 a.m. and revise one small revision. Last minute revision.
After no more classes.
After no more classes. So we thought we will do this on day after tomorrow. But we are like, no. One day before exam, they should have that. Mental peace, no?
Also, there are a lot of PU students who came to us and asked for the classes. So, I think it is favoring both of them.
Both of them. So, it's going to be very useful.
Excited?
We are excited.
Yeah, we are excited because we have to wake up in them. What about them?
I don't love waking up early.
They're not excited.
It's fine.
Okay, bye students.
Students, now we'll be starting off with the last chapter of your ecology and your class 12 syllabus. The last chapter for your class 12 syllabus is nothing but your biodiversity and conservation. So can we start? Biodiversity and conservation. Students, this chapter, I'll tell you exactly what to study here. I'll tell you exactly, yes, it will be tomorrow. Exactly tomorrow 5 a.m. live on Vedantu Neet English channel. We'll be coming live and we'll be discussing the entire, let's say it's like a small last checkpoint for all of you. Before you go to the examination, that is going to be the last checkpoint from our side, because we are making, we did the diagrams, we finished we did all the, you don't want if you keep it, I'll read it. We did all the diagrams is done. We did most important questions from your every single chapter. We also taught you the entire chapter in the mere 5 hours. So that is our promise. So last checkpoint we would like to do from our side is going to be tomorrow morning. We'll be coming live and we'll be telling you a small, it's going to be a 1 or 2 hour session, not more than 1 hour. So we'll be coming live tomorrow and we'll be doing the entire syllabus. One small something is there, small twist is there. So you'll know tomorrow morning at 5 am. Okay. So can we start, students? Can we start biodiversity and conservation? Show some energy in the chat, because students, we have been doing from past 5 hours and I want everyone to be attentive now. Everyone, be attentive now. Students, if you read your NCERT, right now, the entire biodiversity can be grouped, can be studied in 3 different levels. That is, levels of biodiversity. There are 3 different levels of biodiversity. The first level of biodiversity is the genetic level. For example, students, we have the best example is your Rafflesia. Yes, Rafflesia vomitaria, which is the type of your flower, has many different varieties. Yes, genetic diversity. Similarly, we have many different genetic variety of rice. We have many different genetic variety of mango. Yes, that is the genetic variety. Then we have something called as diversity at species level. Yes, diversity at species level. At diverse species level, the example which is given in your NCERT is that, see students, if you see the India map, if you look at the India map, if you look at the bottom part of the India map, yes, this is called as your Eastern Ghats. Eastern Ghats. This part where the Karnataka everything is there, it's called as Western Ghats. Western Ghats. The amphibian. You know what is amphibian? All of you know what is amphibian. The amphibian biodiversity is much more higher in the case of your Western Ghats compared to your Eastern Ghats. So what is it telling you? The species of biodiversity is changing based on the location also, and the example is your amphibian population. That is, amphibian population is higher in your Western Ghats compared to Eastern Ghats. The last one is your diversity at the ecosystem level. That is, at the ecological level. Example, India. Look at India. If you look at the top of India, we have tundra. Yes, much more colder climates. If you go to the Rajasthan, we have desert. If you go to Arunachal Pradesh, we have tropical forest, right? So every single place, if you come to the south side, we have a lot of your mangroves, coral reefs are present. Yes. So the India is such amazing diversity compared to a Scandinavian country like your Scotland and everything, which has less diversity. That is your diversity at ecosystem level. Diversity at ecosystem level. Clear? Now let's learn about your, where has it happened? Global species diversity. That is, how is the distribution of species toward entirety of your globe? That is, if I ask you a question, I asked you yesterday also, which is the phylum which is largest in nature? That is going to be your insects or arthropoda. So again, can you see? Insects are the highest. Second is going to be your mollusca. Mollusca is highest. What about the rest of them? Rest of them are combined here. Then we have separate crustaceans. Crustaceans are separate here. And if you look at in the case of your vertebrates, all of you know the entire earth is covered by ocean. The 3/4 of earth is covered by ocean. So obviously, the population of fishes will be much more higher compared to mammals, birds and reptiles and amphibian. The last diagram is also very simple to remember. That is, kingdom fungi is separate, kingdom plantae is separate. So hence you can see the entire population of fungi is comparatively larger, right? And we also can see Angiosperms is much more higher. That is the reason. The reason behind it, the current era is the current atmosphere is predominantly dominant for your angiosperm development compared to gymnosperm. Gymnosperm was before. Gymnosperm era is over. That was gymnosperm was highly developing in the previous era, but not right now. That is your entire, that is global species diversity. Now students, the next thing which you need to understand is pattern of biodiversity. Here question can come. Here question can come here. That is, what are the different pattern of biodiversity? The first pattern here is longitudinal gradient, so latitudinal gradient. The other one is your latitudinal gradient. Latitudinal gradient is very simple. Latitudinal gradient is basically telling you, if this is the earth and middle one is equator, as we move away from the equator, the species diversity is decreasing. Clear? If they ask you a question, where do you find the highest species diversity? The highest species diversity is found at the equator. Apart from equator, Tropic of Cancer as well as Tropic of Capricorn. Those are the places in which maximum biodiversity can be achieved or seen. Why? The answer is very simple. Again, you should ask why. Why, sir? Why near the equator? The answer is very simple, students. In the equator as well as your tropical Cancer and tropical of Capricorn, there is more sunlight is coming there. If there is more sunlight, more plants are able to do GPP, more NPP, more biodiversity. Simple. More productivity is there. That is, species diversity decrease from the equator towards the pole. Tropics, that is latitude ranging from 23.5 north to 20 south, have a more species diversity than temperate or polar. This is basically the trying to tell you here is, all the trying to tell you here is, this 23 north is your Tropic of Cancer, 23.5 is Tropic of, that is Tropic of Capricorn. In Tropic of Cancer and Tropic of Capricorn, they have more species diversity compared to the temperate and polar regions, because in temperate and polar regions the species diversity is going to be the least. There. Remember that point. The next one is your species area relationship. Very simple. I explained yesterday. Species area relationship tells you that the more you search, more biodiversity you will find, but only up to a certain limit. It was given by whom? The great Alexander von Humboldt. He told that the more area you search, more species you will find, but only up to a certain limit. Yes. And if you plot it in a graph, you get a rectangular hyperbola. What do you obtain? If you plot species and area relationship, you will get a rectangular hyperbola. The more you search, the more area you search, the more species you get, but only up to a certain limit. This is the limit. Clear? And it is given by the equation, that is, if your logarithmic equation is log s is equal to log c plus z log a. Log s is the species richness, log c is going to be your y-intercept, then z is going to be the slope of the equation, and a is going to be the area. Very important, remember that. Clear? Species richness increases. The more you the more area you search, more species richness increases, but only up to a certain limit. That is very important line. The next one is your loss of biodiversity. See, log is equal to c plus z log a. The next one is your causes of biodiversity loss. What are the reasons for biodiversity loss? Diversity loss is happening. That is, the first one is your habitat loss and fragmentation. Now they are called as evil quadrate. They are called as what? Evil quadrate. The four main reasons for biodiversity loss. The first one here is called as what? Habitat loss and fragmentation. In habitat loss and fragmentation, we mainly notice what? The example given here is the Amazon forest. That is, Amazon forest. Before it was around 12% of entire earth. Now it is just 6% of the entire earth. The decrease in the Amazon rainforest. The next one is called as over-exploitation. That is, when need turns to greed. That is, more and more you exploit, more species are going to be destroyed. Example is your passenger pigeon and stellar sea cow. Examples are very important. That's what they're going to ask you. The next one is going to be alien species invasion. That is, you take a foreign organism and introduce into our native one. Example is Nile perch, that is introduced in your Lake Victoria, which almost killed around 200 species of your cichlid fish or keyclid fish. The other example is your lantana camera, Parthenium, water hyacinth, and also the catfish. All of these are different examples of your alien species invasion. Then we have the co-extinction. Co-extinction is very beautiful. That is, if a parasite is dependent on an organism, if the organism is dying, can the parasite survive on its own? No. So if the host is being extinct, the parasite is also becoming extinct. The other example is your wasp and the fig fruit. Yes, they are in mutualistic relationship. If the fig is gone, even this case, there is a possibility that if the fig is gone, there is a possibility that even the wasp is also gone. So both can become extinct at the same time. That is also like how one pollinator and plant has a mutual relationship. If both are getting one get extinct, the other also will get extinct. Now, this is your habitat loss and everything. But what are we doing to conserve it? Now, before we go, how are we conserving it? Let's understand why are we conserving it? Why should we conserve nature? Why can't we just leave it as such? There are three main reasons for conserving nature. The first one is your human need. That is also called as your narrow utilitarian. Narrow utilitarian is the human need. That is for our food, medicine, timber. So all the things which we need from forest, that is why we need to save the ecosystem. That is, more than 25% of the drugs are derived from the plants, and not only drugs, lot other things are derived from the plants. So we need to save them for ourselves. First reason. The second one is going to be your broad utilitarian. That is, obviously students, if you remove the forest, oxygen less, that is, provides oxygen, pollination will reduce, aesthetic pleasure, all the other reasons for your broad utilitarian. Clear? Then we have the ethical issue. That is, earth is not only ours, that is, earth actually belongs to all the other organisms. Can we be so selfish that we completely destroy the earth? No. That is, moral duty to take care of the biodiversity. Moral to take care of the biodiversity. Those are the three main reasons why we need to conserve the biodiversity. The last part of the chapter is how are we conserving it? There are two methods, very favorite question for your NEET as well as boards, that is the conservation methods, that is in situ as well as ex situ. Now what is in situ? In the case of in situ, we are conserving the organism within its natural habitat. Within its natural habitat, we are conserving it, right? That is national parks, sanctuaries, biosphere reserves, sacred forest and hotspots. These are the main reasons for doing that. These are the different types of in situ conservation. Now here one question comes, what is the meaning of endemic? Endemic, that we learn in your biodiversity hotspot. Endemic organisms are the organisms which are found only in one location, not anywhere else. They're endemic to only that spot. And also there is a question called as, what is something called as keystone species? Keystone species are the species which are integral part of your nature. That is, if the keystone species has been removed, the entire ecosystem will collapse. Example, elephant, honeybees, your banyan tree, all of these are keystone species. Clear? Then we have ex situ conservation. That is, in ex situ conservation, we are taking an organism out of its natural habitat and keeping it in an enclosure. Examples is going to be your zoological park, botanical garden, gene bank and also genetic resource centers. There we basically do nitrogen, we use liquid nitrogen to preserve different gametes, different seed bank and everything. All that is ex situ conservation. All that is ex situ conservation. Students, with that, the entire class 12th biology is done and dusted. Entire class 12th biology is done and dusted. Do you realize that? What you study in the entire year is done in 5.5 hours. And if you remove the break time, almost 5 hours. Within 5 hours, the entire class 12th biology is over. And students, if you want to revise the entire biology, you can come back to this video. But before I go, let me tell you something. And students, if you are from Pune, let me bring you on spot. If you go to the description of this video, that is, if you go to the description of this video, if you are from any of the centers, in which Vedantu is currently making offline centers, you can go, there's a link here, you need to use your email ID and login. Use your email ID and login here. Once you log in, even if you're from any of the places which is Vedantu centers near you, you can enroll, have a visit before you take an admission. You can have a go visit there, go visit the place and see how technology as well as education is mixing up. See, this is technology, but here this is offline. How are we implementing technology in, this is online. How are we implementing technology in offline? That you will only know if you go and visit at the Vedantu Learning Center. If you go to the Learning Center, you will notice that the entire atmosphere is so different that we have a doubt counter, we have a smart board is there, we have smart clickers, every single thing is so advanced there. Then we have something called as your, this is the offline center, oh it's there. I don't know why this is the one page is there for this, it should have come as such. And students, if you go to Telegram channel, right now, you will be able to download the entire PDF. The entire PDF of today's class will be uploaded on the Telegram channel here. On this Telegram channel right here, if you want to download. And also you can join my Telegram channel, that is Baswaraj Sir Biology, where I exclusively post some other content, where I talk about different extra things and I'll provide other information, okay, biology content. Only if you want to contact me. If you want to contact me, you can join my Telegram channel. The last thing is your crash course, which I've been telling you, students, Crash 5.0. It's 4.0, you know why it became 5.0 right now. If you have seen, see here, I don't think it's given here. Oh, it's not given here, students. See, the price is given here. Before, this course was around 5,000 rupees. Then we made the course for 4,000 rupees. Right now the course is for around 2,000 rupees. And this is going to be for NEET students. This is going to be for only and only NEET students. Just 2,000 rupees. The entire course is going to be there, where they'll be doing entire syllabus in a mere next 45 days. In the next 45 days, the entire syllabus will be over. So go join the crash course right now, and I'll see you in the next session, students. Take care. Bye bye, all of you.